And in my project, I mainly gathered leafy liverworts and mosses. Bryophytes have a lot of ecological roles that pertain to their water retention. First off, they have evaporative cooling effects in the ecosystem. They provide a lot of erosion resistance by slowly releasing the water that they absorb, which would otherwise be lost as surface runoff. They maintain humidity through their evaporation of the water they retain. They provide microhabitats to microorganisms such as tardigrades and protozoa, as well as hosting a lot of arthropods and their larvae. And finally, they contribute to nutrient cycles both with their own organic matter and through the water that flows through them, catching nutrients along the way to the forest floor. My project has a lot to do with the changing hydrology of Monteverde's cloud forest. Studies have shown that climate change is both extending the duration and amount of dry periods in the cloud forest, as well as raising the altitude of the orographic cloud bank, which is characteristic of the cloud forest. Additionally, it's increasing the quantity of rainfall and the intensity of downpours. Deforestation also has a large effect on the cloud forest. It increases surface heating and moisture content, which in general leads to thinner, higher, and shorter-lived clouds. So with all of this in mind, I had two main research questions. First, do bryophytes capture different forms and quantities of precipitation in pasture versus forest? So this is looking at, do the bryophytes in the forest, for example, capture more horizontal precipitation than in the pasture? And to give context, horizontal precipitation means fog and mist. And my second question is, how does bryophyte water retention vary in different precipitation conditions? Do bryophytes retain or capture more horizontal precipitation than vertical precipitation or vice versa? So on to materials and methods. I had 4 sites, 2 in the forest of the Campbell farm and 2 in the pasture of the Gwindon farm. I set up horizontal precipitation collectors at all 4 sites and 1 rain gauge in an area where the pasture met the forest. I went to these sites every day and collected a sample of bryophyte from each site and took the precipitation measurements from each apparatus. These are some photos from my forest sites. One of my forest sites was more wind protected and had more tree density than the other. And these are some photos from my pasture sites, which both had pretty sparse tree density and lots of sun exposure. Here are my precipitation apparatuses. On the left is my rain gauge, where vertical precipitation would fall into the funnel. On the right is my horizontal precipitation catcher. And the way it would work is fog and mist would condense on the plastic strings, flow through the funnel, and end in the collection bottle. Each day, I would pour these amounts of water into a graduated cylinder to measure how much precipitation they had caught. I would take my samples back to the Monteverde Institute to take their wet weights, put them in the plant dryer until they were dry, take their dry weights, and then calculate overall or general sample water content of the original sample. All right, so on to my results and discussion. So this figure is basically a visual aid to show how at all sites, F1, F2, my forest sites, P1, P2, my pasture sites, over the collection period, the x-axis, their sample water contents were following similar trajectories. So you can see how they all sort of dip at the same time, dip again at the same time. You can also see that the forest samples, which are the green lines, were always wetter than the pasture samples. This figure shows the horizontal precipitation collection values over the sampling period for all 4 sites. The y-axis is water collected in the horizontal precipitation collector in milliliters. All 4 of these horizontal precipitation collection trends were relatively uncorrelated using a Pearson's R correlation coefficient, except for the 2 relationships that I have on the screen, which F2 and P2 had a strong correlation, P1 and P2 had a moderate correlation. But overall, all of the horizontal precipitation collection trends were uncorrelated. So what does this mean? This suggests that microclimates at each site were heavily influential on the horizontal precipitation conditions. So this might mean that on a given day, the fog and mist that are passing through site F1 are not the same quantities as the fog and mist passing through site 2. This figure is a little more complicated, but I'll explain it to you. This graph is only focusing on site F1. The green line shows F1 sample water content, and you can read it using the y-axis on the left. For example, you can see on November 13th, the sample water content started around 80%, and on November 19th, it dips down to around 20%. The other 3 lines represent precipitation measurements, and they can be read using the y-axis on the right. For example, the purple line is total precipitation, and you can see that it starts around 180 milliliters on November 13th, and then on November 16th, it goes down to around 40 milliliters. The R correlation coefficients show the relationship between each precipitation measurement and the sample water content. And you can see that the relationships between them are all ranging from moderate to strong. This is the exact same type of graph, but at one of my pasture sites, site P2. You can see that the correlation coefficients are all very weak. So what do these figures show us? First of all, forest samples were always wetter than pasture samples. Second of all, forest sample water content had much stronger correlations with all precipitation measurements than the pasture sample water content. This suggests that pasture bryophytes were generally more affected by other climatic variables such as wind speed, wind force, sunlight, temperature, maybe faster wetting and drying cycles caused by faster wind, and the moss species present. This is very supported by previous research, which has shown that intact forest in cloud forests tend to absorb the momentum and speed of wind passing through, which allows for lingering humidity and more moisture in epiphytes in general. This is very relevant to bryophyte viability, because bryophytes can only sexually reproduce when they're fully saturated in water, because the sperm needs to travel through water to reach the archegonium, which is the bryophyte female sexual organ. It's also important to mention that precipitation types were not temporally isolated in my project. There were times when rain events would overlap with fog events in the same 24 hours, making it difficult to show how bryophytes at these sites interacted differently with the different types of precipitation. There is previous research which has shown that horizontal precipitation is generally more valuable towards bryophytes in performing their ecosystem services, because it can penetrate into deeper layers of the moss rather than, or the bryophyte, rather than on more superficial layers. In the future, I would like to do more temporally isolated experiments to see how these bryophytes interact differently with the different types of precipitation. And this will provide really important insight into how bryophytes are going to respond to the rising orographic cloud bank, how their reproductive success will be affected if they're being less deeply saturated by horizontal precipitation if it's higher, or whether they could be useful towards climate resilience when there is less horizontal precipitation, depending on the results of how well they retain these different precipitation types. So in conclusion, forest bryophyte moisture was correlated with precipitation patterns, whereas pasture bryophytes were much more affected by other climatic variables such as wind and sunlight. It's important to do temporally isolated research on how bryophytes interact with the different precipitation types in the cloud forest, because this will provide very important insight into how they will respond to climate change and the changing hydrology of the cloud forest in Monteverde. I would like to thank Naomi Gwindon, my primary advisor, for all her reassurance and expertise, and Frank for all his thoughtful feedback. Thank you to the Gwendon and Campbell Farms for allowing me to do my project on their properties, which were so beautiful and a pleasure to go to every day. And to my host family, the Ugalde Rodriguez family, thank you so much for opening your home to me. And yes, thank you. Any questions? Yes, Kaylin.
You mentioned on one of your slides climate change and dry periods in Costa Rica, and then I think on one of your other slides, all the moisture gets done like November 19th or something. I thought that might have been another dry period, but do you see anything with dry periods in your data that may have applications for like health or whatever?
Yeah, so what I didn't include in my presentation was a smaller part of my experiment where I I examined the absolute water content capacities of morphospecies in the pasture versus the forest. And that was a really interesting part of my experiment because I saw that the average absolute water content ability of morphospecies in the pasture was higher than the absolute water content abilities of the morphospecies in the forest. I wasn't able to get statistically significant data on this. However, I think it's a really interesting path that I or someone else could go farther down to examine whether bryophytes in pasture areas or deforested areas or drier areas in general have been selected for drought-resistant adaptations, such as more leaves on moss shoots or deeper pockets where they can keep the water, because there has been previous research on the mosses that are able to proliferate in drought conditions, because they tend to be able to retain water for longer, which allows for more of their sexual reproduction and general proliferation.
Thank you, Ruby.
You can come here. A little bit.
I like this bit.
Thank you. Preach.
Sorry for the extra suspense.
No worries.
What's the Monteverde Institute? Spotify? Isn't it free? I don't know. Wait, hold on. Capital?
Monte with capital 2020
Monte dot
2020.
20
Monte 2020
dollar $10 then?
Trying to nerf me.
No, HDMI. On this side.
But everyone, I could tell everyone had a lot of questions.
So how you chose which species to
I just like stopped it. There was no. That's why I took it out, because it was like the unscientific part of my experiment. I just like grabbed things that I thought was cool. Well, I tried to make sure I was getting it. I would have had
Oh no.
Sorry for the technical difficulties. We're figuring out what it is. It could be the cable.
So we didn't hear one?
I want to know what grade was that. You give me an A?
Alice, can you hand me my charger?
Wow. Thank you guys. Thank you.
I'm the one right for it. So I just did this.
go out to play with it.
That's a good idea. I brought it.
¿Quieres que cambiemos a esto? Voy a quitar esto.
Oh yeah, place shirt. Okay. Okay, what am I doing?
You have lice.
What? I was doing UN from
Quitarme lo que sí. Por la pantalla. Pero no, se hace. Que intentamos cambiar el ahora sí. No, ¿verdad? Mételo está logrando, mételo está logrando. It's the connection with the word. I don't think so. Nacho, cuidado de pega, no tengo poderes para poder ver su máquina. Primero que funcionó la primera vez. ¿Cómo? ¿Todo va bien? Sí. Falta un ganchito. Does anyone know there's a little hook?
Oh, I was gonna say, there's a piece.
I'll just hold it like this. Yeah. We need some zip ties. Or a piece of coriander. Leo. Necesitamos una mesa. Una mesa. Creo. Fui a buscarlo a llegar a ver esto y aquí se ve. Y cuando llegué, no está. Pero ya se ve. ¿Qué? Yo fui a buscar dónde estaba. No tiene. No, sí, claro, usted no está viendo. Usted lo mismo sí visto, recientemente. ¿No será el pantalón? No. Quitarlo y apagar esto y todo. Yo no sé si es una oficina. Tiene que jugar, hay que poner
Pero igual es.
si ponemos el pie aquí arriba, no sostiene. Pero suavecito, ok. Estaba pensando en eso.
A mí, voy a recibir esto para no dañar el tabaco.
¿Cuál tabacón? Ah, yo creí que el piano. Creí que el piano se llamaba tabacón. Ya, olvídenlo. Ya, va a ser como Navidad. No tiene proyector de acetato. Proyector de acetato, no tiene. ¿Es el que ustedes? No, el instituto está en Estados Unidos, ¿verdad? A ver, ¿dónde está? Los controles están adentro. Préstame eso, por favor. Gracias. Frank, también tenemos los papeles. Sí, del pastel. El super pero tenemos cables de ese. Hay cables de sobre. Sí, pero ya. ¿Ah? No, pero ahora estamos buscando. Pero no es la mano que te pasa, ¿no? Sí, el problema es de cable muchas cosas. Si lo metes de extra, los números van a cambiar. Eso es suyo. Está $20,000. Donación al instituto. Qué bueno. Usted sigue siendo negocio. ¿Así o allá? Póngalo por favor. Claro, se nota tu claro, es un lente. Ah, ya. Pero ha tenido problemas con el tele, simplemente se fue. Qué bueno. Afuera sí. Pero no está conectado a la parte de roots. Ah. No, pero no tienen proyector de acetato. Ellos tienen sus proyectores de acetato. Ah, no, it's cool, no. No lo sabe el arte, ¿verdad? Do I put your presentation in photos?
Yeah, everyone did that on the same day. I drew all my pictures. I was thinking, it would be so
Eso es todo. No, pero está bien. Bueno, pero está mejor que antes. Porque antes no había. Can you click on the next extra muscles?
It's this one.
¿Qué pasó? So,
Es porque está.
ahora,
Porque está compartido. Sí, sí, sí.
ese.
Sí, ese es.
No tiene sentido. Es que eso va a brincar, va a ver.
¿Usted prefiere hacerlo usted? No. Es que Jaime me está diciendo que prefiere. Pero Jaime ha hecho actividad.
O sea, yo lo puedo hacer bien, a mí me parece bien, pero a mí es demasiado. Porque yo sí llevo el tiempo y yo estoy viendo a Alex.
La cagada mía porque yo estaba pensando que eran 12 de...
Sí, usted me dijo que eran 3 minutos, y ¿no?
Y yo súper seguro.
Do you have the thingy?
Yeah.
Do you want to see if it works? No, not that. I mean, yes, keep it there. That's good.
Yeah, it works.
It works? Good. Okay, I know.
Thank you.
Ya sé que ya.
Schläge, Schläge.
Es que ahora tengo que hacer como buffer sound. What? Yeah, it's a
The boy?
little dangerous now, but okay. Thank you everyone for your patience. As you can see, we can rely on the predictor always. The super smart TV was not so smart today, so we have to rely on the predictor. But we can keep going finally. And yeah, a lot of suspense before this very great following presentation by Tommaso Abrahamson Ward, who's going to teach us about allelopathic effects of triggers. Thank you.
Hi, my name is Tommaso Abramson Ward, and I studied the allelopathy of tree ferns and its effect on the epiphytes on them and on non-tree fern trees. This is a tree fern. They grow to much greater heights than many other kinds of ferns, and they are able to do so because of the structure of a trunk-like structure called a rhizome, surrounded by densely packed roots. This allows it to support its own weight, where other ferns wouldn't be able to. This is also what differentiates it from a regular tree, as it doesn't have secondary or woody growth. Like all ferns, they thrive in high moisture environments, and they grow up to 20 meters tall with a crown of 2 to 4 meters wide. This specific tree fern is of the Cyatheaceae family. These are the scaly tree ferns. And take special note of the structure of the trunk, because this is how you differentiate it from another common tree fern family found in the Monteverde area. This is the Dixoniaceae family, and these are the hairy or spiny tree ferns. These trunks, you can tell the difference because the Cyatheaceae are more gray-brown, thinner, and smoother, and the Dixoniaceae are a darker red-brown and spikier or hairier. Epiphytes are plants growing on usually the trunks of other plants, usually trees. And Monteverde supports a great diversity of these epiphytes, as well as a great diversity of ferns. Tree ferns specifically are a great substrate for epiphytes to grow on. You can find them growing on them very often. However, not all species of epiphytes are able to grow on these tree ferns. And this is due to the allelopathy of the tree ferns. Allelopathy are secondary compounds in tree ferns which prevent the growth of other surrounding plants. The fact that there are epiphytes growing on them, even though they have these secondary compounds, brings up my question: how do species richness and diversity of the epiphytes vary with respect to the tree ferns and the non-tree fern trees? I measured this by choosing tree ferns further from the edge of the forest, further from trails, and further from other tree ferns to avoid the effects of disturbance and reduced epiphytic growth. Once I chose a tree, I measured the diameter using a diameter at breast height measuring tape, as well as a laser distance measure to measure the height. I counted the number of epiphytes, and I measured the percent coverage of plants growing around the trunk. I did this by assigning a rank value of 0 to 20% green plant growth around the trunk of the tree, or around the trunk of the tree fern. And a rank of 1 was 0 to 20%, 2 was 21 to 40%, and so on. I also took field marks and pictures and gave morphospecies to the epiphytic species I found in order to identify the epiphyte families. After taking the measurements on the tree fern, I took the same data for a pear tree. This is a tree of similar diameter close to the tree fern. I found that there was no significant difference between the number of epiphytes on the trees and the tree ferns. However, I did find a significant difference in the ground cover ratings, which were lower for the tree ferns. I also found no significant difference between the species richness and the mean alpha diversity of epiphyte families between tree ferns and non-tree fern trees. But I did find a significant difference for specific epiphyte families between tree ferns and their pairs. This graph shows the difference in species richness of tree ferns and pear trees. I want to highlight 3 families: the Aspleniaceae, the Urticaceae, and the Bromeliaceae families. Aspleniaceae and Urticaceae were found to be much more rich on tree ferns, while bromeliads were found to be much more rich on pear trees. This last column is species I was not able to, epiphytes whose family I was not able to identify. This graph represents the mean alpha diversity of tree ferns versus non-tree fern trees. The same 3 families were shown to be significantly different: Aspleniaceae, Urticaceae, and Bromeliaceae. But I would like to highlight how the Polypodiaceae and the Aspleniaceae, which are 2 epiphytic fern families, were much higher in diversity on tree ferns as opposed to the pear trees. I found that there was no overall difference in epiphytic families between tree ferns and tree fern trees. However, there was a significant difference between the specific epiphytic families of some epiphytic families between tree ferns and tree fern trees. They showed a preference for different substrates, which were the root mantles of the tree ferns. This is the root structure around the rhizome, as opposed to the trunk of the non-tree fern trees. The root mantles are in a much shadier environment and provide much more humidity, as opposed to the more light provided by angiosperms or the pear trees. I also found the difference in the ground cover rating. This is significant because it shows that there is a strong allelopathic effect by the tree ferns. When you consider this with the not significant difference in epiphytic family growth on the trees, you find, or it's very interesting to consider, because the root mantle significantly changes this environment. It provides the ability to store water and nutrients much more at increased levels, as opposed to the trunk of the pear trees. This could account for why the allelopathy doesn't affect the overall growth of epiphytes on the trunks. Further research could focus on the growth of specifically epiphytic ferns on the tree ferns, because these were found in much greater diversity on the root mantles of the tree ferns. I would like to thank Naomi, Fede, Frank, and Leo for all their support and guidance. I would also like to thank Carmen, Marvin, and David for allowing me to use their land to conduct my research. Yeah.
Do you know the mechanism by which like allelopathy, like the allelopathic chemicals are spread? Is it like secreted by the plants or like through the ground?
Yeah, so allelopathy, the mechanisms are different by each plant. Specifically, these tree ferns are, the chemicals come off of them when the water runs on the fronds or the leaves of the tree ferns, and it runs down to the area around. Yeah.
I was wondering if you know of any like medicinal or otherwise uses for the particular compounds made with allelopathy?
That's very interesting. I have not found any of that in my research. The allelopathic compounds are much more related to anti-herbivory compounds, but there may be some medicinal value that I am not aware of. Yeah.
So to clarify, you didn't find like allelopathy on the trunk itself, but did you find an effect on the ground cover?
I did. I found a significant difference in the ground cover rating, which told me that there was a strong allelopathic effect of the tree ferns, but it wasn't affecting the allelopathic growth. And I believe that to be because of how good a substrate the root mantle is for epiphytic growth. Yes?
Are the epiphytes actually detrimental to either the trees or the ferns?
Good question. The epiphytes are not detrimental. Epiphytes are defined as being like non-parasitic. They just use it as a substrate without harming the host tree. Yes.
Tomaso, can you tell us a little bit about the dispersal mechanism of these epiphytes? Is it always similar, or did you find variants?
No, the dispersal mechanisms for epiphytes are generally very different across families. Usually, they'll disperse by wind, but it kind of depends on how they do that. A lot of ferns, or all ferns, disperse by spores, but other epiphytes disperse by seeds. But yeah, actually, it's usually by wind. Yes.
Artist renditions of the planet many, many millions of years ago often include tree ferns in their work. And I was wondering if you know, by chance, if tree ferns are more ancestral than the smaller little ferns.
Yeah. They are some of the oldest species of ferns still living on the planet. There are smaller ferns that are also have existed for very long, but tree ferns are some of the oldest.
Thank you so much. Can you let me move this a little bit? Let's just move it. We're a little, you look a little trapped in there. Amazing job. Here's the bee. Wicker thing.
Forward, backwards.
Yes. Cool. Yes. Okay. A benefit.
They belong to the family Orchidaceae. They belong to the family Orchidaceae. They're commonly epiphytic. They depend heavily on orchid mycorrhizal fungi known as OMF for their establishment, and many orchids continue this relationship throughout their life. OMF is highly species specific. Generally, they will have preferences for the types of orchid species that they'll colonize. It's worth noting also that many other canopy species rely on mycorrhizae for nutrients in the canopy. Observing this relationship in Monteverde with its many elevations led me to wonder whether these elevations impacted that relationship at all. I predicted that it would have little to no difference, but I was excited to do the research to find out. We chose 2 field sites, both with a lot of primary forest. First was in Monteverde, let's try this pointer out, up here. It was on property behind the Estación Biológica Monteverde. Elevation was approximately 1550 meters. Second site was down in San Luis on private property at 1050 meters near the Estación de Campo San Luis. Monteverde was sampled on November 17th, and San Luis was sampled a few days prior on November 14th. Once in the canopy, once in the field, we chose 2 ficus species that gave us access to canopy, safe access with lots of epiphytic growth. They were around the same DBH, about 120 centimeters diameter at the base of the tree. All accessible orchids visible within that canopy were sampled from until we got to 20 per tree. To sample an orchid, we took at least three 3-centimeter slices of orchid roots from a plant. Those were stored in quarter-folded coffee filters here. Then they were kept moist in a Ziploc bag until we could get them back to the lab. Once in the lab, those coffee filters and the 3 pieces of root were cut down to 5 pieces of approximately 1 centimeter root, roughly this size. They were then treated with the following procedure. They were bleached with, I should say too, they were wrapped in little coffee filter envelopes before treatment and then labeled. They were bleached with a 2% hydrogen peroxide at room temperature. They were cleared with a 10% potassium hydroxide at 90 degrees Celsius and then acidified with a 1% hydrochloric acid at room temperature. They were then stained with a 5% blue ink solution so that the mycorrhizal structures could be viewed. This part of my project required extensive optimization to get the right blend of chemical concentrations, time exposed to each treatment, and temperature. It was very frustrating and definitely the most time-consuming part of my project. So once I had everything treated, analysis involved taking these 5 treated roots and taking off very thin longitudinal slices, putting them on a slide, and then analyzing those for clarity. The 3 pieces that were deemed the most clear were then selected for subsequent observation. That involved taking 3 random observations per root piece. You can see here it was magnified at 400 times. So for those 3 pieces of roots that each had 3 observations, we came away with 9 observations per plant. Those observations determined percent colonization, which was looked at by counting the number of cells on a single plane in the microscope view, and then counting how many of those cells were colonized by hyphae or pelotons. Hyphae are the blue streaks that are running through the cells. That's the fungal network that colonize and reach through the cells. Pelotons are these watermelon seed-looking pieces in the cells. They are tight coils of the hyphal network. They're stored in the cells and then digested by the plant later for food. So percent colonization was determined by dividing the colonized cells by the total cells. These are my results grouped by site: San Luis, which is the lower elevation on top, Monteverde, which is the higher elevation on the bottom. Species was identified after analysis. I found it really interesting too that each plant ended up being a different species. They were processed in order of collection, which I really think just speaks to the the amount of diversity in the canopy. You'll also notice that there are a couple Araceae species that snuck in. They were misidentified as orchids and treated. I decided to include them in my analysis because I think they give a really important context to the special relationship that orchids have with mycorrhizae. You can see the colonization rates are a lot higher in the orchids, ranging from 68 to 98%. This observation average was taken from those 9 individual observations, as was the standard deviation. And I found it interesting also that the lowest observation average and the highest observation average both come from plants of the same genus. Again, just speaking to the diversity of relationships that individual species have with mycorrhizae. So all of that was compiled into a box plot per site, Monteverde here on the left and San Luis on the right with the lower elevation, Monteverde's higher. This blue point in the middle is the observation average for the site. This y-axis is the average mycorrhizal colonization percentage ranging from about 60% here up to about 100% here. The individual plants are plotted in red. You can see there's not a lot of difference between the two sites in average, and a Mann-Whitney U-test indicated that there was no significant difference in colonization between the sites, which means just that there was a lot of overlap in the colonization rates between the sites. You will also notice, however, there is an outlier up here, which is that 98% colonization rate. It had the lowest standard deviation, so it was unlikely to be an error, but just for fun to see what happened, I did run the test without it because you can see that that point is drawing the average up closer to Monteverde. So the Mann-Whitney U-test ran without that outlier, came back less insignificant. It was only a p-value of 0.06, so still insignificant, but it did indicate a trend between higher colonization rates in higher altitudes. These results are similar to those seen in a 2004 study by Rutzeline et al. That looked at root fungus colonization of alpine herbs that were assessed along an altitudinal gradient in northern Norway. In this study, they found that colonization did not significantly vary with altitude, not consistently anyways, and that species level responses may differ more than site level ones, which is pretty similar to what I found in my study. Results that I did find supported my initial expectation that colonization rates would be similar at both elevations, and excluding that one outlier revealed a weak trend of increased colonization at the higher elevation site, although additional research would be needed to determine whether this pattern is meaningful. Any future research, I would definitely recommend prioritizing species level replication so as to attribute any differences seen specifically to the elevation gradient and not individual relationships that species have with mycorrhizae. Definitely a larger sample size. That lab optimization hindered me there. And looking at multiple trees per site, sampling from multiple different trees to exclude any individual differences that a host tree might have. So, thank you. They do, most orchids tend to have OMF, the mycorrhizal fungi, that will colonize just with them, and vice versa. There are mycorrhizal fungi that are known to just colonize with certain orchids. They have maybe like 3 or 4 that they'll cycle through throughout their life, according to the research that I found, but that's the only pattern that I could find, if that's what you're asking. Okay. Kara.
You mentioned frustration in the lab. What was like your biggest breakthrough that you were super proud of in that process?
The day that I let go of the idea of processing all of my samples. That was my best day. But yeah, it was just a lot of soggy samples and then unclear samples, and big shout out to Laurel Schmidt, who had a procedure from a couple years ago that I was able to follow when I got frustrated. Thank you.
Hi, Emmy. Yes. I was wondering why everyone was so loud. There's a microphone.
Oh. Yeah, but that's for YouTube.
Oh, never mind. Okay, everyone's naturally loud. did talk very loud.
but just in general.
It's good love. But it's good. It's good.
No, yeah.
All right, and now we move, sorry, and now we're moving to the world of animals and particularly insects. So Ty Brown is going to teach us about insects that are also found up in the canopy. Thank you, Ty.
Thank you, Emmy. Okay, my name is Ty Brown, and as you know, I can talk about insects forever, and I only have 9 minutes, so please talk to me after if you have any questions. I studied insects in the canopy, and this is at the intersection of 3 understudied biota, 1 cloud forest, 2 tree canopies, and 3 insects. Cloud forests, where we are now, are unique environments that are categorized by steady misting. They're also located in the tropics, which is understudied in comparison to the general abundance of the world and how understudied that this place has been in comparison to the world. And tree canopies are also understudied, given they are more inaccessible without specialized personnel and equipment. And they also contain a great majority, or I thought a great majority, of insect biomass in the tropical environments. And then insects, also understudied, given they comprise over half of the biomass of the animal kingdom, and research just isn't there yet for that. And they also are on the decline, which you can imagine the consequences of that. I set out to study a diverse range of insects, so I wanted to use a lot of methods. All of my methods are attractant methods because we only had 3 days to collect data. The first of which I used a flight interception light trap, which was invented here at the Institute. It was very innovative, and Jorge helped me so much with that. And it was also a pretty low budget method and ended up collecting around 400 insects, which was a great portion of my data. And what happens in the trap is the insects fly into this wall that's in the middle, and then they fall into my collection bucket. And these usually catch insects that are worse at flying, like beetles, or because of the light, moths or other insects that are attracted to light. And then I also used a fruit bait trap, which is a common low-budget method of determining insect abundance. They're just attracted to fermenting fruit and beer, and yeah. And then the euglossy bee trap, I had read that it also collects things other than euglossy bees, but I was curious as to the distribution of euglossy bees between the canopy and the understory. And so we set these up in a large fig tree, left it for 3 days, and then collected the insect soup, took it back to the lab, and then determined insects to sort, to order, and then family, and then morphospecies. Morphospecies is like basically just what the insect looks like is different than the other insect, and that is how you determine different morphospecies. So in total, collected 610 total insects, 306 of which were in the canopy, and 304 of which were in the understory, which is pretty interesting, pretty similar distribution. I was kind of scared at first that I'd done something wrong, but no. And then 172 morphospecies, and 38 families, 8 orders. This is just euglossy bee, one of the prettiest ones we found. And yes, just to go more in depth on the order portion, the majority of the understory is categorized by Diptera, which is understandable because they're attracted to fermenting fruit, and that's mostly where they got caught, and fermenting fruit is on the understory, and so that makes sense. And the canopy was more evenly distributed in the major orders between Diptera and Coleoptera. And I also don't know if too many people know about Psychoptera. Those are book lice. Archaeonatha, those are primitively derived insects. They are pretty interesting. They eat detritus, and I also wanted to mention Dermaptera. You probably see those in your bathroom. They really like moist environments. They're earwigs. But you ask me more about that another time. Okay, so, whoa. All right, that says 84, 25, and 63. This is a this is a distribution of the morphospecies I found. So let's say 25 of the morphospecies can be found interacting between both the canopy and the understory, whereas a majority of them I found to just be located in the canopy or the understory. And then with the canopy having the most diversity of morphospecies. Okay, so to get more into depth on morphospecies and families, although as you can see there is a lot, so I can only touch on so many. We have just a few, and the ones in bold are unique to the understory. So let's see. I know that I said that Diptera flies are the most abundant in the understory. I do want to touch on some beetles though, because there were quite a few beetles. First, Cursileonidae, that is weevils, and I know that does not look like a weevil, but it is. And then these in the middle, these are click beetles. They are very cool, and I found that in the Euglossy bee trap, which I found to be kind of interesting because I was wondering what I would find in that, given they're trying to attract Euglossy bees. And then Cantharidae, these guys can be mixed up with Lampyridae, which are which are fireflies. Whoa. And that's just like visually, but these are soldier beetles, which are very common, and I found a lot of them in both the understory and the canopy, because it's not bolded. The only click beetle I found was in the understory. Okay, and then Staphylidae, this is another important one. These are rove beetles. This does not also look like a rove beetle, but because they have like kind of a butt that goes upwards usually, but it is a rove beetle. This one was unique to the understory because it was kind of hairier than the other ones that I saw. But this was another family that was common between both areas. Okay, flies, very important, touch on flies. Drosophilidae, these are fruit flies. They're characterized by their wing venation. They have really big red eyes, and you can't see it, but they're striped on their back. Not going to say anything more about that. Phoridae, these are scuttle flies. They have a very small head, and they look like this. Okay, hold on. I want to show you these guys because these are also fruit flies. They're called true fruit flies, and their wings are so beautiful. Their eyes are very psychedelic, and they have little stripes on them. Okay, fruit flies. Let's just move on. Canopy. All right, so these are all bolded except for Apidae, just because their morphs were unique to the canopy, and I wanted to touch on them. Patilidae, very cool. These are feather butt beetles. They have a feather butt, feather wing. Sorry. But it's just so cool. Why do they look like that? I have notes on them. They're very small, and they feed on spores and hyphae, and I only found like 4 of them in the canopy. Cursileonidae, these are also weevils. I showed you them before. I just wanted to show you just 2 different morphs next to each other. And then Phenagonidae, this is very cool. They're glowworm beetles, so their larvae glow, different from Lampyridae, which glow as adults. And their antennae, you should look them up later or right now. Phengonidae, they're very feathery antenna. Okay, probably just being very excited about insects right now. Okay, and then Apidae, we found the 7 of the Euglossy bees in the canopy and only 1 in the understory, which was very interesting and makes sense because of no Leo. Okay, it's over. Ask. Oh. Okay. There's a lot of orchids and flowers in the canopy, and so that makes sense that these orchid bees are pollinating in the canopy. Phengonidae. These are two peristoid wasps, and their hosts are in the canopy, so that makes sense. Oh, these are very cool. Look this up later. They just fold in on themselves, and I only found one of them, and it was in the canopy. Okay, so overall, lots of flies in the understory, and Phoridae was the most abundant. They're attracted to light, so that makes sense. And then canopy, let's just say that was very diverse and abundant. And then, yes, thank you. This special thanks. I can't choose.
In this experiment, what designated the difference to you between understory and canopy? Was it light levels? Was it number of branches? Was it just where you put different traps?
This is a great question. I set up a bunch of traps on one tree in the understory, and then we climbed pretty high and set up the rest in the canopy. So,
Did you measure the height of it?
no. More than 15 meters. 15? 15 to 20 meters. It was very tall. Yes. Okay, so yes, I'm more unconfident in explaining the slide, but I got this for Leo, I see you nodding, and Fede. So yeah, there was higher, there were more species, unique species to the canopy than the understory, based on this. I don't know. Well, that's kind of what I expected. I think there's not a lot of research done on the canopies, and from what I've learned from my entomology courses, there are just like so much more in the canopy. So that's what I expected to find, and so finding the same number of insects in both was surprising to me. Yeah, but there is more diversity. And I also found more insects that I couldn't identify in the canopy, probably because we have less research on them, and yeah.
Thank you, Tyler.
Yay. I'm taking your water. I was like, it's so. Thank you. Oh, I'm excited for this one. Oh. Sorry.
Oh, thank you. Anyone on the presenter mode on a
in my computer. We could create it. In my computer, I open it in a different app. So if I connect my computer, it works as like a.
Do you prefer to do that? Use a computer?
Yeah, I can do that. Let's use my. Usually the PDF is going to be the same. So you have to use your own computer to open it. Did you make it on Canva? Just put it in presenter mode on Canva. That's what I'm putting. Right? Wait. Yeah, I can do it on my computer. I have. Yeah, I need that. ¿Para qué necesitamos?
Para esto.
Ah, para eso.
Tiene, sí.
Whoa. There we go. Okay. Oh. Quit? No. No. I know. Giant arrow. Wait, what did you just plug in? Because before it was working. What is that? Oh, for this? Yeah. I don't need it. I think that might be messing with it. Ah, Or I need to put it in present mode. Okay. Yay! I can just point, because was that for this?
Yes. I don't think it's gonna.
Okay, you can try and plug it in.
Oh, yes. Oh, there you go. Ah, it was the thing.
Wait. Okay, it's working. Yeah, it's working. Just leave it. Yeah.
Right.
Okay.
Sorry again for the technical difficulties. You're very nice audience, patient audience. Thank you so much. So our next presenter is Haley Penfield, who's going to teach us even more about insects that are attracted to light in the understory, and more particularly with moths. Thank you.
Hi, my name is Haley, and I'm going to be talking about the effect of artificial light color on moth abundance in Monteverde, Costa Rica. So for some background, artificial light at night, there's been an increasing dependence on it, and it's also called ALAN. ALAN is used to light up homes, streets, and businesses, and typically has a negative impact on the natural ecosystem of many nocturnal organisms. The visible light spectrum for humans ranges from about 380 to 700 nanometers, ranging from violet to red. And while there's diversity between moth species, typically they see between 300 and 550 nanometers from UV to green. So what are moths? Moths are insects within the order Lepidoptera. They are nocturnal and serve as pollinators, decomposers, and prey for many organisms. Moths are a very diverse group with about 160,000 species worldwide, and about 5,000 of those can be found here in Monteverde. Moths also use transverse orientation for navigation. This means they keep a constant angle between themselves and a distant light source, such as the moon. Because the moon is so far away, keeping this constant angle allows moths to fly in a straight line. But when there's artificial light, this messes up their navigation system and they confuse it for their distant light source, angling their body towards the light, causing them to spiral around and around. I was interested in human impact on nocturnal insects, specifically moths, which led me to my research question: how does light color influence moth abundance and taxonomic composition in Monteverde, Costa Rica? My study took place over 12 nights. I had 4 groups: red, blue, orange, and an unfiltered control. And I had the light trap set up like there with the white sheet in the back where insects such as moths and others could land and be observed. At 5 p.m. each night, I set up the light trap before sunset so that at 6:30 p.m. I could take 30 minutes of data collection. And during this time, I took photos of each moth that was about 2.5 centimeters or bigger, which was the size of the segment of my pinky. And I also took note of wind level, ranking it in low, medium, or high. And at 7 p.m., I took quadrant photos of the white sheet so that I could later estimate abundance. I used online resources to record moon illumination, and I used field guides and iNaturalist to identify my moths to families and, if possible, genus level. So these are some results related to abundance. On this graph over here, we have light color on the x-axis and moth abundance on the y. I ran a Kruskal-Wallis test, which showed that these results were not statistically significant. While it looks like the control and blue have higher average abundance, there was great variation within the data, and the p-value was greater than 0.05. So this means for my study that moth abundance was not dependent on light color. Looking at this graph over here, we have moon illumination as a percentage and moth abundance on the y-axis again. There is a weak relationship with the r-squared value of 0.28. This also means that for my study, that moth abundance was not dependent on moon illumination. Moving to this graph, we have wind level on the x and moth abundance again on the y-axis. I categorized wind level into low, medium, and high. And as you can clearly see, there is a relationship with low wind level resulting in higher moth abundance and high wind level resulting in lower moth abundance. This box plot shows the range of values within each group, and these red X's represent median values, and the letters indicated by A and B represent statistically significant differences in groups. So the A in the low wind level and the B category in the high wind level indicate that these groups are statistically significant in terms of differences in moth abundance. However, medium wind level has A and B. This means that between the low and the medium wind levels and the medium and the high wind levels, there's not a significant difference. But overall, my study does show that moth abundance is dependent on wind level. The other part of my study focused on the diversity. Over the 12 nights, I observed 14 families and 117 genera, and I wanted to understand if the diversity was different between different colors of light treatment. So I ran a chi-square test, and this resulted in a chi-square value of 10.13 and a p-value of 0.02. These results are statistically significant, meaning that different light treatments do result in differences in diversity. This graph here shows unique moth genera by light treatment. The C, B, O, and R represent each light treatment, and this graph shows the individual as well as light treatment groups. And as you can see, the control light treatment resulted in the highest amount of unique genera, followed by blue, orange, and then red, while the groups of treatments, whether they're groups of 2 or 3, typically resulted in lower unique genera. And the group where all treatments were present had also a low value for unique genera. While the high number of unique genera for control and blue may be related to moth sensitivity to shorter wavelengths, the short amount of and limited data cannot confirm this with certainty. So I observed 14 families. These are example pictures from these 14 families. Euribidae and Geometridae had the most genuses within them. I saw them every single night. And here are the other 7 with Pyrolodia, which is actually a superfamily, being the most abundant, seeing many individuals every single night of those. So in summary, light color did not impact total abundance in my study, but wind intensity did, with higher wind resulting in lower moth abundance and vice versa. I also found that light color did influence composition and diversity of moths. And in general, longer wavelength lights, such as orange and red, likely have less negative effect on nocturnal insects. For broader applications, again, using warmer color lights when possible, also reducing the use of ALAN, whether that be dimming lights, turning them off when they're not needed, or pointing them more downwards, will all help improve the negative effects of ALAN on nocturnal insects. And it is also important to continue to raise awareness and support future research surrounding human impact on tropical insects, especially in such a biodiverse and complex area like Monteverde. And I wanted to thank Emmy, who's my primary advisor, and Leo for helping me develop this project and complete it, and Jorge Mora for helping me create the contraption to change the color of my light. La Estación Biológica for giving me a safe place to conduct this study, and Noel and Antonella for coming with me to the station every night and helping me with data collection. Thank you.
Just out of curiosity, do you know what the moths do on high wind nights? Like if they're hiding out somewhere or what they're doing?
I am not for sure, but I think that flying in the wind is more difficult, so they more stay where they are and don't hunt as much.
Were there other insects besides moths that you just saw every single night that you're just like that you wanted maybe to be like add to the study, but just didn't think you were only doing moths?
Yes.
So many other insects. Most interesting were beetles. There were a ton of different beetles every night. They were super cool colors. I don't know a lot about them, but it was very interesting. And they would typically stay on the sheet for the whole duration
and not move much, which I thought was very interesting.
Is there a beetle diversity to light in wavelength?
I did not study the beetles. There was usually only like 1 to 2 per night. They were just interesting to look at and cool to see. Yeah.
Can you tell us how you calculate moon illumination?
Yeah, I used an online calculator and put in the coordinates of the location, and it gave moon illumination for each date that my study was conducted.
And in the graph of light treatments, the different treatments it means that you mix lights, or it's just the modes that came to this different lights and share in the diversity of these different.
On this graph?
Yes.
Yeah, so when it says like C plus B, I was looking at unique genre that were only present in control and blue. I didn't mix.
Yeah,
but
you don't put them in the same.
No.
there any difference between sex and light difference or no?
That's a very interesting question that I did not think about. I do not know how to differentiate between males and females, but I think that would also be very interesting to see their patterns.
Thank you.
So fast. Oops. No. Okay. I finished a little before. Oh.
Is that on?
You don't have to do anything, just like plug it in. I mean, not plug, but.
Oh wait, did I just mute it? No.
We don't know
Wait.
how to present a PDF.
What did you want?
We don't know how to make it in like presenter mode.
What form did you want it as?
Maybe if we open it with something different.
Wait, what were we supposed to submit it as? A PowerPoint? I thought it was a PDF. I thought they said a PDF because a PowerPoint if there's no Wi-Fi. Huh? It does? Oh. I could submit it right now. Pretty sure I was told to submit a PDF, but whatever. What email do you want me to send it to on this computer?
Do you want to use your computer?
I could just send it as an email.
I think it's easier if you just use your computer right now. It will take a lot of time. I mean, I don't even know whose computer it is.
Okay. Dude, you told me to put a PDF.
It's okay, it's okay. It will work.
Yeah.
For 9 minutes, your time for speech.
So when you put up the 5, it's been 4 minutes?
Yes, you still have.
Yeah.
Yeah.
You still have.
Plus if you
Stop Sable, guys. Huh? No. Entire screen.
Well, it's weird because I write things on this side and this back, or whatever.
I don't think I could use those because my brain is process
Okay.
I think I can read this. Like my hand. Yeah, so that I remember like I'm kind of visual. Where's the share the screen? I don't know Mac.
I have to see an image.
Choose content. Start mirroring.
All right.
Tranquilo. Tranquilo. Is this the pointer? Okay.
And you can use it
Yeah.
to like forward and backward.
Okay.
Okay, it took us a while to get to the final presentation, but we finally did it of this first session. And this is going to be the last one about arthropods by Jared Humphreys, who's going to teach us about arthropods in coffee potters. Thank you, Jared.
Yeah. Yeah, thank you for that wonderful introduction, Emmy. My project was on arthropods, and arthropods are the phylum that contain the class of Insecta and Arachnida. And I chose to do on arthropods because I was looking at insects and spiders. And so my project was on arthropod diversity between two coffee farms. So the two coffee farms that I compared both supply coffee beans to Cafe Monteverde, which I assume many of us have enjoyed. And these two farms are Life, which we all previously visited, as well as Turin, which is a bit more like out in the boonies. And basically, they had many similarities, such as the age of the plant, the size of the field was about the same, and they all had trees interspersed throughout the fields. But where they differed was in surrounding conditions. At Turin, most of the fields were surrounded by like one layer of trees as windbreaks, then more coffee fields. Whereas at Life, they had a lot of like native forests on multiple sides, and then one side was like a tree windbreak and coffee field. So surrounding conditions differed a bit. But the area that I was interested in that differed between the two was the plants that existed between the coffee rows. So at Turin, they regularly apply about 3 times a year herbicides to suppress weeds, and then they plant beans or frijoles for eating between the rows. Whereas at Life, what they do is 1 meter around the plant, they use a machete to cut the weeds down to suppress them. But between the rows, as you can see right here, they allow the weeds to grow to full maturity. So this difference in the plants between the coffee rows led me to ask the question, how does plant presence between coffee rows influence arthropod diversity? And so getting into the methods involved a bit of getting into the weeds, quite literally, as you can see right, thank you. And I took 10 quadrats at each site for the plants between the rows. And Life included a lot of weeds, as we can see here, but Turin, I was mostly taking quadrats for beans. And what I took was vegetation cover, which includes, vegetation cover is where the percent of the ground that is covered by leafy material, as well as I took the plant morphospecies diversity for each quadrat. So for insects, I did 5 transects spread out throughout each field. Each were 10 meters. And what I would do is I would use a net, and I would sweep those plants between the rows to collect the insects. Once I collected the insects, I would freeze them, put them in alcohol, and then look at them under a microscope. My identification involved like classifying on morphospecies, which is like shape, size, coloring, clear marcations. And I would basically classify morphospecies and then the number of individuals for each morphospecies at each farm. So moving on to my results, I found that Life had higher vegetation cover and plant morphospecies compared to Turin. And that was something you could kind of see within the pictures already, but I just want to quantify it. And here on the left, you can see the graph of vegetation cover, where on the x-axis, we have Life in dark green, and we have Turin in light green. On the y-axis, we're looking at the average vegetation cover. And as you can see visually, Life had a greater average vegetation cover compared to Turin. And running a t-test between the two groups for the 10 quadrats collected at each, found a p-value of less than 0.05, which indicates that this difference in average vegetation cover between these two groups was statistically significant. In a similar fashion, for plant morphospecies, I found that Life had, on average, a higher number of plant morphospecies per, for the farm. Moving on to insects, I found that I took insufficient transects to determine the full amount of morphospecies per farm. So essentially, I didn't do enough transects to be like, I caught all the potential species there. If you remember from Fede's class, we had the cumulative analysis, where on the y-axis, we're looking at the cumulative species, and on the x-axis, we're looking at the transect number. This is basically showing us how many new morphospecies are added per transect. And because you see that neither of the graphs reached a clear like limit or like reached like a flattening point, and they weren't flat for long enough. So that shows me that I didn't take enough transects to really say I fully surveyed each farm for the insect morphospecies. Now for diversity, I did a true diversity index, which is basically an equation where you can manipulate the Q value, and based off of the different Q values, give you an estimated value of like different species to kind of different like classifications of species to give you an idea of diversity. So when Q equals 0, we're just looking at the raw species richness. So common species that are in high abundance and rare species that are in low abundance are all weighted equally. So you're just counting the number of species per farm. And what I found was that life had more insect morphospecies at 98 compared to terrarium, which had 44. When we set Q equal to 1, you're basically weighting each species proportionally. So adding to create the final value, more common species are going to add more values compared to rare species. And so it's basically the number of effective like balanced species. And terrarium had 20, whereas life had more at 34. When Q equals 2, we're looking at dominant species. This is based off the Simpson's diversity index. And terrarium had 11 dominant species, whereas life had 19. All of this to say that life had greater diversity of insect morphospecies. So in terms of quantity, we could see here by this visual that for number of arthropod individuals, life had almost 4 times as many insects as terrarium. So to discuss these results, life had higher arthropod diversity and quantity, and this could be because of the higher plant presence between the rows, as we saw with vegetation cover, plant morphospecies. But a confounding variable could have been that life had more surrounding forests, which literature has shown increases diversity and quantity. In terms of the implications of this project, literature has shown, oh look, some insect diversity around us right now. It shows that higher plant species between crop rows increases arthropod diversity, and interestingly, this increased diversity reduces pest pressures on farms. And this could be because at life I found 59 orchid spiders which control pests, whereas at terrarium I only found 1. And so overall, this study could show the positive impact of life's practice when you allow more plant species between the rows. That increased diversity could be allowing for greater predatory arthropods, which control pests, as well as increased pollinators, which could increase pollination rates of the coffee plants and increase overall yields. So overall, showing the potential benefits of life's practice. With that, I'd like to thank Emmy and Naomi for really like helping me learn about the scientific process and about myself as well. I'd like to thank Sadie back there for being an awesome homestay mother, Diana for helping organize things in terms of research, Eladio for helping classify the plant morphospecies, and especially I'd like to thank Eddie. He was like one of the on the grounds coffee like farm managers, and he really helped me understand like the on the ground reality of what it is to manage a coffee farm. So thank you. Owen.
So if you go back a few slides to the cumulative species graph,
Yeah.
unfortunately my eyes were not good enough to see the y-axis.
Yeah.
Can you just tell us the numbers there?
Yeah, so the numbers are 30, where like you see that terrarium reaches 44 species and hasn't clearly leveled out. And then for life, it reaches 98 up here. And so it basically, the final transect is reaching like the species richness or the Q equals 0 for each.
Okay, so each step is by 30 then.
Yeah, each step is by 30, yeah.
Ruby?
I was wondering if you, maybe in talking to the people at this farm,
Yeah.
like learned about like which obstacle they're like choosing to combat more, like pests versus herbicides, and like if the choice to let weeds grow is like a deliberate choice towards natural pest control.
That's a great question, because what I found was that the reason why they don't usually do what they do with life, where they let like the weeds grow, is simply because like using manual labor to weed just costs way more compared to herbicide, even though there's like the benefits of increased insects. Also, another interesting thing is that you do, to a certain degree, especially once the coffee plants are mature and you're harvesting it, you do have to clear the weeds to a certain degree before harvest, because like snakes can hide in the grass, and like that could then impact the workers, and so that's the important social aspect of sustainability. Ewan?
You mentioned finding around like 50 orchid spiders. Was
Yeah.
there any other arthropod species that was in like big abundance that you found?
I found a lot of leafhoppers between both farms, and that's the one that looks like this right here. And and at both sites, I found a lot of dipter, a lot of flies, and I feel like that made me really fully hit me in the face of the biodiversity portion of this program. I was like, wow, there's a lot of flies to classify. Sarah? Mhm. Yeah, I dug into the soil at Life. Unfortunately, I forgot to when I went back to Turin, so that's why it's not part of the project. But at Life, like digging into the middle, like definitely a lot of roots because of the plants, and I'm assuming that would have a lot of beneficial impact for the microbes in the soil. It also helps control disease. That's what Eddie told me. Another shout out to Eddie, is that like the diversified roots can kind of like suppress the spread of disease if it, instead of just being only like coffee and beans.
Just to ask, I don't know if you have a sense of, in terms of richness, it's very different.
Mhm.
But do you have an idea about how similar are the compositions, like the variation? Are the Turin ones a subgroup of the light ones, or are very different in terms of the beta change?
Yeah, I didn't specifically look into like similarities and differences, so I don't want to like talk too much on that. But there definitely were a lot of shared species, like the leafhoppers, a lot of shared fly species, shared beetle species. But where Life was unique is they had 59 spiders compared to 1 at Turin. I also caught more grasshoppers at Life versus Turin. Although that could have been because they're kind of trapped in the grass, whereas like in Turin they had more open space to jump away. Yeah.
Could be very interesting to see functional.
Mhm.
Very, very nice project.
Yeah. Oh, but an interesting thing though is I found a tiger beetle at Life, which is a really important predatory insect. Yeah, which can kind of show the benefit. Yeah.
Thank you.
You can ask me after. Time plug it.
Thank you to all the presenters from this amazing first session. Now we're going to have a little break, and then we'll come back. 10-minute break.
There are refreshments outside. Wait, keep pulling. That's the truth. You can get all the refreshments.
You know how much it was? How long it was?
How long?
It was 9 minutes.
Whose computer is this? Can I make sure mine's
This
on
is
there?
Jared.
Yeah.
Is everybody bringing up their own computer?
Should I unplug it?
Mine doesn't work with stuff.
The thing is that they had their sensitivity, and
Yeah.
I was just like,
Mine is a Google slide, but
Thank
I can
you.
also have mine as a
I really
PDF
appreciate
sent
that.
in an email.
so
I don't have mine
this
hard
is the
drive
kind
because
of
my
thing
computer's
that would be really, really great to be able to do a vlog on, or to
Yeah.
pamphlets on, or a social media post.
Yeah. That was my whole goal. I basically I want to make an insect guide and a plant guide for Cafe Monteverde, and then maybe I'll set up a Zoom meeting and present them.
they
my
definitely
result.
should
Yeah.
get it. But also other coffee farmers. I mean, this is regenerative.
Yeah.
This is just
Yeah.
really, really cool to be able to show that.
Yeah.
The money thing is really significant.
Yeah. Yeah.
But did you do a video?
No. No.
Okay. Because we gotta share this,
Yeah.
not just
Yeah.
to light,
Yeah.
but in general.
Yeah.
Tomorrow there's a Agro Regenera is
Yeah.
doing the Association for Regenerative
Yeah.
Agriculture is having a meeting mostly focused on dairy farmers and what they can do.
Yeah.
This is the kind of thing that they should really have access to.
Yeah.
Really,
If they have the
good.
meeting tomorrow, I could send my presentation.
Well, there's not going to be time for that kind of stuff
Yeah.
at that meeting,
Yeah.
but it's good to be able to mention it
Yeah.
to keep people from that association. Because
For sure.
this is really cool.
Thank you. I really appreciate
I'd
it.
be really curious to know the difference in cost.
Yeah, it's about 200 more to like hand weed versus
200
dollars, like US dollars. So per like field is what they found. To use manually hand weed versus herbicide.
Yeah, I wonder what that
Which usually like some
translates to.
yeah.
Bueno, si usted puede ayudar, genial. Sydney, just use your computer. It's easy enough to.
Okay, if it's on my computer, then it needs to have a USB-C.
We have that.
Okay.
It works.
I was just gonna say something because I was the last one, but I'll change it.
Can you send it to me, Sydney, to my email?
She just hit as hard.
I don't think it's a lot. Okay, here's the thing. On this one.
Because all it is like.
You should have said no.
I'll just say it. I know you're gonna do sections. And then I'm also the pictures and videos in this presentation. Yeah, they're mine. I did that.
It's all me.
Look at this girl.
I always try and hand myself up. I get excitement over anxiety. That's why I try to, yeah.
Está viendo
No, I
No, as long as it's the right brand.
Hello everyone, we're starting again. So sorry, we apologize for the delay. Sorry. Nothing we can do about that. Well, that's not entirely true. Anyway, the next section is about small things, living things in the sea. The folks who are presenting this elected to go to the fishing village of Guajini Keel and tough out the mosquitoes with dengue and a variety of other challenges in rural Costa Rica. And so they're going to be sharing what they learned. And our first speaker is Kim Cardona from UC Santa Barbara. And she will be talking about an amazing phenomenon that EAP students witness, which is bioluminescence. And just a plug for the small things in the world, arguably plankton and some of the things that Kim studied are some of the most important creatures on the planet. Thank you, Kim.
Hi, my name is Kim Cardona, and today I'm presenting bioluminescent branches and species abundance among different sites. For my introduction, so bioluminescence is a production of light emitted from living organisms common among aquatic communities. The production of this light is caused by chemical reactions involving luciferin and luciferase. The ecological drivers of the glow are used to distract predators and even are considered as possible signals within common dynamics. The gradient of the onshore to offshore locations provides ecological and environmental conditions for planktonic species in varying light conditions. I wanted to study this topic because it is an under-researched, understudied phenomena that proves that within the littlest amount of water there is so much life and so many interactions happening between the visual iterations of the flashes. So this has led me to the question too: how does plankton abundance relate to bioluminescence brightness across a nearshore to offshore gradient? My materials and methods. This is a map of the location that I chose to collect my studies and my samples in. So this is a Blair equipment of Nico. Site 1 is used for the onshore location, Site 2 for mid-distance, Site 3 for offshore. And between these distances, there was a measured amount of 384 meters between Site 1 and Site 2, and 318.99 meters between Site 2 to Site 3, and in total between Site 1 and Site 3, there was a total of 639.83 meters. The amount of meters between each of these sites is good for accuracy between trying to measure my results. I also collected my results at Site 1 on November 17th and my results on Site 2 and 3 on November 26th. For the rest of my methods, I used a 5.25 liters of water and filtered it with a plankton net and poured it into a tank. And the tank was put in a dark and disturbed area. I'm sorry, before that, 50 milliliters of plankton sample was used to be poured into the tank so that observations can then be made. And then the tank was put in a dark and disturbed area for Site 1 on the onshore distance. For Site 1, the onshore distance, the tank was put on the sand, and for Sites 2 and 3, the tank was put on in the cabin of the boat. The observations were made in green intervals between 30 seconds, 1 minute, and 5 minutes, and within all of these time intervals, I measured the flash frequency, and I basically counted the amount of flashes within each sample. Then I collected another 50 milliliters of plankton samples in each location and stored them into water bottles, and they were taken to the refrigerator. Using these plankton samples, I identified and counted the organisms under a dissection scope, and I also used alcohol to stop the organisms from swimming for more accurate identification and count. I also, after identifying these taxa, I taxon for the presence of bioluminescence and inputted the data into bar graphs. So these are my results. The bar graph represents the bioluminescent flash frequency across all the sites. So the x-axis represents each of the sites, 1, 2, and 3, onshore, mid-distance, and offshore. Then the y-axis represents the number of flashes that were counted in each site, and the difference between the bars and the colors are the 3 intervals of 30 seconds, 1 minute, and 5 minutes. So here I found that site 2 had the highest amount of flash frequencies between all 3 intervals, with 5 minutes being a number of 141, being followed by site 3 with a number of 83 flashes, and being followed by site 1 with a number of 42 flashes. And the other time intervals followed the same, follow a similar pattern. Site 2 being the most abundant flashes, and site 1 being the least. This bar graph represents bioluminescent taxa abundance. The x-axis representing the taxa identification, and the y-axis representing the number of each taxa, and the bars representing each site. As you can see, the most abundant bioluminescent taxon was the Nauplius, being the most abundant in site 2 and site 3, followed by site 1. The next most abundant were the Balanopsis and the Copepods. The Balanopsis was the most abundant in site 2, and the Copepods were the most abundant in site 3. This graph is similar to the first graph, except this is a non-bioluminescent taxa abundance. So as you can see in site 2, once again, there's a greater amount of certain taxa in the Cladocerian, being followed by site 3, then site 1. And the next two most abundant taxa that I counted were the Neonisis and the Hygroche, being the most abundant in site 1, being followed by site 2. The most abundant was unidentified zoa. So for my discussion, as we saw, for site 2 had produced the most amount of flashes and had the most abundant non-bioluminescent and bioluminescent taxa. Site 1 produced the least amount of flashes and had the least amount of bioluminescent and non-bioluminescent taxa, while site 3 stayed in the middle. This was a pattern that was followed for each of the graphs. So to go into detail about each of the 3 most abundant bioluminescent taxa, we have the Nauplius, the Copepod, and the Balanopsis. The Nauplius is an herbivore's early infant stage with a trophic level of 2. The Copepod is an herbivore and omnivore with a trophic level of 2 to 3, and the Balanopsis is a gelatinous predator with a trophic level of 3 to 4. The most abundant non-bioluminescent taxa included the Cladocerian, Neomycete, and Hygroche. The Cladocerian is an herbivorous zooplankton with a trophic level 2, and the Neomycete is a tetrivore and omnivore, and has a trophic level of 2 to 3, and the Hygroche is a predator and scavenger with a trophic level of 3. So for the rest of my discussion, my findings support my prediction. My findings support my prediction that the bioluminescence brightness is closely related to plankton abundance and species interactions along the on to offshore gradient. This confirms the findings that bioluminescence can act as an indicator of community structure and ecological activity. The factors that have influenced the results include the plankton in the tank that changes the natural flow, and it might have increased the mechanical triggers that are unusual for the organisms, and the larval forms were very challenging to identify, but despite these limitations, the consistency of flashes across the time intervals tells us that the results reflect real differences between different sites. I would like to give a special thanks to Lil, Frank, Maya, Janice, and Emily for making this project possible and helping me with my research. It's supposed to say thank you, pura vida, but thank you, pura vida.
frequencies of flashes in the bioluminescent plankton, is that how you judge what was bioluminescent or not? Or did you use like a sort of catalog or plankton sort of directory that lists certain species as bioluminescent? How did you judge?
So counting the amount of flashes was more separated to see the abundance and flashes within each location. But actually identifying each taxa that was bioluminescent or non-bioluminescent required me to do extra research, going to different websites and looking at different books, especially looking at a lot of different books. And from there, I compared what was and wasn't bioluminescent, and then I counted each of those taxes.
Were there any that were listed as bioluminescent that weren't bioluminescing?
I couldn't see which ones were bioluminescing or not bioluminescing. I think maybe with there's not really a way to look at it through a microscope. I was able to, I know Frank helped me, I was able to see some organisms flashing under a microscope, but it wasn't accurate to identify which organisms those were. So I just had to assume which ones were by doing my research on which ones didn't.
Oh, okay. Sorry, I guess you kind of just answered this, but I was curious if you noticed a certain organism that produced a lot more light than some other ones that you studied. And I know you couldn't see them all in the microscope now, but I was just curious.
I couldn't see like which organism did or didn't produce light, but I can see that the most organisms that are bioluminescent were the Nauplius, so I would assume that that organism produced the amount of light out of all of the other organisms.
Okay. Oh, so they can let this. Okay.
Trinity.
I'm glad we're on this graph. I was wondering if of all these taxon, are these actual taxon that you saw, or were thinking and like predicting you would see but didn't see?
So these are actual taxon that I saw. You might not be able to see exactly the amount or maybe the graph because it's too small for you guys, but there were about one or two of some of these taxa, which is why like the bars are so low. But I did look at the dissection scope and I identified them. Like when I saw these pictures here, I took a lot more. I just uploaded the most common and abundant within the most abundant taxa that I found within my presentation. But I took like a million pictures like these for each of the taxa, and that was how I was able to identify it.
Your last question, FNL.
Would you change anything of your project to give me more time?
I think if I was given more time, there would be more of an accurate way to detect the amount of flashes. For example, instead of using tank observations to detect flashes using my eyes, I think that if I was given more time, I would use the fluorometer which is a device that can detect fluorescence and bioluminescence in the water, and I think that would be more accurate. And also going into timing, I think that I would also measure abiotic factors in the future because I wasn't able, when doing my research on abiotic factors for this project, when doing research for abiotic factors within this project, they didn't make much of a difference or a change between the 2 weeks that I had for this, but I think if I did a long-term project of over a year, then they would make more of a difference because of the seasonal changes.
Awesome, thank you, Kim. Sí, está listo. Nacho wants to trade the microphone. Sí, gracias.
Yes. See? It works. It's a thingy. You don't have to scroll.
So exciting.
Next speaker comes to us from UC Santa Barbara. She is Lainey Wake, who will be telling us about the process and arrival of little creatures on available substrate in the ocean.
Awesome, thank you. Hi, I'm Lainey, and this is the impact of environmental conditions on germling and meroplankton settlement. All right, first to start off with a few definitions. Settlement is when free-swimming planktonic organisms find and attach to a benthic substrate. Metamorphosis is a physical process of transformation from planktonic organisms to juvenile organisms once it's on the substrate. A germling is an organism produced post-germination from an algal spore, and meroplankton are organisms that don't spend their entire lifespan as planktonic and will eventually settle to either be nectonic adults or benthic organisms. All right, so many benthic dwelling organisms rely on settlement from pelagic free-swimming organisms to benthic individuals to persist, and settlement is cued by environmental factors, and this settlement can be delayed if environmental factors around organisms are not favorable, and this period of delay is called the settlement delay. And settlement delay is evolutionary essential, especially towards sessile organisms, because it determines where they're going to spend the rest of their life, the resources they'll have access to, and where they'll eventually reproduce. This period of settlement delay can last anywhere from minutes to months, depending on environmental conditions as well as larval types. Algal gremlins also have agency in their settlement location. It is a lot more difficult for them as locomotion is more difficult for algae. And because of this, something called the laminar sublayer is essential for larvae. And the laminar sublayer is a really skinny area of water right above substrate that has smooth water flow. And algal gremlins in this area have a much easier time settling. And a few examples of positive settlement cues are chemical, acoustic, surface textures, as well as the presence of algae and coral. All right, so this led to my question of how do differences in environmental conditions impact gremling and meroplankton settlement on a rocky substrate? All right, so I had two sites, Playa Jicote and Bajo Rojo. Playa Jicote was a coral reef in a bay. It had high pocillopore concentration, low surge, and pretty low algae concentrations, whereas Bajo Rojo is a rocky reef with high algae concentrations, high surge, and pretty low coral concentrations. On November 20th, I put out 8 rocks on each of these sites, and then over the next 9 days, I monitored for settlement in terms of morphospecies richness, and as well as percent cover. In order to do this, I took photos using an underwater camera. And with that, I was able to identify each individual morphospecies, and I also took notes on my dive slate while I was at the sites. And in order to calculate percent cover, I used a 15 by 15 grid overlay over each picture. I counted each of the squares that contained over 50% of the square containing rock, and I counted that as a rock square. And then every square that had over 50% algae cover, I counted that as an algae square. And to determine algae percent cover, I simply divided algae squares by rock squares. All right, here are all the morphospecies I found. Just to give you a little bit of intel of what they are, morphospecies 1 through 7 are red algae. Morphospecies 8 is in phylum Annelida. 9 through 12 are in phylum Cnidaria. 13th and 14th are in phylum Mollusca. Then 15 through 21 and 26 through 28 are all brown algae. And finally, 23 through 25 are green algae. And for the different sites in Bajo Rojo, I found morphospecies 1 through 24, as well as 27 and 28. at Bajo Rojo, and in Playa Cote, I only found morphospecies 25, 26, and 2, which was really interesting, and I will get more into later. All right, I first graphed morphospecies richness across sites, and I found that Bajo Rojo had a mean of more than double morphospecies per observation than Playa Cote. On this graph, I have the x-axis is site, the sites being Bajo Rojo and Playa Cote, and the y-axis is total morphospecies. And each dot on the graph is one observation, and I counted one observation as one rock on one site on one day. So in total, I had 112 observations. Then I graphed percent cover across the sites by day, and I found that Playa Cote had a mean percent cover of over 20 times more than Bajo Rojo, which is really interesting. And again, on the x-axis I have site, and on the y-axis I have percent total cover. All right, then I wanted to see if there was a correlation between percent cover and morphospecies richness. And I got a correlation value of -0.28, but the p-value was statistically significant. So I was able to infer that as percent cover increases, morphospecies richness generally decreases, obviously with some exceptions. I finally graphed morphospecies richness by morphospecies across the sites by day, and this graph really just does a good job showing differences in richness through colors. All right, the first factor I looked at was surge, and surge has a complicated effect on larval and germline settlement because it decreases the laminar sublayer, which, as I mentioned, makes it more difficult for algae to settle, but it also can increase nutrient availability through increased upwelling. So I thought that was initially very interesting. Additionally, Bajo Rojo being in the open ocean has grounds for a lot more genetic diversity in gametes than Playa Cote being in a bay has a lot more localized genetic gamete diversity, which could be one factor causing it to have less morphospecies richness. Additionally, Playa Cote has a much higher coral population. So while coral obtain 90% of their energy needs through their symbiotic relationship with zooxanthellae, 10% is through carnivory of plankton and gremlins in the water column. So coral could be eating the larvae before it has the chance to settle. And interestingly enough, scientists actually think that this percentage of carnivory in corals' diets is actually increasing with ocean acidification. Finally, I observed something called algal monopolization, which is where in Playa Cote, I observed 2 species of algae really overtaking a lot of the substrate. In addition to decreased surge, allowing much more gremlins to settle, this could be because of less presence of algal grazing organisms and fish in Playa Cote, which would allow more of the same species of algae to settle and dominate a substrate. There were some limitations. I had only 2 sites. It would be really interesting to do many more sites to get a more comprehensive kind of feel of how variables impact settlement. And also, I would be really interested in studying, doing a longer term study, maybe even like 5 years long, to see how communities changed after that initial biofilm was developed. I think that would be really interesting, as well as seeing temporal and seasonal changes in settlement. And of course, there's some difficulty in underwater identification. Thank you, Frank, Leo, Fede, Naomi, Emmy, everyone, Coiny Keel, host mom. Thank you all. Literature, thanks for listening.
Yes, Monica.
You mentioned like 5 years for
That was kind of an arbitrary time period, but I was really looking at after the initial biofilm developed, especially in Playa Jicote, just because coral coral larvae are often cued by something called crustose coralline algae, and that takes a second to develop. And once that initial biofilm develops, it might have differences in settlement, once you already have that biofilm. So I really only monitored the very early stages of settlement. Hmm?
Was the depth similar in terms of where you?
Yes, that's a good question. It ranged anywhere from 1 to 3 meters depending on tide. But yeah, they both were around 1 to 3 meters, tide dependent.
Can you explain when you said the bay conditions would have lower genetic variability?
Yeah, just because in the open ocean, there's a million directions where gametes can come from, whereas in the bay, it's kind of a smaller, calmer opening. It's getting less water flow, less in and out, and it could have preference towards more localized gamete selection.
Thank you.
Thank you.
Let's go.
Jensen? Here's the thinking.
Next speaker is Ewan Jensen
the
from
following.
UC Irvine.
This is what
He's going
people
to
are
be talking about sea cucumber diets and grouping patterns.
Hello, everyone. My name is Ewan Jensen, and I will be talking about the diet selectivity and grouping behaviors of Holoturia lubrica. A little brief introduction into this genus. This genus in Costa Rica, Holoturia, is composed of right around 13 species in the ACG alone where I studied. And a big reason why I studied these creatures was because Holoturia lubrica in specific had no direct information on its diet or anything about its predation, anything of the sorts. A little bit more background. Research about the diets of these creatures are mainly subject to only the creatures that are commercially viable. So a lot of these creatures are consumed globally, and it's drastically decreased their overall populations over time, and a lot of them are getting close to endangerment and almost extinction. Holoturia consists of around 120 species. As I said, Costa Rica has a lot of them, but the ACG has 13 of them. And sea cucumbers, a broad description of them are they're detritivores. They mainly feed upon sediment and sand, and they will filter it and take off organic matter that is trapped upon the sand. And they have many functions that benefit us and the world. They act as slight buffers for ocean acidification, and they also bioturbate a lot of ocean sediment to allow oxygenation. A little bit of how to identify these creatures. There's two main ways that I used to identify them, which was the calcareous ring at the top at A, right here. And then these are all ossicle formations that are used to identify them, that are all species specific. So that led me to my central question of, does Holoturia lubrica exhibit sediment selectivity, and do they have different grouping preferences based on microenvironment differences? These are my two sample sites. Islita on the left here is a little island peninsula location that has a lot of different tide pools, and it is a public beach. And Playa Jicote on the right is it's a private beach, and it's a cove with two sides of rocky substrate. Methods of identification I used for these creatures was ossicles and appearance. Ossicles on the left, these top two are my ossicles that I found from dissecting a H. lubrica species, and the bottom two are from another study that we used to identify them. And they're mainly identified by this kind of C-shaped ring. And then on the right here is all the different color differentiations I saw between adults and juveniles. Colors varied in kind of a brown color, and they're mainly identified by these spots that are along the back of the adults. A little bit more about my methods. 58 species, or 58 specimens were found all under rocks for my study. The trials of my group ranged from 4, 8, and 10. A little bit more about what I did was trials were run for 2 to 4 hours with 15 to 30-minute increments in between, and they were separated into 2 categories based on rock presence or rock absence. Fecal samples were then collected after my grouping study had concluded, and these were all looked at under a microscope and categorized into 6 different categories. One was micromollusk, another one was larger pieces of mollusk, spicules, foraminifera tests, microplastics, and other organic matter. And these fecal samples were then compared to the same categories in the sand found next to the Holoturia lubrica colonies. So into my results, a total of 1,720 pieces of substrate were counted by me in feces alone. And A is kind of an example of, on the left, we see a larger piece of mollusk, and then on the right is an example of a micromollusk. B is a little bit of a closer up picture of that micromollusk. C, it's kind of hard to see, is a sponge spicule. D is a foraminifera test. E is a microplastic in the middle, very hard to see, it's kind of a bluish color. And then on the left is a kind of organic piece of wood. And F was really shocking to find, which was an entire ant in their feces. So into my data, the y-axis here is the mean count per sample of the said substrate, and this is substrate type. This is not comparing each site, this is just showing the difference between feces and sand. And the main ones to look at is micromollusk. We can see a ginormous increase in feces of micromollusk in Islita and Playa Jicote compared to sand. This is kind of the stats. All we really care about right here is significance, and we can see that significance was only found in micromollusks, spicules, and foraminifera tests between both sites. A little bit more about site comparison. This is just between feces, this graph alone. The x-axis is substrate type, and then the y-axis is mean count per sample. And we can see that the one significantly different category was micromollusk. And lastly, the grouping tests that I ran were mainly significantly different by a large margin between grouping patterns between no rock and rock presence. So a little bit of discussion about my selectivity and diet selectivity studies. That significant increase in micromollusk foraminifera test and spicules in both sites compared from feces to sand substrate shows a presence of selectivity between, or in the diet of H. lubrica. And these papers kind of go into other more studied sea cucumber species. And this one mainly talks about how a lot of these species will sometimes go for more beneficial sediment with more organic matter in the presence of it. And this one just kind of talks about how the different shapes and sizes of the tentacles of these creatures leads to a selectivity in different substrate types. The differences between sites were mainly due to sediment absence, and this suggests that H. lubrica does have adaptability to their diet based on location. And this just talks about 3 different species that had 3 different site locations with the same categories that they were eating, and all had different abundances of each categories. Into grouping, significant grouping activity when rock substrate was not present. So this meant that these kind of groups would form between the different 4 to 8 to 10 groups, and they would be these kind of clumps when there was no rocks. But once rocks were placed into the tank, 70% of the study group moved onto the rock and were kind of under the rock in this kind of formation. And this paper kind of talks about how artificial reefs and a different type of sea cucumber will prefer to have a 30-degree angle of their said environment and to be on a rough substrate rather than a smooth substrate, which supports my rock activity. And this paper kind of talks about how crevice is beneficial to the fitness of overall sea cucumbers, and it increases their overall sediment intake when they're in a kind of rock crevice. And lastly, microplastics. This was kind of a side part of what, a side effect of my study that I found. A total of 40 pieces of microplastics were found in all different samples, and an average of 2 per sample of each feces sample was found across all sites, which was shocking. And these studies kind of show how microplastics in another species, the kind of big brother of Holothuria lubrica, Holothuria scabra, which is a very commercially viable species, had 89% of all samples had presence of microplastics and around 2 per sample, so supporting my study. And this is just kind of the harmful impacts that microplastics can pose on Holothuria as a general. It can destroy their gut microhabitat and all sorts of things. So that leads me to a little bit of conclusion, and I would hope to kind of investigate more into this microplastic issue, because as we produce more plastics in the ocean, we can see that a lot of these different effects can have on Holothuria as a genus. And I want to say a massive thank you to Leo, my primary advisor, for giving me so much guidance on Holothuria as a genus in total. Federico is my secondary, Frank, my host family and host brothers below that, and the rest of EAP. Thank you.
I saw that you compared two different site locations. Was there any point to the experiment where you considered an additional site location? And if so, how did that go?
Yeah, I mean, I considered doing Bajo Rojo. It had a different species of sea cucumber that I wanted to compare to, but in terms of time constraints, I couldn't implement it. Yeah.
When you were comparing like the depositions of like what was being deposited in sand, like what was the point of that? I was just trying to understand that.
Which graph are you referring to?
It was like earlier on.
Like this one?
Yeah.
This is kind of referring to feces versus sand samples. So I took sand samples from around the locations where I found them in the natural environment. Mm-hmm.
What was the commercial viability of the sea cucumbers that you were referring to?
Yeah, a lot of these sea cucumbers are exploited globally for certain purposes in other countries for consumption, for proposed health benefits, but I don't know how true those are, but yeah.
How many individuals were you able to dissect, and did those dissections teach you anything other than specific?
Yeah, so I was able to dissect 2 specimens. The first one, I did a gut analysis of it, and it showed a little bit of differences. I didn't want to dissect a lot of different specimens because it's just not the most ethical study. But I would just say that it supported some of my gut content, or feces content, but it did support another study that I found with more larger pieced mollusks within the gut content.
Thank you. Leo, are you happy to change frogs for sea cucumbers?
So happy, Leo. Thank you. Oh.
It's okay. I'm like, I don't know what to do.
The next speaker is Kira Gray. She's from UC Santa Barbara, and she will be talking about marking and tracking sea urchins.
All right, as Frank said, my name's Kira Gray, and I looked at marking and tracking the displacement patterns of sea urchins in Guajini Keel, Costa Rica. So what are sea urchins? Sea urchins are part of the phylum Echinodermata, and as we just heard from Ewan, sea cucumbers are also in this phylum, as well as sea stars. They are marine invertebrates and they're grazers. Grazers are a really important component of an ecosystem, and as we just learned from Lainey about coral and algal settlement, urchins can help control algal populations by grazing and leaving open space for coral to settle. They also have a decentralized nervous system, which means they don't really have a brain, but that doesn't mean that they're not actively responding to things. They respond to light, to different touch around them. They are smart for not having a brain. This is the species that I studied, Diadema mexicanum. They exhibit this aggregation behavior in which individuals will cluster together. For the purposes of my study, I counted urchins as one aggregation if their spines were touching. And here's a close-up shot of a Diadema mexicanum. It is believed that they hide or aggregate during the day and then at night explore to forage. This is believed to be to avoid getting predated by predators who are diurnal or active during the day. Two studies of interest in my initial research looked at a random walk model. And so this model compares the behavior of urchins and their movements to a model that effectively models dispersal of gas through a room. And so the initial study concluded that their movement is primarily random. It's no different than if gas molecules were to disperse through a room. That's how these urchins were moving in their study. And an additional study assumed random movement and then mathematically proved that they could replicate the patterns that urchins display in nature. So all of this to say that they move randomly, maybe, but there's definitely motivations happening. So where are they going and what are they doing? First question in this study, what is an effective way to mark and track urchins? And second question, what are the displacement patterns of the urchins? As far as methods, I selected 3 sites around Kwajini Keel in the Pacific Ocean off the coast of Costa Rica. First site is Bajo Rojo, second site is Playa Jicote, and third site is Islita. You've heard of all these sites in some of the other presentations. Bajo Rojo was a more exposed site with more wave action and surge. Playa Jicote, you can see this peninsula keeps it more sheltered, and Islita was a little more exposed over here. At these sites, I would snorkel. We would snorkel. I had a lot of help with this. And we would find aggregations. We would select an aggregation, and then I would collect the urchins. And I would mark the aggregation with a painted rock that was numbered. Once I collected the aggregations, we would swim them back to the boat or to the shore. This is one of my lovely assistants. And at the boat or at the shore, we would mark them using beads, same beads you would make friendship bracelets with. And these beads we would fill with either plasticine, like a modeling clay, or super glue. And then we would apply the beads to the spines. We applied the beads to the spines with tweezers. It's very easy to get spined when working with urchins. And as you can see here, the urchin is responding to this stimulus. So again, for not having a brain, they think a lot. Here's an example of some of the beads used. The beads were all different shapes and colors, and so that allowed me to track individual urchins and come back to the same urchin over and over and again. And once marked, we returned the urchins to the exact same spot of the aggregation, thanks to that painted rock that I had left, and left them for various amounts of time, ranging from 7 hours to 24 plus hours. Once returned to the site, I looked for my marked urchins. Here's an example of a marked urchin in a larger aggregation. The rest of the aggregation isn't marked. This one is unique in this aggregation. So I would measure the displacement from the origin, which was somewhere up here, with a transect to the urchin, and then I would take a compass heading to determine the direction of displacement. So from all of this data, I brought it back to Monteverde, and I sat and I processed it, and I sat in R, and I coded, and I coded, and I kept coding, and there was a lot of coding, many hours at the computer, But from the displacement distances, the time intervals, and the headings, I was able to calculate the rate of displacement between each observation, and I was able to recreate their movement patterns make little maps of their movements. So in total, we marked 93 urchins, and we rediscovered 53 of those urchins. That's a 57% rediscovery rate. We found 11 lost beads, and a lost bead is just a bead not attached to an urchin. By site, at Islita, we recovered none of our urchins. They all disappeared. At Bajo Rojo, we rediscovered 20%, and at Jicote, we rediscovered 75%. Pretty early on, we saw this rediscovery rate much higher at Jicote, and so we invested more time in marking and tracking there. The higher wave action at Bajo Rojo and the more traffic, more tidal action at Islita likely contributed to this. Islita and Bajo Rojo also are a lot more rugose and textured, and so the urchins have an easier time hiding, and we have a harder time finding them. The bead by itself was not retained on the urchin spine, so that's why we resorted to plasticine and superglue. With plasticine, we had a 44% rediscovery rate, and with superglue, we had a 76% rediscovery rate. With superglue, we had one instance where the superglue cured, and one instance where it did not cure. When it cured, we had a 91% rediscovery rate, and when it didn't cure, we had only a 63% rediscovery rate, but both are still higher than the plasticine. This here is an example of a bead that was lost that we were able to recover. This is an example of a bead that was lost with the entire spine still intact. And so this bead was applied with superglue. Because the entire spine is still intact, we think this could be a case of autotomy, which is when an animal or an organism voluntarily removes part of their body for survival. We also found instances of broken spines with the bead still attached, and former EAP students have noted that urchins may break spines to remove some sort of irritant, like perhaps a bead. So our rediscovery success over time, longer intervals between observations meant less success in rediscovery. We had a 24% reduction in tracking success per hour between observations, so the longer we waited, the less we found. In future studies with more regular observation, you can note where the urchin was and check a smaller perimeter of where they might have gone. With longer periods of time, you're not sure how far they could have moved. Urchin movement rate. We had an average rate of 11 centimeters an hour, a minimum of 0.74, and a maximum of 36 centimeters an hour. This rate is not consistent through the time interval because it's just a measure of displacement and divided by the amount of time between each observation. Comparison of movement rate, we found the first step to be significantly faster than subsequent steps, and this is likely a flee response from the disturbance of actually marking them, and so they wanted to escape this disturbance. For future studies, this might be important to note to potentially drop this first step because it's affected by the disturbance. As far as displacement patterns go, here are some maps that I animated. So this is where they went. And so this black square is the origin where they began. And this timestamp here shows the dates and times over which they were observed. Each point, each color is an urchin, and each point along their path is an observation. And here we can see a scale bar and the displacement from the origin. Here are the rest of my sites and the rest of my maps. The directionality here is likely due to the texture of the reef. And so at this reef, there were some ridges that went this way, and they liked to hide along those ridges. So in future studies, additional data about habitat and substrate will help explain why they go where they go. So in conclusion, the best way to mark and track urchins is with superglue and with frequent observation. We didn't see a definite pattern in the urchin displacement, which maybe will support the random movement hypothesis. I think looking at urchin movement on a large scale and a fine scale is necessary to really understand what's going on. And Thank you to the Monteverde Institute, Estación Biológica Monteverde, ECAP. Thank you to Frank for advising me through this whole process. Thank you to Leo for marking 93 urchins and spending 50 hours looking for them. Thank you to Guayaquil Diving Center for shuttling us around and keeping morale high. Thank you to Flor for cooking the best food in Costa Rica. And thank you to Taryn for braiding my hair every day. And that is all.
So you had a rediscovery rate of 100%.
Mhm.
How do you discover your data?
With more data points, I would have liked to compare my graphs to the random walk model. I think this data is not sufficient to compare to a model, but with more points, it'd be really interesting to see how it compares.
Did you find any groups that kind of would move and then stay in one location for a long period of time?
There was one particular urchin at this location, and it liked to stay in this one rock crevice, and it kind of hung out here for a while. And there was one urchin at, I believe, the previous location. This location, yes, that actually returned to the original spot of the aggregation on the very last observation day. So yeah.
So how many data points would you need to generate a random walk model, and how would you redesign your experiment in order to achieve that?
I'm not sure of an exact threshold for a model. I would think in the hundreds likely to get an accurate view. But I think other studies have used different types of tags like pit tags and other, like a transmitter tag would be fantastic. Then I would get consistent data and I could look at step length and turning angle and then throw that into a software to determine behavior patterns. Yes, yes, there's like something going on there. They're moving in response to something, so yes.
Did you tend to see when you revisited these urchins that they go to, like they just settle on their own alone, or were they like in different aggregations, or did you see a pattern just visually?
Yeah, so I did take note of the number of urchins in each new aggregation. A common number was 8 that they happened to group in, but I also found them alone and I found them in groups of up to 36. Usually within, I did a circular search pattern, so within that radius, yeah.
Thank you, Kira.
Here's the thingy.
water between rocks. So my methods kind of wordy, but I took observations from the 18th through the 24th of November, and I did 5 days at each site, so Bajo Rojo and Playa Cote. All observations were taken between low to medium tide in order to retain consistency. I sampled each site with 3 5-minute or 15-minute trials per day that were separated by 15-minute intervals in order to assess trends across repeated disturbances. And those disturbances were myself. I approached in a standardized, threatening manner, typically with my arms up. And I stayed within roughly or on the 2-meter line from the shore. So I recorded crabs that were between 0 and 2 meters and 2 to 4 meters from the active tide line. I categorized each crab with one of 4 escape behaviors or responses that I noticed. One was that they would hide into a crevice or take refuge. Another one is that they would jump to another rock. Next is that they would enter the water to escape from me. And the last one is none or unresponsive, which in itself is a response. So for my data analysis, I observed a total of 1,767 total responses across the 10 days. I had 991 at Bajo Rojo and 776 at Playa de Cote, meaning I saw marginally more at Bajo Rojo. However, from the distance, I found 1,361 between 0 to 2 meters from the tide and 406 between 2 to 4 meters. So I found a lot more, or roughly 3 times more between that first section. So they like to be closer to the water. I used the data to compare location variability, tidal distance, and visualization, and all visualizations and tests were done in R, similar to. So first is for location variability, I used Fisher's exact test to compare the responses between sites. Bajo Rojo is in red and Playa de Cote is in blue. On my x-axis, I have the response category, and on my y, I have percentage of total responses. And this is both distances combined. So I found that the crowds at Bajo Rojo more often used crevices than those at Playa de Cote. And in contrast, Playa de Cote often, the crowds will jump to other rocks much more often than those at Bajo Rojo. I found a marginal increase in unresponsive behavior at Bajo Rojo compared to Playa de Cote. And in water escape, I did not find a significant difference between the sites. So next is tidal distance, or difference in like between 0 to 2 meters and 2 to 4 meters. I did a Fisher's exact test again in order to determine the difference. I found that between 0 to 2 or 2 to 4 meters, the crowds were much more likely to go into a crevice than those within 0 to 2 meters. I did not find a difference in jumping to other rocks or hiding in crevices. However, from 0 to 2 meters, the crowds would enter the water exclusively, with none in the whole trial period entering the water after being 2 to 4 meters away.
It says hibernating.
Just went in.
El de él tiene que estar rojo.
Distance. Finish this. Okay. And the last one that I looked for was habituation, which I used simple linear regression to look at how the percentage of non-responsive crabs differed between each trial, which is the 15-minute sections that I did with 15 minutes in between. And I labeled Bajo Rojo in red and Playa Cote in blue again. I found an average increase of 17.6% per trial, meaning that each trial, it was steadily increasing for the amount of crabs that were not responding to me or were not moving when I had approached them. And to discuss, I found that habitat structure does drive site-specific behavior, specifically with refuge availability. So finding that more crabs seeked crevices at Bajo Rojo than Playa Jocote was because they were more readily available, like I had said before, and it is the lowest energy cost option. And at Playa Jocote, because those crevices are not available, jumping to another rock is an intermediate response to make it to a crevice because there's not one available for them. I found that tidal position also influences escape choices, mostly in terms of entering the water. Because I found no crabs 2 to 4 meters that would go to water, meaning that they do not seek it out, but if it is there, they will use it. The next one is the low reliance on water entry overall between sites and amongst the distances, likely meaning that the predators in the water, which can include more eels, octopuses, or carnivorous fish, are prevalent, and also with the crabs staying out of the water most of the time, meaning that even when they would jump in and I would observe them, they would quickly come out. Also, I did find significant habituation across repeated trials with typically the third trial of the day. I'd have a lot of crabs just looking back at me and not moving. This could indicate the conservation of energy as well, to not, because the ecosystems change and they have so many diverse predators, if they see me as not a threat, it could, like over time, they could get more used to me. And this is also shown with the literature saying that crabs do have a short-term memory and they can understand repeated disturbances. However, I did not see habituation across different days. So when I would come back, it would reset. So yes, I'd like to thank Frank, primary advisor, Emmy, my secondary, all of the other staff members, Dive Center, staff of the Estacion, and my host family. So, thank you. Yes. So what I had read is the decision that they make is the first one or whatever is most convenient. So with between 2 and 4 meters, I did not find any going to the water, assuming that in that distance there was something else that would be either a crevice or another rock that was more, I guess, like a better option. So yeah. Yes.
So obviously you make a very menacing kind of stimulus, but do you think seagulls resemble one of their more natural predators, like the raccoon, that they exhibit less habituation over time?
I don't know. I was also considering looking at habituation in different sites because Bajo Rojo would not have any terrestrial predators. So that would be an option, but I'm not sure how different it would be. I would go to the same locations in the areas, but I wouldn't. No. Yeah. Yes.
Did you notice any active predation other than yourself on these crabs?
Yeah. So we saw a lot of the crab-eating hawks or common black hawks, especially in the Maya and around, as well as I had a good time finding octopuses, and we saw eels as well. So all around. Yeah.
Do the crabs have spots they prefer to hide in? And do you notice them like congregating around there, or do you like try to focus on areas where they would have?
So one of my observations was that they were commonly in groups and would be together in these locations. I also noted on the data that I took, but I didn't include, the first one to respond to me in a group, so, to look at and see if like the first one responding to me was responding to me and other ones were not. They were responding to the other crabs. But it seemed like they were in groups in protected, like sheltered areas. Yes. So when I left 15 minutes in between, when I would come back, they would be all over again. So it was, I think, a good amount of time to leave. Yes. I don't know. Especially in Kinikil, they are not typically eaten by people, but fishermen will use them as bait. So that would be an interesting thing to research.
Thanks, Scott.
Thank you. We're not gonna switch then?
No.
Aaron is not gonna go first?
No, we're just gonna switch this.
And then we'll switch.
Remember just to. We're not gonna take this out. Okay. We're just gonna, yeah. I'll just put it down here, okay? So, this out. This one. This one. We, you on this side or this side? Just while we're getting set up, just to give you a quick heads up, we are going to skip the next break and also skip lunch. That's a joke. Everyone is invited for lunch, and we're hoping to have that right at 12:30. So we're going to start the next section after our two next talks about other sea life, and then at 12:30 we will interrupt that session and have lunch. Okay?
I think. Wait, it's okay. I can just press it.
When you're speaking, make sure that the mic has a little green light on.
Hello. Oh,
The
okay.
next speaker is Sydney Kwan, coming to us from UCLA,
Okay.
and she's going to be talking about octopus chemoreception.
Okay. Hi, my name is Sydney Kwan. My study focused on the chemoreception, behavior, and individuality of octopus mimus. So firstly, octopuses are in mollusca, they're cephalopods, and they have a very distinct lineage from humans. However, they display convergent evolution in things such as the development of camera lens eyes, just like us. Their diet consists of crabs, fish, bivalves, gastropods. Our octopuses specifically loved clams. They also have abilities such as camouflage, jet propulsion, ink, and many others. And their primary three senses are vision, touch, and chemical sensing. And they usually utilize a combination of all three for survival, exploration, etc. And octopuses have up to about 500 million nerve cells, most of which are in the peripheral nervous system, which means that there's more emphasis on sensory information coming in from their arms and their suckers. And their suckers have abilities such as olfaction, touch, and adhesion to other objects. And interestingly enough, some stimuli encountered by suckers actually doesn't even reach the central nervous system, meaning it doesn't get processed by the brain. This is like reflex, as in when they encounter a stimuli, they can react faster because it doesn't have to go through their whole central nervous system. And some studies show that proprioception, which is the awareness of body position and their arms position, also doesn't reach their brain sometimes, which means that to some degree, octopus arms can act of their own accord without direct control from the brain. And their suckers also specifically utilize chemoreception in prey detection and also just exploration in general of their surroundings. So this leads to my central question, which is, how do octopuses utilize and react to chemical cues in the absence of visual cues? So I wanted to look into chemosensation or chemoreception and seeing how that aids their survival, their foraging, and looking at their behavior in terms of how they're going to react to foreign objects, and then also investigating the relationships between their three primary senses. So methods, we went to Bahia Tomas for 12 days, and we located octopuses in their dens every day with the help of some Guajinikil locals. And so I set up GoPro camera, recorded for about an hour, and I would test the water current and attach floats to signify where each den is so I could come back and find it. And I used these plastic film canisters, which have holes in them to allow chemicals to escape. And some of the canisters, the bait canisters, are filled with these live clams, which is their favorite food, and some of them just had rocks in them, and that was served as my control. And this is an example of my experimental setup. So basically, these are my two canisters. This is the bait canister, and the octopus chooses to grab onto it, put it under itself, and put it back. And this is the control canister. I don't know if you can see, but there are holes here, and that's where the chemicals can come out. So some of my results, I looked at the first interaction with canisters. And a canister interaction I define as an intentional touching of the canisters with the suckers facing the canister, since that's how they receive chemicals. And I had a total of 28 octopus videos, and 12 of these octopuses physically interacted with the canisters, and that's what I have on my graph. So 8 of these 12 interacted with the bait canister first, 3 of them interacted with the control canister first, and 1 actually touched both like in the same arm movement. So it was kind of investigating both at the same time. And I got a p-value with a binomial test of 0.12. And then I also wanted to look at if there was some sort of significance with the length of time of the interactions with the canisters. So these canister interactions are in the order of increasing values, and the purple diamonds are bait canisters, and the blue circles are control canisters. So as you can see, there's a pretty wide range of interactions. It's measured in seconds. This top one was, I think, 463 seconds, and some of them were like 3 seconds. And the average for bait and control were both pretty similar, actually. And it's really interesting because there's this like barrier between 25 seconds and 180 seconds where I recorded no data points. So all of the octopuses that interacted with the canisters either interacted them for time below 25 seconds or above 180 seconds. And it's also interesting to see that for the higher section, 4 of the interactions are with bait and 2 of them with control. And my p-value was 0.9 for this graph. And then this is another table, and this one actually shows 4 individuals that interacted with both bait and control canisters. So I wanted to see if there was some sort of disposition within each individual octopus to see how they interacted differently with the canisters. And so there are only 4 individuals, but it does technically have a general correlation between these 3 on the bottom. These individuals all interacted with the canisters for less time, while this one individual on the top, number 2, interacted with both canisters for far longer time. And so both the maximum values were just from one individual. And this, I don't want to draw any definite conclusions because it is only 4 octopuses here, but it's interesting because it could imply that this one individual in particular had some sort of reason or motivation to interact with the canisters for longer. And then some more observations. Each octopus really interacted with the canisters very differently. I couldn't predict like what they were going to do with any of these, if I was going to see them or what they were going to do. So they had behaviors such as tapping and nudging the canisters, just staring at them, fully picking them up, moving them, pulling them closer to each other, enveloping under their bodies, siphoning water and sediment at them, or putting their arms inside the canister. And this is one of my favorite observations. There's actually 2 octopuses in, this was a video. There's the white octopus, which is in the den, and it actually saw the pink octopus, like the visiting octopus, interact with both bait and control canisters. And then as it passed the den, the white octopus like lashed out and like attacked the pink one. And you can actually see the arm in the left picture is in the canister while it's getting attacked. And then later, this white octopus also interacted with the canister after watching the first one do so. Okay, and so my discussion, there is a general tendency towards the bait container for first interaction because 8 out of the 12, or 66% of the octopuses did interact with the bait first. However, because of my p-value, this first choice interaction is not significantly different, although it's close. However, previous research does support that octopuses use chemosensation, chemoreception, to detect their prey items, and that first touch behavior doesn't necessarily mean that that's the food that they're going to eat, or that's like a commitment to do anything. It just is an exploratory behavior, and there is like random touch behavior that people have seen before. It's just them being curious and exploring. And then also potentially there was contamination, cross-contamination between the canisters with chemical compounds. Plus the rocks could have had chemicals that could have been interesting to the octopuses. Plus octopuses just have general curiosity towards foreign objects, so they might have just been interested in the canisters themselves and not necessarily the chemicals that are coming out of them. And then also what was really interesting is seeing all of the variety of interactions with each octopus. Some of them were just staring, didn't physically interact, siphoned water at the canisters. One of them actually did it to the camera too and touched it. This is actually not an octopus touching the camera here. Yeah, and then the interaction lengths were pretty varied too, which is interesting to see. And this also is supported by previous research that shows that personalities and individuality is characteristic of octopuses, and they have different strategies for foraging and figuring out tasks, which actually is evolutionarily beneficial to their species' persistence and dispersal and their predator-prey relationships. And they also have been shown to learn from observations of other octopuses, such as my video with the two octopuses, them learning from each other to do certain things, learn certain things. But yeah, so octopuses in general do have a lot of curiosity and display individual behaviors. Thank you so much. These are my references. Thanks.
Time for questions.
In the time graph, you have that p-value of 0.9, and I know that's not significant, but what is that actually showing? What do you think that means?
Well, it's showing that the between the bait and control canisters, there isn't a significant difference of like one having, like for instance, it's not like the bait canister had a lot longer of time interactions versus the control. It basically, having a high p-value shows that there was a pretty wide range of lengths of interactions between the canister types.
Yes.
If you were to do this study again, like what would you do differently, or like what would you kind of like expect to see?
Yeah, if I were trying to really hone in on the chemoreception of the octopuses, first of all, I would want to more solidly reduce the cross-contamination of the canisters and also like sterilize the rocks or use some sort of other object that I know doesn't have any chemicals on it. Plus, I also think it would be interesting, for my study, I found almost different octopuses every single day. So I think it would be also interesting to focus on a smaller group and retest them because you can see more of their personalities there and learn more about the individual octopuses since that's such a like variable factor. Plus, some of the octopuses, they all were different obviously, but I actually expected some of them to be able to open the canisters, but I tried smaller ones. Is probably a factor that into my study, but yeah, definitely relative age and like more life experience. Definitely.
Thank you, Sydney.
Sí, por eso.
We're gonna stop like after maybe one fish. Maybe not.
Do you know if the video will play by itself?
If you,
It's in there?
yes.
Okay. Okay. If not, I can just use this to press play.
Yeah, yeah.
Okay.
If not, then you look for it.
Okay. Okay. I can do that.
It's like right here.
Sounds
Only one
good.
that's open.
Got it. Got it. Wow. So
Our next
fancy.
speaker is Taryn Hosnick from UC Santa Barbara, and she will be talking about fish following octopuses.
Okay, hi everyone. My name is Taryn. I'm a third-year marine science student at UCSB, and I'm going to be discussing my findings on octopus-fish foraging interactions, specifically between Octopus mimus and Serranus piscitacinus. Okay, first, I know this session's going a little bit longer, so I'm going to do my best to grab your attention. And I also did want to say that all of the pictures and videos that you see in this study were of octopuses that I studied and taken by me. Okay, first I want to introduce you to two key species. Sydney already talked about Octopus mimus, but I'm going to refer to it by its common name, gold octopus, and the barred serrano, which is a fish in the family Serranidae. The gold octopus is often confused with Octopus vulgaris, which is a species subset called the common octopus, and this leads us to not have super definitive research on octopus. I only found one peer-reviewed paper on it, and it didn't have to do with its behavior in the wild. So in terms of octopuses as nuclear species, there is previous research done showing that fish follow octopuses, specifically wrasses, goatfish, sea basses, and groupers. However, it's still a relatively novel concept. I would like to point out when I was originally pitching this project to Leo, he was a bit hesitant because even a taxonomist as great as him doesn't know everything. Oh, sorry. I mean that some things are still new and not everyone knows about them. But it's a great help though. My initial question did ask then, do fishes follow octopuses when foraging? Because again, I wasn't sure. And here is an octopus in den 2. This one was a favorite of mine. So in terms of methods, this is my main site, Bahia Tomas. It's characterized by being a rocky reef with lots of algae-covered boulders and Poeciloporus species, which is a type of coral. I did also look at Isla Muñecos and Bajo Rojo, but did not observe foraging there, and it was a lot more difficult to sample those sites because they're less shallow. Okay, for my methods, this is a very simple version, but for the majority of observations, the first step was to find a den with an octopus inside. This was done by looking for middens, which are heaps of shells. Octopuses, particularly these ones in Bahia Tomas, seemed to eat a lot of clams, and so they eat the clams in their dens and discard the shells outside of their dens. It's a clear indicator that an octopus den is there. Or sometimes I would use barred serranos to see if octopuses were nearby. Then metadata was taken. I have a plethora of metadata, including algae cover, coral presence, initial fish presence, midden composition, and whatnot. I deployed a float and a GoPro. GoPro stayed there for about 30 minutes to an hour, depending on GoPro battery life. And then I swam away, and I did not get anywhere near the octopus until the session was completed. I collected the GoPro and analyzed the data. And in terms of the types of data I collected data on the presence and behavior of follower fish, foraging trip lengths, and then intimidator fish interactions. I have a total of 41 observations, 18 unique dens, and 3 of those observations were done with me with my own eyes and not a GoPro. Okay, in terms of results, this is the main question, answering the main question I sought to answer in terms of fish presence. We have 20 foraging trips across 9 unique dens. I would also like to mention I wasn't able to specifically determine if they were different octopuses because I didn't interact with them, so that's why they're referred to by den IDs. So we have 22 instances where there were yes follower fish present and 6 where there were not. This was statistically significant. I used a 2-sided exact binomial test and Bayesian analysis to determine this. In terms of the ones that had no follower fish present, that was 6 instances. Those were GoPro videos where I did not see it with my own eyes, and that just meant that there were no follower fish leaving or returning with the octopus. However, when I observed 4 foraging trips myself, I noticed that although fish did not leave or return with the octopus, at times they would take note of the octopus foraging and start foraging it with it when it had already left its den, meaning that fish presence can fluctuate during the trip. So it is possible that those 6 instances with no follower fish present did actually gain follower fish. Here is an example of the follower fish following the octopus. You can tell it mainly follows the arms. That's what it's focused on, since those are the parts that are flushing out organisms. Yes. This is O2 in den 2. And then here, the fish, you can really see how they kind of attack the material very rapidly and begin to take note of it foraging. But yeah. Okay, in terms of foraging trip lengths, they were short, averaging at 7.6 minutes. This also further supports that these are Octopus mimus instead of Octopus vulgaris, because previous studies show that during the day, Octopus vulgaris would take foraging trips averaging at around 2 hours. Potentially, Octopus mimus takes shorter foraging trips during the day. From this data, though, and the behavior of their foraging, I was able to kind of get to know the personalities of these octopuses better, which leads me to believe that it is the same octopus in each den throughout the duration of my study, which was 2 weeks. For example, Octopus 2 was in that big coral head, liked to leave a lot by swimming, whereas Octopus 4, or O4, liked to leave its den cryptically by crawling on the ground. And O13 was actually the only octopus that would leave its den in my presence, and I observed all foraging trips with my own eyes from that octopus. This is a flow chart. Now we're talking about intimidating fish species. This wasn't something that I sought out to discover, but did. So we have the octopus in a neutral state, and then an intimidating fish or observer approaches. No physical interaction is needed. And we have 5 main responses: color change, ink release, defensive disc, jetting away, and contact with intimidator. The most common one was under color change, and it was transforming to a stark white modeling. You can see that in the top left. The progression of that is O13, and that was when I got too close, and it kind of hides back in its den. I observed 20 interactions with intimidating fish, 10 different species, and the most common ones were common silver belly and the long-nosed puffer, of which I have a video right here, where you can see the long-nosed puffer. That's real time. It rapidly swims towards the octopus who is foraging. You can also note the barred serranos following it, and this defensive disc as it moves back to its den. This is possibly due to the fact that both long-nosed puffers and octopuses prey on clams. So if the puffer thinks that this octopus is maybe foraging for clams and has some, perhaps it's attacking or being aggressive towards the octopus to try and get some of those. So the main conclusions that I have are these observations are the first documented instances of barred serrano following gold octopus. Barred serrano tended to target small, unidentifiable prey that the octopus did not show interest in, and they benefit without competing with the octopus. Oops. Couldn't have done it without them, especially Freddie and Juanca, who are the 2 ex-fishermen that helped us, by Tomas, and that's it. Yeah, so this was actually a huge thing that I wanted to really get right. So Octopus vulgaris is dubbed the common octopus. It's a species subset. It's all over the world, in like Japan, in the Mediterranean here. They lack, or science lacks, recent identifications of morphospecies within Octopus vulgaris, making it difficult to determine different types in different locations. So because of that, initially I wasn't sure if it was Octopus mimus, but Leo has a colleague with the Schmidt Ocean Institute. She's a mollusk specialist, and I worked with her to figure out if these were Octopus mimus. She said it's evident by the brown channels on them, and then it's hard to tell here, but they have crimson eyes with gold in it, and that's how we helped determine that it was Octopus mimus. Mhm.
So good. Did you typically see more than one serrano following the same octopus? So, and kind of connected with that, I was wondering, did you notice any response by the octopus or the fish or any reaction?
Too. Bigger one would try and like get rid of the smaller one and like swim at it aggressively, so it would be the only one following the octopus. But the octopus didn't interact with the barred serranos. They also didn't get in its way, so it was able to forage freely, and they kind of just took advantage of that.
Can you tell us if you think there's any benefit that the octopus gets from being followed by fish?
I didn't like necessarily study it, but I'm thinking it's maybe not great. Like like I don't think that if anything, if there's any impact on the octopus, it's not going to be a positive one, because they don't benefit from the barred serrano taking these small unidentifiable prey items that they don't care about. And then also because it's such a cryptic animal, it really relies on its camouflaging abilities. But if you have a bunch of follower fish following your every single move, predators are going to be able to tell where you are a lot easier.
So what about the idea mentioned by some brilliant reviewer that maybe there's a many eyes effect and those fish can also help detect predators?
Possible. I don't know. I don't know how smart barred serranos are.
Good news and good news. Lunch is going to be ready now. It is ready now. So everyone is invited. The menu is vegetarian rice and beans and delicious salad and the let's go. And the also great news is that we're going to try to inch back to the schedule and resume with another marine session at 1:15. Okay, so that gives us 55 minutes. Should be plenty of time to leisurely have lunch and talk about the wonderful presentations. Thank you, John.
Voy a revisar qué firman las otras. I know you were
Hi everyone, good afternoon. My name is Leonardo Chacon. I am here for moderate this session about fish behavior and ecology. And our first speaker is Alice Dodge, and she is going to talk us about the aggressive behavior of damselfish in some reefs in Kwajini Kill.
Thank you, Leo. All right, so my name is Alice Dutch, and my presentation is about the Acapulco damselfish and the giant damselfish's responses to a breviary on their algae gardens. All right, starting off with the Acapulco damselfish, or the Stegastes acapulcoensis. It's about 18 centimeters and is commonly found in the tropical eastern Pacific Ocean. Similarly, the giant damselfish is found in the same habitats and rocky coral reefs in this region as well. It sits at a length of 31 centimeters, so it's quite a bit bigger than the Acapulco damselfish. And they both express very territorial behavior towards any competitors for their algae gardens. So their algae territories look like this. This is one. You can see the little Acapulco in the corner over there. And this is its own little algae garden. All right, so what does it really mean when they're aggressive and territorial? They're aggressive by showing displays of chasing towards other herbivorous fish. For example, the giant damselfish typically chase other giant damselfishes and also other Acapulco damselfishes. However, and then also rainbow wrasses and blennies, as you'll see later. Meanwhile, the Acapulco only chase their own species and they don't chase the giants, which is important. They also bite sea urchins. As mentioned earlier in Kira's presentation, they can feel things, so we'll get into that a little bit later. They also weed out unwanted algae species. They're nicknamed algae farmers because they do little weeding on their territory to get rid of any other algae. The specific algae that they like. Oh yeah, sorry. One more note is that they exhibit these behaviors mostly during the day. They're diurnal, which means all of these are during the day, not the nighttime. So moving on to Polysymphonia. This is a red algae, which is red and grows very fast. It's very nutritious, and therefore it's widely eaten by other competitive species, such as urchins. They really like it, and so do the damselfish. However, these damselfish do not like it when other species prey on their algae gardens. They put a lot of work and time into it, so you can see here, the giant damselfish is getting really mad at this Diadema mexicanum by it typically grasps the spine with its mouth and tries to get it to go away. It's important to note that these are just very curious animals. I accidentally put my GoPro a little too on the site, and it was just kind of like going after it. Yeah, I personally have an affinity to the Acapulco damselfish because they're so small and mighty. Leading into my central question. So, all right. How do Acapulco damselfish and giant damselfish protect their algae gardens from herbivory? And a follow-up question, is there a difference in technique, efficiency, and intensity of aggression between the two species? So the sites I chose, like many others, is Jocote, Playa Jocote. At this site, what was most notable to me was that there's a lot of abundance of coral and a lot of Acapulco damselfish. Again, you can see this guy just looking at the camera, a little confused on what's going on. We also have Baja Rojo, which is surrounded by the open water. So that means there's a lot of surge. And I observed that there was a greater abundance of giant damselfish at this site as well, and lower density of corals. So for my methods and materials, I use slates to mark down all of my data. I also use tongs, which is a kitchen utensil, but I used it for this purpose of scooping up the urchin. I also used a glove to not get spined, and 2 hand tally clickers. I used these clickers to take down a lot of data points, but I'll just note the important ones. One was the urchin bite. So every time that the fish grasped onto the urchin spine, I noted with my right hand. With my left hand, I noted how many times it approached the urchin, which I'll further explain later. I also took note of every time it chased another fish. And I categorized this by every time it sped up visually and tried to chase another fish away from its site. in total. Oh yeah, and one more thing is my trials were 10 minutes. I cut it off at 10 minutes, even if the urchin was still on the site, just in the sake of time. In total, I took 42 trials from all the sites, including both species. So, on to my results and discussion. I found that there's little variance between the central aggressive tendency of both species. I created the Z-scored aggression index, which is basically all of the aggressive behaviors: the weeding, the biting the urchin, and the amount of times it chased fish. I found that there's little difference between the two species, which is to be expected. They're in the same family, and they both exhibit aggressive behaviors. So I made the data basically this bites per urchin event. I'll explain this right now. One urchin event happens when a damselfish swims to a sea urchin and bites it, and bites it, and then it keeps biting it. And in this case, it had 4 bites in one event. It ends when the fish swims away to do something else. So I did find that in both species, as the bites per urchin event, which means it bites it a lot in one event and swims away, decreases, the bites per urchin event increases, and the amount of fish chases tends to decrease with this. You can also see that I did say that they are both quite similar in species, the results, but right here, these 2 points are 2 instances in which the giant damselfish bit it about 80 times and only had 5 events. So those are 2 outliers that kind of skewed my results here. So I made another graph just to explain it further. So as you can see here, it exhibits this negative correlation, and the difference between species isn't very much. However, between the 2 species, I did find individual differences in technique of urchin removal, and I'll explain that now. So I categorize urchin removal as being not removed, which is just it being on the rock, or it being removed via a current. So for example, when the Acapulco bites it and lifts it with its mouth, then the current tends to take away the urchin, and that is the light blue in the middle here. The retreated is when the urchin is harassed enough times that it just kind of picks up and walks away. So I found that more urchins retreated from the giant damselfish attack than the Acapulco. This makes sense. I observed much longer trials for the giant damselfish because they did not have the ability to lift up the urchin with its mouth. I found it very interesting that the Acapulco, although much smaller, was able to lift it up with its mouth and kind of pluck it and throw it away. Therefore, the giant damselfish had to rely a little bit more on harassing it until it just scurried away on its own. And this is kind of a reiteration of what I was saying earlier. For urchin removal, the Acapulco displays faster time duration, and this is significant. So yeah, in general, the average of the trial durations for the Acapulco was faster than the giant damselfish. So in conclusion, overall, the total territorial aggression between species is similar. They both tend to chase less fish when biting the urchin more times. And the Acapulco damselfish tend to have shorter trials. The giant damselfish tend to make the urchin retreat more than the Acapulco damselfish. So a little bit more into discussion of this. Wait one second. It's really interesting to see this kind of, I didn't know about this before. And when seeing the sites and seeing the urchins aggregating right outside of the territories, it shows how much of an impact they can make on the coral reef communities. Because the urchins are algae grazers and tend to be grazing whatever they can find, they're opportunistic feeders. This means that when the algae is taken by all these damselfish and kind of mass produced, and it takes up a lot of light and space, that means corals cannot compete as well. And that also means that urchins have to compensate because they can't eat the algae that's on these rocks, protected by the damselfish. All right, so further studies, I would like to see the relationship between coral bioerosion and the presence of damselfishes around. Thank you. All right. Preguntas? Tommaso.
Do you know how the urchin spines affect the damselfish and if like they're sensitive to it and what the damselfish bite urchin does to the urchin?
Yeah, so I observed that when the urchin was bit a lot by the fish and I would like swim over to the site to see, kind of what happened, there would be spines littered on top of the algae. So they are able to like twist them off with their mouth. I don't know if this hurts the fish. I didn't observe like bleeding in their mouth or anything. And they do it a lot. Like different species of damselfish also do this. So I'm not sure how negatively it affects the fish. For the urchins, I do know that probably is not good because their spines get ripped off and they have to like leave and they don't get the food they want. Owen.
Were there any differences within the territories between the two types of damselfish? Like was the giant damselfish found in a certain type of location or depth or area of space compared to the Acapulco, or was there a pattern you observed?
Yeah, that's a great question. Most times, the algae territory of the giant damselfish was a lot larger, and it also had denser amounts of Polysymphonia, the red algae. I also found that they typically were a little bit in like shallower waters, like higher up, and then the like closer to the bottom where it has more like ability for competitors to kind of go on their algae gardens were the Acapulco. And I hypothesize this is because of like Acapulco not being able to compete and chase away giants for that optimal spot.
Sorry.
I'm wondering if you noticed that the algae gardens were only for one fish or if they shared it with maybe like damselfish or
Yeah, another good question. For the Acapulco, they like exhibit more teamwork behaviors. Sometimes there'd be a pair, a male and a female, on one site, and they would. I only observed this once, but they both took off the urchin like at the same time, and they're both like, ah. So that was really cool. But I didn't see that for the giants, so yeah.
Yes. Is there like a mechanical difference between the two species, the giant versus the Acapulco, in ability to like lift the urchins, or is it more of a behavioral difference, like the larger one being more just pushing versus their ability to actually lift the urchin?
Yeah, that's a great question. I was really confused by seeing this in person and why there was such a difference between the two species. My guess is, because I haven't seen much research on it, is that because the Acapulco is like really big and has like and it's known for being very aggressive, can continually bite it. Whereas the Acapulco, like I noticed at least, swims away and then it comes back really, really fast, and then it kind of latches on and is able to like pick it up. Yeah.
Alice, did you say for the first fish, you say just now Acapulco when you meant giant damselfish? Yeah.
No, the Acapulco, or maybe I did. I meant to say that the Acapulco are the ones that are able to lift it up and they swim away and kind of latch onto it.
We don't have more time for questions. Okay. Thank you so much.
Thank you.
spot to get that footage because we saw them, I was with you, we saw them for like 1 second and then they got scared and ran and they never came back to that rock. They're like super elusive.
I think it's just like being really patient and attentive to your environment and where you are, and it's just, yeah. I think it's also just luck. I was just out with Kim one day when we were out there, and we left the GoPro in that little crevice because we saw adult rock skippers like hopping around, and we just happened to get like really amazing footage of them. Yeah.
Sorry. Lovely talk. I wonder about interpretation of territoriality versus like mating figures and how one can distinguish if GoPro limited footage with the using.
Yeah, I was also wondering that. I don't know why they were displaying territorial behavior. It might have been because they were both males. Just like, yeah, it might have been because of a main reason, but it's unknown. Like, I haven't read anything about.
I guess my question was, were they both males?
It's a great question. I wish I knew. Yeah, I don't know how to identify them.
No, never mind. I was wondering, how would you identify male and female bunnies?
Go ahead. How about this? Me?
Yes.
Okay, yeah, I was asking about the territorial behavior too. It seems so bizarre to me that they would be territorial on land, which you don't expect to be fish territory and not in the water. So I was wondering if there are any ideas out there about what the motivation for the territorial behavior would be, what kind of resource they would be guarding.
It could be due to, I also observed the adult rock skippers, like, and like little, like, I observed adult rock skippers and little rock bowls where they would go to every single day, and it would like be like their little homing behavior. So, maybe it was just because it was where it's like a place that they go to every day, and it's like their territory, similar to like how we have like a room and like, you know, something, yeah, but I don't know. It's just, yeah.
We don't have more time for questions. Thank you so much.
How's it going? Good to see you guys.
Thank you.
first, what are nursery habitats? So over here, I have an example of an ocean nursery habitat. They're relatively shallow areas that have rocks or other sources of protection. In this example, there's rocks with little crevices that the fish can hide in. There's also an abundant food source like the algae in this picture, and they're typically used by juvenile or fry stage fish. On the other side, I have a picture of a nursery in the mangroves. As you can see, the visibility is very low, and that provides a lot of protection from predators. There are also stilted roots on mangroves that go into the water that the fish can also hide in. The abundant food source in the mangroves is usually found in the mud or like floating around in the water. And they're also used by juvenile and fry stage species and have a relatively shallow depth. So a little bit about the fish life cycle. They start out as eggs and then hatch into larval fish. They can't really move around very much. They're very small and very vulnerable. Then they'll go through some ontogenetic changes, which are different shifts that the fish go through their developmental phases, and they move on to the fry stage. So they look a little bit more like what we think of as fish. They're able to move around a little bit more. They can feed on their own. And then they'll shift to the juvenile stage over here. Some fish don't have a defined fry stage, so everything after their larval stage will just be considered a juvenile fish. As juveniles, they are still using nursery habitats, or in fry and juvenile stages, they'll use the nursery habitats. Usually they'll look very different. They'll have a lot of physical differences in their coloration as juveniles. And then sometimes they'll also move from their nursery habitats into an adult habitat while they're still in the juvenile phase. Once they are adults over here, this is when they reach their full maturity. They're a lot larger. They participate in different behaviors, sometimes different schooling behaviors or feeding behaviors, and they're in their adult habitats. So a little bit about mangroves. This is a picture of the mangroves in Quahiniquil. There are 3 species present there: the red mangrove, white mangrove, and black mangrove. They have stilted roots to help with the changing tides. So this ecosystem is very, the tide changes a lot, so the depth will be very different throughout the day. The soil is also very anoxic, so there's no oxygen, but there is a lot of organic matter, which is some of what the fish will feed on. The mangroves are also filled with brackish water. They have fresh water coming in from the river and salt water coming in from the ocean. And for ocean habitats, here we have an ocean habitat at one of my sites. I focused on nearshore zones. So nearshore zones are also shallow in depth. They're more affected by waves and surge, so things will be moving around a lot. They're also affected by the tide, so some areas will be exposed, and then at other times they'll be covered by water. They tend to have, or most of what I focused on was rocky habitats like this. You can see there's lots of boulders in here, but it's also very common to have sandy or coral habitats. So after looking at all of that, it led me to the question, are there fish species that have nurseries in both the ocean and the mangrove habitats? So I studied at 3 different sites. I had 2 ocean sites and 1 mangrove site. For the ocean sites, I had Bajo Rojo and Jicote, which you've all heard a lot about. And then in the mangroves, I had 1 site near the town of Quahiniquil over here. So to identify nursery sites, I first looked for areas that had at least 3 juvenile fish that stayed there for about 10 seconds or more. And then I set up a GoPro and left it there for 10 minutes while it was videoing. I also left the area to try and minimize any disturbance, any sediment I might kick up, or just to not scare the fish away. During video analysis, I identified the species present in the video, what stage they were, and what habitat type it was. So I had rocky habitats, large rocky habitats, sand, coral, and mud. In the mangrove, I also used a sign net. So this is what the sign net looks like. One person stands on each side and drags the poles through the water. And then with the help of Leo, we put them in buckets over here, collected all the fish in these buckets, and then took them out and measured them, took pictures of them, and found and identified the species. So for my results, I found a total of 8 species in the mangroves and 43 species in the ocean. There was one species that was not present on the video. It's not on this graph, but there was a larval flatfish or a flounder that was found in the mangroves while using sign nets. In the ocean, yeah, there was a much larger amount of fish. And this is for juvenile and adults. So over here on the y-axis, I have all the different species, and then this is mangrove, and this is ocean, and the blue represents that the species was present, and if it's white, that means it was not present. So of the juvenile species, I found 2 that were present in both the ocean and the mangroves, and those 2 species were the schoolmaster snapper and the big-eyed trevally. In each site, I also calculated the ratio of juveniles to adults. So in Bajo Rojo over here, I have on the y-axis, I have percentage. So there were 51% adults present and 49% juvenile or juvenile and adults. At Jicote, at Bajo Rojo, I took 13 trials, and of the species present, 51% were adults. In Jicote, there were four trials, and of the species present, 65% were adults and 35% were juvenile or juvenile and adults. And in the mangroves, out of the 14 trials, 70% were juvenile or juvenile and adult, and 30% were just adult. So for the discussion, the juvenile fish that I found that were in both habitats, so the schoolmaster snapper and the big-eyed trevally, were in slightly different stages. They were still juveniles, but the schoolmaster snapper, when in the mangroves, I observed them in schools of about 5 to 10 fish, and then when I saw them in the ocean, they were alone. And I saw them quite a few times alone in the ocean. For the big-eyed trevallies, they were in schools in both locations, but the juvenile fish in the ocean were a lot larger and a lot more defined. I also noticed that the habitats like sand, rock, coral were very important, even within the larger habitats. So in the ocean, I found most nurseries to be in the rocky habitats. I didn't find many in the sandy or coral areas. Sand doesn't offer a lot of protection or food sources for the juvenile fish, so they kind of stayed away from that. And then coral, which you would expect to be a very good nursery habitat, sometimes emits different chemicals that are either harmful to the fish or just not very favorable. And when there were since there were rocky habitats present, they just tended to stay over there more. The ocean had more species overall, but of the species present, most of them were adults. And the mangroves, while it had a lot fewer species present, they were majority used by juveniles. So the mangroves are a very important habitat for juvenile fish. I want to thank Leo and Frank, my advisors. I want to thank Kwahinikeel Dive Center for helping us out on the boats. I want to thank Gloria, my homestay mom, for giving me a wonderful stay while I was there, and thank you to the Monteverde Institute for making this possible.
We do have time for questions. Anyone?
Are there any fish that are like juveniles in the mangroves that will eventually migrate to the ocean? If so, like how does that work? And is there like a certain age or time of year that it happens?
Yeah, so the schoolmaster snapper is one of the species that migrates. Some species have a specific age and some don't. Like for the schoolmaster snapper, many of them will migrate at about a year old. I'm not sure exactly what age the fish I saw were, but I'm guessing roughly around a year old. The big-eyed trevally were very similar. They also migrate to the ocean, and then many other snapper species that I didn't personally observe, but can be found in the mangroves, also migrate to the ocean later.
Other question? Tomas?
How were you able to identify like what fish that you saw, whether it was a juvenile or an adult?
Juvenile fish often have like very different coloration. So like the schoolmaster snapper, for example, it'll have that like very defined black stripe through its eye as a juvenile, and then as an adult, it's not present anymore. There's other fish like the Cortez rainbow wrasse that will have horizontal stripes when they're juveniles, and then they'll get brighter in color as they get older, or if they're male, they'll have a completely different pattern with like vertical stripes.
Did you memorize that going into this project? How did you know which like groups of fish were juveniles around the nest?
It was a lot of like identifying them as a species first and then identifying if it was juvenile or adult, or some of them are just, they look very different as juveniles. So when I was looking through different like books and websites and stuff, it was very clear that it was a juvenile and not an adult.
Sorry.
Ruby.
Ruby.
I know this is sort of a more general question, but do you know the like physiological mechanisms that the fish that spend parts of their lives in the mangrove and the ocean, like how they're able to endure the brackish water and higher salinity?
Yeah, I was also wondering a lot about that. I'm not super sure. There's not a lot of research done on juvenile fish, so I couldn't find any specific answers.
We don't have more time for questions. I'm sorry.
Sorry. Ah, no, no es with me. Ah, es que está... Sí, no ocupa el disco duro. Sí, a veces estoy cerrando el disco duro. Yeah, I know, it's not working.
It's loading.
Need. ¿Será este?
Simon?
It wasn't a ploy. I was just.
Thank you for waiting. Our next speaker will be Sophia Simon, and she is going to tell us about the aggressive behavior of the saber-tooth blenny.
Hello everyone, my name is Sophie and I'm going to be talking about the aggressive and social mimicry in the saber-toothed blenny, also known as Plagiotremus azaleus. And so here we have two different species. On the top we have the juvenile Cortez rainbow wrasse, also known as Talisona leucosanum, and it's a fish tropical in the eastern Pacific from Baja California to Peru. The juveniles form small schools that eat fish eggs, crustaceans, and plankton, and they're also known to feed on the ectoparasites from other species of fish. On the bottom, we have one that looks very similar, and this is a completely different species known as the saber-toothed blenny. And this species also has a very similar range to the Cortez rainbow wrasse, and it's also known to be solitary and territorial, living in old tube worms from mollusks holes. This blenny is also in the Nemophini tribe, which holds 6 different genera that are also known as fang blennies because they have large canine teeth. In the Plagiotremus genus, these species also are known to feed on the scales and the tissue of other species. And so with this, we have a very interesting relationship where these saber-toothed blennies will mimic the juvenile Cortez rainbow wrasses to feed on other species. So here, where do we think the blenny is? Can we find where it is? On the top? Ta-da! Wow! Okay, so for some more introduction and background, just to define what mimicry is it's when one species looks similar to another for a beneficial purpose. There's various types of mimicry, including Batesian, Müllerian, aggressive, and social mimicry. And today I want to highlight both aggressive and social mimicry. Aggressive mimicry is when a predatory species resembles a beneficial or harmless species to gain access to prey. And social mimicry is when a similarly colored species will benefit from associating in the schools of an unprotected species. And based on other studies and research that I have done, many members of the Blennidae family are commonly known to mimic species in the Labridae family for protection from predators as well as increases in access to food. So with this in mind, I wanted to ask, do the Cortez rainbow wrasse schools affect the aggressive behavior of the saber-toothed blenny? And are there preferred target species for the saber-toothed blenny? How do they react to this blenny? And so for my methods, I conducted samples at both Playa Jicote and Bajo Rojo. Playa Jicote is in a bay and has a larger diversity of coral species, and Bajo Rojo is more open and has a lot more, is kind of more faced with the surge and the currents of the ocean. And within my trials, I conducted 47 wrasse present trials and 44 wrasse absent trials for a total of 445 minutes of direct observation. And within one trial, the way that I approached this was finding one blenny that was either already in a school of the Cortez rainbow wrasses or if it was alone. And from this trial, I would estimate, one, the number of wrasses that were present, and then I would also count the number of attacks by the blenny on other species, as well as noting which species they attacked. And then I did the same thing with counting the number of received attacks towards the blenny by other species, as well as recording those species. And so here I want to play a video of what this behavior looks like. And so here we can see the blenny attacking mainly damselfish when it's alone. And it kind of is a little bit aggressive, and even within these videos that I've slowed down tremendously, it's hard to see this fish attack them. And so in these like coral reef environments, it also plays like a more complex relationship between different species and different mimicry relationships that occur. And so here it's like kind of even hard, you can't really tell where the blenny is. And so now moving on to my results, this graph shows the number of attacks per trial in both wrasse present and wrasse absent schools. So we have on the x-axis absence and presence, and then on the y-axis the number of blenny attacks per trial. And we can see that in the presence of the rainbow wrasse schools, there is a slightly higher attack per trial, which indicates that when the blennies are in schools, they may have a higher aggression rate. And conducting a Mann-Whitney test, this p-value is not significant, but since I didn't conduct that many trials and the data might be like not as many data points, this could show a current trend of how aggression may be related to the presence of these wrasses, with higher aggression in the presence of wrasses. This graph shows basically the same on the x-axis with absence and presence of the Cortez rainbow wrasses, but on the y-axis we now have the received attacks on the blenny in the wrasse presence and absence. And one thing that is interesting to note here is that in the absence of these rainbow wrasse schools, there is a higher number of received attacks with a median of 3 attacks per trial compared to when they're present in the rainbow wrasse schools, it was a median of 2 attacks per trial. So this also shows that when the blennies are in these rainbow wrasses, they benefit from the protection of being in this school based on like also having similar coloration. So then I wanted to also, since I took a lot of data on this species distribution, I wanted to also look at how the blenny attack rate kind of shifted across the species that they attacked. So these are just the common species that I noted, the most like common species. So we have Acapulco, Cortez rainbow wrasses, giant blue damselfish, Pacific sergeant major, and saber-toothed blennies. And on the y-axis, we have the attack rate per minute that the blenny is attacking on other species. One thing I want to highlight is looking at the saber-toothed blenny, we have a higher amount of attack rates when the blennies are completely absent, so when they're alone, versus when they're present. And this may indicate that like the behavior of these blennies are changing when they're completely like solitary versus when they're in these schools. And so I thought that was also interesting to note that they're attacking their own conspecific species. Okay, and then we have another graph here that's showing both presence and absence of the blenny, but now we're looking at the received attack rate towards the blenny across species. And so here I wanted to also highlight the dominance in the Acapulco damselfish. Both in the wrasse presence and absence, the Acapulco damselfish are the ones that are attacking the blenny the most. But also I want to highlight that more blennies attacked, like again, we see in the absence that more blennies that were swimming nearby would also recognize these of their own species and would attack them the most. And so this is also interesting because it kind of shows the benefit of being within a school. And so for my discussion, I want to just go over that blennies attack target fish more when they're in wrasse schools compared to when they are alone. And even though I did not receive statistically significant results, my p-value may indicate that this could be a pattern and something to investigate in future studies. I also want to talk about how the saber-toothed blenny benefits from associating in rainbow wrasse schools to decrease the number of attacks against them. And this may be because when they associate with the juvenile Cortez rainbow wrasses, the other species that may try to have retaliatory attacks on them cannot either identify them as a predatory species or they just cannot identify the specific individual. And so this kind of shows that they receive protection when they're in the schools. And then I also want to end with when the blennies are in these wrasse schools, they adopt free swimming patterns from wrasses, mainly because of an increase in food. In one of my graphs, I also showed that the blennies would also attack the Cortez rainbow wrasses in their presence. And I also want to indicate that when they're solitary, the blennies may exhibit more territorial behaviors and a higher aggression rate on their own species. So this may be an indication of facultative mimicry, where they only show this type of mimicry of the Cortez rainbow wrasses under certain conditions. And I want to thank Leo and Frank for helping me conduct this study, and Federico and Naomi and the Lara family for taking me out on the boat every day, and all of my fellow classmates. And yes, thank you.
We do have time for questions.
Scott?
Scott.
The attack of the blenny on other species? Since they are a carnivorous fish and their main diet relies on biting the mucus and the scales of other species. Yeah. Yeah.
Sorry, that question made me think, like, are they really hurting those fish when they eat their scales, or are they helping them get rid of dead scales?
That's a good question. I think it's mainly like they're hurting the fish. Yeah, because they're trying to bite part of them. So. Yeah. Yeah.
So, yeah, I was wondering when the blennies attack the wrasses, do the wrasses like respond by retaliating or trying to get away, or anything that might create a trade-off between using the between attacking the wrasses and using the wrasses as cover for other purposes?
So this was like an interesting result that I got, because when I was first like thinking of this study, I was like, oh, they mainly just use the wrasses as protection and they won't go after them. But I observed countless times that the blenny will attack the wrasse, but the wrasse will do absolutely nothing, and it'll just like kind of swim in the school again. And so maybe this type of mimicry also works with the Cortez rainbow wrasses, where they can't identify the blenny as being a different species because of this coloration and also the same swimming behaviors that they demonstrate.
When there is a blenny biting the fish, are they biting once, or can you see biting several times?
It's mainly like one time. Yeah, one time per fish, because usually they like dart and then the fish will swim away. Yeah.
Thank you. We don't have more time for questions.
Should be in green.
Second.
Perfect.
How does, is this the clicker?
Yeah.
Is that-
Forward,
okay.
backward.
Okay.
Oh, it's running out of battery.
Can you use, if it does, can I just use the
Yes,
laptop?
exactly. If it stops working, then just use your.
Okay.
This is what people are seeing, and this is the next slide.
Got it.
Well, and sadly, we are in the last of our wet and salty talks. And for this enclosement, Monica Banas is going to tell us about the effect of snorkeler presence in fish surveys.
Hi, my name is Monica Ballas, and I'm a biology student at UC Santa Barbara. And like Leo said, my study investigated how snorkeler presence affects the species richness and abundance of fish in northwest Costa Rica. So snorkeling is often used in marine fieldwork to observe and survey fish. As you've seen, if you've been watching the studies, lots of people have used snorkel to do their work. And there's an inherent bias of this, however, where the presence of a human can scare off fish and cause them to, have a different behavior or act differently. And this is a very understudied topic. I couldn't find many papers that looked at how snorkeler affects fish. And this has never been studied in Guayaquil, Costa Rica before, which is where I did this study. Guayaquil is right up here in northwest Costa Rica. And I used remote underwater video in the study, which is basically just underwater cameras, GoPros. And I looked at the richness, which means just the number of species, and the abundance, which is the number of individuals of a certain species. So my central question was, how does snorkeler presence affect the observed fish species richness and abundances at different sites near Guayaquil, Costa Rica? And my materials and methods were split up into the fieldwork and then the video and data analysis. So for my fieldwork, this was conducted from the 16th to the 26th of November 2025 at these 6 sites. A lot of these you've heard about before. However, Isla San Jose was one that was quite a bit further away, not that close to Guayaquil. It's part of the Islas Murciélagos, which is a protected site, so it's a little bit different. I conducted 22 paired trials. Each paired, and these were just across all the sites. Each paired trial consisted of a 10-minute snorkeler treatment and a 10-minute control treatment. In the snorkeler treatment, I was swimming around where the area that was being recorded by the GoPro, and every 2 minutes I would dive down in view of the camera. And in the control treatment, there was no human presence around the camera, and this always began exactly 5 minutes after I left the area to keep a consistent buffer time. And then after doing all this fieldwork, I had a lot of videos to analyze, which I did using this GoPro player application. In each video, I determined species richness by looking at every new species that would appear in the video. I would identify it and then record it. And then for each of these species that was in the video, I determined the abundance using a metric called max N. and this is just the maximum number of individuals of that species in any given frame of the video. So this picture right here is part of one frame of a video that I analyzed. So a little example, you can see these are king angelfish. There's 7, 23 barber fish here, 3 Panamic surgent majors, that's one there, a green jack up here, and this is actually an Acapulco damselfish, that little blob. So it was a lot of work to look at each individual fish. And I did that for almost 7 hours of the video, which I found out takes a lot longer than 7 hours. And I looked at over 2,000 fish that were recorded, and these represented 54 different species. So I found that there was, using a paired t-test, there's a significantly lower species richness when snorkelers are present. In all the averages of the control trials, there was 11.7 species, and the average amount of species for the snorkeler trials was 9.2. So this is a mean difference of 2.5. And this graph here shows there's control species richness on the x-axis and snorkeler species richness here. Each dot represents one of 22 paired trials. So like for this dot here, it had 15 species in the control, 10 in the snorkeler. And if there were any dots on this line, that would mean that that dot had the same amount of species for both treatments. And so dots on this side of the line had more species in the control. Dots here had more species in the snorkeler, which only occurred 3 times. So this decrease in species richness corroborates similar findings from studies done in Thailand and Mexico, which suggests that this effect is not just limited to certain locations, and that it might be a global thing that you can see in other locations. And one potential reason for this is just predator avoidance behavior. Fish might view humans, something much bigger than them, as potential predators and either hide or leave the area or just change the behavior to avoid the predator, and this would decrease species richness. However, more work is required to better understand this cause and all the nuances of this. And then these pictures here are from my Isla San Jose trials. Up here is the control treatment. You can see there's a ton of fish. I remember just sitting down to look at this video and I was like, oh my gosh, that's a lot. And then I was skipping ahead just to see what it looked like, and I got to this part and the fish are just gone. So then for general species abundance, each different species in each different trial had a max N value for the control treatment and a max N value for the snorkeler treatment, which was sometimes 0 if it wasn't present. And this graph kind of shows that. Each point represents an encounter of one species in one paired trial. So for example, this, I have no clue what these points actually are, but this might be Acapulco damselfish in trial 1. This might be Acapulco in trial 2. This might be yellow snappers in trial 2. So there's just a ton of different data points from different species in different trials. And so I conducted a Wilcoxon-signed rank test to determine whether this difference is usually something besides 0. And this was significant. So there was 164 encounters of species that had a higher abundance in the control. This graph is the same concept as the species richness one. On this side were encounters where it was more, there was a higher abundance in the snorkeler trial, and then ones that are on the line had the same amount. So there's 164 here, 99, 54 are the same. However, when doing a linear mixed model, which I did later on to look at a site effect, this also looks at just treatment as a variable itself. And this showed that this didn't show a significant result, and I interpreted that as snorkeler presence isn't really a consistent predictor of decreased abundance for all species. And so these 2 tests show that there's like a small effect of snorkeler on just all fish abundance of all species as a whole, but it's not super consistent and clear, not as much as species richness. Sometimes species are gonna decrease in abundance when snorkelers are there. Sometimes they'll increase. Sometimes they'll stay the same. Sometimes the same species has done all 3 of these things at different trials or different sites. And then another aspect of this is that not all species will react the same to snorkelers. So there might be some that actually do show significant response consistently, which is what I looked at next. I tested 14 fish families and 28 species that had enough trials that had at least 5 observations, so enough data to analyze. And I found 3 families and 4 species that showed a significant response, and all of these were a decrease with snorkeler presence. And then these are a list of the families and species, which I will talk about on the next slides. So the pufferfish family and the porcupine fish family were 2 families that showed a significant decrease. And then this species right here, the spotted sharp-nosed puffer, was one species that decreased on its own as a species, as its own category. And so puffers are captured for the aquarium trade in Quahina Keel frequently, which might well explain their aversion to snorkelers. However, porcupine fish are not as commonly targeted, so this behavior might just be something that's more of an instinct and not necessarily based on a factor of the location. And then the yellow snapper and the terminal phase of the Mexican hogfish were 2 more species that showed a significant decrease. The family of the yellow snapper also showed a significant response. This family is Letjanidae, and the reason it also did was because this was the only species observed in that family, so it's the same result. Both of these fish are commonly hunted with spear guns in Quahina Keel, so this might show a correlation between species that are heavily preyed on by humans and the species that might want to avoid humans. And this shows that snorkeling surveys might not be super reliable for studying fish like these that are commonly hunted by humans. And then finally, the Acapulco damselfish showed a significant decrease, which was very interesting to me because this was one of my most observed fish. I saw it at every site in almost every single one of my videos. So even though it was almost always present, even when I was there, like I saw them with my own eyes many times, they had a slightly reduced abundance. And one explanation I think might be a cause for this is that they just had reduced activity, so maybe they're hiding more, even if all the same ones are still present, and I just didn't see them all in the video when I was there. And then I looked at whether sites differed and found that they did not. There's, both in terms of species richness and abundance, fish weren't more sensitive at some sites than others, which you can see in this graph. They all show a downward similar slope. And then in conclusion, the biggest takeaway is that species richness decreases with snorkelers, as well as the abundances of certain fishes. So snorkeling might not be suitable for all research purposes. When you're looking at fish, like abundances of fish that are heavily hunted or just trying to find a number for general species richness, it might not be the most suitable. However, there are many circumstances for research where snorkeling can and should still be used. And yeah, I did this study because questioning methods is super important to improving the quality and accuracy of science. And in Quahina Keel, this study can be used to inform future projects that use snorkeling for research, in addition to adding to a large body, or not a small body of literature about this effect around the world. And thank you to everyone that's helped me, Frank Leo, the Monteverdi Institute, Quahina Keel Diving Center, my homestay mom, my peers, my family. Thank you.
We're still having time.
That's a good question. Overall, I saw 54 species. Yeah, I don't actually have a number for like how many was just in total. I think most of the time I saw both species at some point in both trials. Usually on the camera, yeah.
Another question? Owen.
Have there been similar studies in the past on other large animals such as crocodiles or sharks and their effect on species richness?
Yeah, I'm not sure. One of the papers I did look at when doing research before this was like, I think it was about mammals or something, or it just talked about in general, like animal behavior studies can be unreliable if a human is present. And one of these papers mentioned that this might be especially true in aquatic settings, possibly because humans aren't as present usually in the water.
Emmy is not allowing me to take more questions, but thank you so much.
Thank you.
We'll do a absolute no longer than 10-minute break, okay? So there's refrigerio out on one of the tables. There's also a gluten-free section of refrigerio, okay? And some other surprises.
Yeah, I used the flashcard like once.
Amazing.
Yay, thank you.
Flashcard and I didn't.
Yeah, there was one part that I knew. I was like, never mind, we'll stay here. But
There was a part that I was like getting messed up.
I was staring
Thank
at
you.
the
Thank you.
whole time.
Oh, thanks.
And listening, of course.
Oh shit, yeah.
What's up, live stream?
Yeah, I was like,
Single you will be talking about frog diversity in San Bernardino.
Thank you. Hi, Mom. Hi, Dad. My name is Trinity Terranova, and I did frog species diversity at the San Gerardo Biological Station. Okay. All right, so a crucial ecological indicator has been disappearing from its habitats all over the world. Habitat loss, climate change, deadly citrid fungus, as well as other concerningly unknown factors have basically driven frogs to endangerment, and in some cases, like the golden toad from Monteverde, actual extinction. So I conducted this study as part of an ongoing effort to monitor frog populations here in Costa Rica. I asked the question, what is a frog? No, I asked the question, what is the frog species diversity at the San Gerardo Biological Station? Okay, so here we have Emmanuel, Percival, Janet, and Minor. These frogs represent the 4 families I identified along with peers and mentors at the station. We have glass frogs, which are known for being arboreal, as well as their specific skeletal structure. It's very slim. Then we have leaf litter frogs, which later I will show you were the most abundant. Leaf litter frogs are known for, first off, they don't have any webbing on their hands or feet, but also they reproduce directly, meaning they skip the tadpole stage, and just as soon as they hatch, they're little teeny tiny frogs. And we saw a ton of those around the station. Next we have true frogs. There was only one of these. They are known for their smooth, moist skin texture. It's similar to a toad, but they don't have the warts of a true toad. And finally we have Minor. So the tree frogs are known for, of course, probably a lot of people know they have those really big discs on their hands. They do use those for climbing, but many people don't know that they use them to glide when they jump like that. It's really cute. All right, so for my materials and methods, my study site was the San Gerardo Station. I chose this station, it's about 12 kilometers away from Monteverde. I chose this station because I did preliminary observations. I tried to do my study in Monteverde. And when I was doing those observations for several hours for like 2 weeks straight, I found 0 frogs. So I chose to do this station instead. It's situated within a pre-montane rainforest. And it is known to have, from previous studies, it's known to have like a high abundance of frogs. For my procedure, myself as well as mentors and other students went out for 4 consecutive days while we were at the station to collect as many frogs as we could possibly find and photograph them as so. And for my materials, very simple. I used nitrile gloves, Ziploc bags. I kind of use them interchangeably, but always use the gloves because I needed to change them between frogs to prevent them from infecting each other with different diseases and prevent myself from getting salmonella. And then for data collection, I took all of the photographs at the end of each survey day, put them into a Google Sheet, and analyzed them to find out their family specifically, and then later using Eladio Cruz to figure out what their actual species was. This is an example of one of the sheets after a day of surveying. Usually I just tried to keep it at 2 photos. Not all of them were super easy to tell. Again, Eladio was super, super helpful here. Here I should note that there's a glass frog there. It doesn't look like a glass frog. And this is also a glass frog, but this is a juvenile glass frog. So for my results, 26 total frogs were found at the station over those 4 days. Here on the x-axis, you could see the 4 families that I found. On the y-axis, the number that was found over the course of the study. 2 tree frogs were found, 2 glass frogs were found, 19 leaf litter frogs were found, and only 1 true frog was found over the course of the study. I also analyzed the frog's location relative to the station. So near the station was considered around the grassy perimeter before entering the trails. Far from the station was on the trails Sendero Principal, Sendero Saino, and Sendero Catanata trails. So it was pretty interesting what I found here. So near the station is in black, far from the station is in green. No tree frogs were found near the station, and no true frogs were found near the station. But more glass frogs were found near the station than far from the station, and more leaf litter frogs were found away from the station than near, but I hypothesize that's because they are just the most abundant group that I was finding, myself and peers were finding on the forest floor. This is a map of the trails surrounding the station. So that black dot up there is the station. The trails, as you can see, are really vast. The trails that me and mentors and peers were surveying on were the Sendero Principal in green, the Sendero Saino in orange, and then in brown the Sendero Catanata. And I didn't really venture that far past, I want to say maybe right here. Just kind of like that general area because these trails are really kind of vast. So my favorite part, we found 8 species of frogs, including the rufous-eyed brook tree frog, emerald glass frog, both juveniles and 1 adult, the Branford's litter frog, Underwood's litter frog, slim-fingered rain frog, common rain frog, brilliant forest frog, and pygmy rain frog. And I should note that the rain frogs are also not leaf litter frogs. So the rain frogs are leaf litter frogs, but they are a subspecies. Specifically, this one right here, the pygmy rain frog, is part of a subfamily called Pristimantis ridens. But I got this confused, and I almost thought that they were a completely different family earlier. So, all right. So what do these findings mean? For one, based on my study, as well as 3 others, 1 in 2019, 1 in 2023, and then 1 in 2024, San Gerardo is an ecological hotspot for frogs. In every study I looked at previously, San Gerardo had the highest species diversity and the highest individual count of frogs. Additionally, in fall of 2025, it looks like Monteverde just showed a random decline in frog species diversity compared to San Gerardo, and that's just based on my surveys. And finally, continued surveys of Costa Rican frogs are absolutely critical in order to understand their threats and better protect them from extinction. For further research suggestions, for one, I would search a larger area and more sites than just San Gerardo, and I would look for more than 4 days. Additionally, I would collect more detailed data. I would collect data on the soil substrate, pH, temperature, and elevation, because elevation is known to have an impact on frog activity. Additionally, I would survey during the spring and summer because fall is apparently a really not good time these days to find frogs. So I would do the spring and summer, and other studies have found more frogs during spring and summer in previous years. And finally, I would survey amphibians as a whole, not just frogs, because amphibians are all threatened, basically. So thank you to Fede and Eladio, your advisors, teachers, but also dear and caring friends, to all of us students for many, many years now. Additionally, thank you, Naomi Solano, and all the students who helped me frog hunt, especially this knucklehead right here. And Great, thank you. That's not working anymore. Thank you. Oh, there we go.
So you mentioned how you think that there might be more abundance of frogs during spring and summer. How do you make sense of that when fall is normally rainy season?
So thank you for bringing that up. I believe climate change and other environmental factors and habitat loss have been affecting the breeding season of frogs as a whole. It's expected that they would be active during November when I was looking, but there was none. And a large part of that was because there was a storm, but like previous studies that have been done in the spring, all the way back to 2019, which is how far back I looked, have shown that there was just more frogs during those. I haven't looked at another study that was done in the fall. I've only looked in this like spring and summer.
Yes.
So you said there were two parts to your study up in San Gerardo, and they were the close to the station and then further. And was there any significant difference?
Oh, yes. There was, so a lot of the arboreal frogs, so actually I shouldn't say that. I found a lot of leaf litter frogs near the station, but I found more leaf litter frogs away from the station. They just happened to be the most abundant. There were far more frogs as a whole found farther away from the station. I think probably because they were in their natural environment in the forest, not like next to a giant building.
Do you have like a specific measurement to see like if you're close to the station you're in with this parameter, or if you're farther, you're outside of this parameter?
I wish I did. I wish I did. That may have like kind of pushed the study maybe a day longer than the time that we had in San Gerardo. We had like 5 days in San Gerardo, but that would be amazing. And for future, I'm glad you brought that up, because for any future research, I did see another study that was doing amphibians, and to do that, you have to take a transect and just plop it down in the middle of the forest, and you have to accurately measure what you're doing.
Do you think the difference in microhabitat from further away from the station and closer to the station influenced the type of frogs just because of like less trees or something like that?
Yes, absolutely. When I looked near the station, I found glass frogs, but they were up in the canopy when the grass met the forest. And then most of the frogs I found just near the station in general were leaf litter, because there's just a bunch of grass. And then there's like dead leaves there. And as you're walking, you just kind of kick the ground and you'll see a little like, and you'll be like, oh, there's a frog. And it'll usually be a leaf litter frog. So I think, yeah, there's definitely a difference. More arboreal, probably in the forest.
Jared, did you want to ask a question?
Oh yeah, I have a question. It's your birthday, right?
Damn it, Jared!
Happy birthday to you. Happy birthday to you. Happy birthday, dear Trinity. Happy birthday to you.
You made my day.
Happy birthday to Trinity's mother.
Yes, it's our birthday, not just my birthday.
Thank you.
Thank you. Kill it, homie. Huh? Ooh.
Do it, bro.
Okay. It's the clicker.
The microphone's here. Yeah.
Oh, okay.
Yeah.
Can you put it closer to yourself? I don't know how good it is.
Fair enough. I don't want to blow out this microphone. But
All right.
No, that's good. That's perfect.
Is this good or okay?
This is the clicker.
Yeah.
And this is what people are looking at.
Yep.
This is your following slide.
Yep.
Only you can see.
Yep. So I want to like see if I can go like forward and back.
Yes.
Okay. Yeah. And we have this
Our
right
next
here.
speaker is Owen Stewart. And he will be talking about pelicans in Guajira. Let me just say that I admire this gentleman's fervor and enthusiasm for his study subject and his knowledge acquired during his project.
Thank you, Frank. Okay. All right. All right. How's everyone doing today? I said, how's everyone doing today? Woo! I'm doing great. Thanks for asking. So as Frank said, my name's Owen Stewart. I'm going to present my presentation on pelicans in Guayaquil, and specifically how they interact with fishermen. So these guys right here. Oh, sorry, I didn't credit whoever drew this. Let's just pretend it's mine. Anyway, but now raise your hand if you live in close proximity to these birds, if you're used to seeing these birds close to your home. Okay, now keep your hand up if you also live within maybe 5, give or take, miles of a harbor or a dock, or you see, nobody dropped their hands, so this is what we're talking about today. So that said, let's get into this. So pelicans, if you don't know, if you're unfamiliar with them, they're a type of seabird found year-round in Costa Rica. They're considered a sentinel genus, or brown pelicans are considered a sentinel species of seabirds, which means that a lot of other seabirds sort of follow their behavioral patterns. You know, where pelicans go, other seabirds tend to go. And they're generalist feeders, which means they can feed on pretty much anything and that they can find or dive for in the water. Multiple different species that they can feed to their chicks as well. And brown pelicans are especially, they hug the coastline. So they're found between like British Columbia going south to Chile, and then along the east coast of the US from the Chesapeake Bay around the Gulf of Mexico to Venezuela. So they're a marine type of bird. And they've been known or have been previously studied and also viewed by myself and Frank to directly interact with humans. And so this is what has interested me. Fishermen have these fish cleaning events where they clean off their fish and they toss these extra scraps into the water, to which the pelicans surround the boat like little, you know, people waiting, like little trick-or-treaters waiting for candy. And then they receive the food in the water, and then they, you know, they stick around or they fly off once that's all done. So that's what I was interested in. So my central question involves this: how accustomed are these pelicans to receiving the food in the specific location that is the Muelle Guayaquil, or in this case, the harbor that we looked at in the little fishing village? And also the type of boat that the fishing crews feed them from, and if any adjustments would affect their behavior. And it turns out, well, I won't spoil that. We're going to see that in a little bit. So some of the materials used, we used 2 different types of boats. We used a fishing pongo, which is this open-top boat that pelicans are more used to receiving food from, and also a tourism boat or a diving boat driven by the Lara family. So shout out to them for that. We used a supply of bait, which I got from Frank, and he ordered it from this little like fish like bait shop thing that's by the harbor. We had to like unthaw the bait as part of it, so it was frozen solid initially. But and then I used a knife and gloves to cut up the bait and toss some into the water. We used a video cam to get the data at a wide angle, and you'll see that in a little bit. But yeah. There were 12 trials overall that took place. So there were 2 different types of locations that we tried out, and also 2 different types of boats, which leaves for 4 categories, and that's 3 per category. And the camera was, recording happened from before the first piece of bait was thrown until after the trial was done. And so, you know, this is how it worked. We have the boat here, and then we have the fishing boat captain, and there's me, and I got my supply of bait there in the boat. And then you have usually somebody somebody who recorded the camera. This could have been another student, it could have been Leo, could have been Frank Joyce, but anyway. And I'm keep distance away just so that we can hold the camera at a wide angle, and then we set sail in the water. So in order for these trials to take place, we had to observe some pelicans in the area, of course. You won't really get a response without a bird that's not really able to see you. So we looked for one that was flying in the air, and then at least 2 that were stationary, and then we were good to toss some bait into the water. So that's how it worked. In terms of how this study went, which variable do you think mattered? Do you think location? This is the Muelle here up top. That's Isla Muñecos, the unfamiliar location, and this one right here is the boat type. And you can see that's the unfamiliar one. And it turns out both of them were significant. And you'll see that in a little bit. So this is what some basic tables look like. You can see the boat type off left and the location off right. Normally, these would be lined up. I don't know what went on with the computer, but it's fine. We can pretend that they're there. So anyway, it kind of looks like a Punnett Square setup. But yeah. Oh, there we go. That fixed it. There we go. But yeah, so this is just a little basic start. You can see that the tourism boats at Muñecos had an average of 0, and this is no mistake. There were no pelicans that came to any tourism boat that came to Isla Muñecos. There were plenty of them there. There were about 40 of them looking down at us for about 20, maybe 30 meters up. And then they had responses that were few and far between, with unfamiliar boats in a familiar location, or unfamiliar locations but with familiar boats. And of course, you can see up to 25 pelicans showed up at any one time. These are the most that were there present at any one time. And it loaded up to 25 pelicans, which that happened on the last trial. I don't have a picture of what 25 of them looks like. This is 17 though, so you give a good idea. But anyway, this is a slightly different stat. This shows pelican presence in a different way. This doesn't take into account the sheer amount of pelicans that were there, but rather just the seconds that we had with at least one pelican stationary in the water close by. And you can see that it was close by over half of the time with just the fishing boat in the Muelle, which is what they're used to. And you can see some of the results. So these were the account of the total pelicans visited by the video footage. You can see overwhelming results for fishing Muelle. And you're probably wondering, oh, and why don't you adjust that for the amount of time that pelicans were there? Some of them were there longer than others. Fair enough, that's even more overwhelming. What if you adjust it for the average number at any one time, which is what I call companions. If you have one companion for the whole trial, or a number of 1, that's the equivalent to like 1 pelican sitting by the boat for the entire duration of the trial, or maybe 2 for only half of it, et cetera, et cetera. And you can see some overwhelming results again. And that's adjusted for bait tosses as well, because the bait tosses varied and it's still extremely significant. The only unsignificant, so using chi-square analysis, the only unsignificant data point here was between these two, because there were so few pelicans that showed up at Isla Muñecos that these numbers were similar enough to the point where they couldn't be separated from each other. Other than that, the results show a clear trend towards pelicans favoring receiving food at the Muelle specifically and from fishing boats, which shows how much they've been trained to do so. They come to the boat in larger groups for a longer amount of time in the control sample in the Muelle Guajiniquil. And that's what it looks like. They even fight between the food that they get. Meanwhile, the actual opportunistic feeder here was frigate birds. And they showed up at almost every trial, no matter what the fishing boat type was, no matter what the location was. Honestly, my prediction was that pelicans would behave in a more comparable way to this. But finally, a quick point here is that Isla Muñecos might be just as much of an outlier as the Muelle is, because Isla Muñecos is a nesting location. The pelicans were observed nesting, so they may have been hungry, but they may have been too preoccupied with taking care of their eggs, waiting for their mates to go get food. So they may have had other things on their mind. Other than that, I would like to thank all of you guys, of course, for supporting me, but especially my advisors and my translator and assistant with my project, Frank Joyce. I aspire to be like you and your boundless energy with helping us out and chauffeuring us and all of that. But other than that, thank you guys so very much. With that being said, that is my presentation. Thank you guys so much for listening. Any questions? Yeah, Kim, I see your hand question. Actually, yes. So, funny story, the first day that we went out was the 16th of November, and I didn't collect any data that day. I had bait ready, but if you go back 36 slides or whatever to like this setup here with the amount of pelicans, right? The first day when we were at the Muelle, we saw no pelicans even around. And even Frank was like, this is unusual to see this few here. Because we saw a lot more of them in October. We're about like double the amount. But yeah, there were absolutely times where I was like, this is not the right time of day for it. So yeah, good point there. Thank you. Gordon. Probably, oh, that's a good question. There were a lot of challenges that we faced collecting data. First, I would say would be finding the pelicans, as I mentioned earlier. But second of all, it was just to make sure that we, like, I guess the biggest overall obstacle that was a theme was just acquiring bait. And also, once we got the bait, it was often frozen solid. It's a shame that I didn't include the picture, but there's a picture of me holding up the bait that was all just frozen together, and we had to wait over the course of the day to thaw it out. So that was a fun little process. And so we learned over time to have bait that was just like ready the previous day. And so that's something you learn from doing stuff more often. I mean, if I did a follow-up project to this, then we would obviously have that down more. Trinity, you have a question?
What would you do with this project if you had a year?
Oh, probably. These are good. Now, if I had a whole year for this, first what I would do is I had ideas to like track pelican activity by time of day, but also if we were there for a whole year, we would see their whole nesting season and patterns with that. So if we measured like fish collection from all sorts of different months of the year, we could see some significant trends there, especially if the fishermen have a weekly schedule or a monthly schedule and things like that. Yeah, question here? Pelicans, the oldest one on record, I think, was 43. Yeah. Question over there?
Do you believe these were like the same individuals across both locations?
That's a really good follow-up experiment too. So yeah, there's two realities with this. There's one where the group at Muñecos was entirely different and completely unfamiliar with the feeding process, or one where they could be the same group. I'd have to say I couldn't know that if I tried, because if we bird banded some of the pelicans way up there, it would be way too precarious. Like, I've done some precarious things this program. Frank would have been like, absolutely not. Do not climb up Muñecos to try to tag those pelicans. You're gonna fall into the water. But yeah, a really good study would have involved tagging the pelicans and tracking where they go. That's a really good point. Yeah, and by the way, I should be clear, the oldest pelican on record was 43. Usually after like 3 to 5 years, they develop adult plumage. So like in their first couple of years, they're juveniles and they have different plumage. But yeah, is that all we have time for?
Like and subscribe, by the way.
Well, you woke up the audience.
Yes, perfect.
Perfect. Here.
Thank you. And then,
So this is just, yeah, that's forward, backwards, and this is the laser.
oh, okay. Thank you. What?
This is what everyone is looking at, and this is the following slide.
Okay.
Is Sophia Newton from UCLA, and she will be telling us about barefooted tiger hens.
Thank you, Frank, for that introduction. My name is Sophia Newton, and I'm going to tell you about my study, which was comparing the foraging behavior of adult and juvenile bare-throated tiger herons in the Quahina Keel mangrove. So a little bit of background. Bare-throated tiger herons are part of family Ardeaidae, which is the heron family. And since little is known about bare-throated tiger heron foraging, I'm going to tell you a little bit about Ardeaidae foraging. And what you need to know is that these are wading birds. So they're known for standing in the water when they're foraging and eating prey there. And when they forage, they slowly and methodically walk until they find prey. They have very good eyesight. And then they stand very still, staring at the prey until they make a jab at it. Mangroves are a very essential ecosystem for Ardeidae as a whole, herons as a whole, because there's so many fish there and so many crabs. There's just an abundance of prey items. And further studies before this have shown that they primarily eat small fish in the mangroves. And then what we do know about bare-throated tiger herons is little because they're very unstudied. There's not much research out there, but there is this paper by Kushlin and Anger in 2007, which studied their foraging in Panama on the rocky seashores. And juveniles and adults are both abundant in Rio Quajinaquil. I observed them before I did this project. And there are a lot of studies out there that compare juveniles and adults, other birds and other herons, just not these specifically. So that led me to my question, is there a difference between the foraging behavior of adult and juvenile bare-throated tiger herons in the Quajinaquil mangrove? Here is a juvenile and here is an adult. And as you can tell, I hope they're pretty different, so they're easy to distinguish, and that's why my project was based on comparing these, because I could tell the difference very easily. Okay, so what I did was I went to Rio Quajinaquil every day. I would look for bare-throated tiger herons. When I found them, I would determine if they were foraging or not. And if they were foraging, I would start my observation. Here's an example of what I would do. I would write down the time I started. I would observe them, and then I would write down the time I stopped observing them, which was when they flew away or were out of sight, something like that. And then I had a total observation time. And then during an observation period, what I recorded was the successful prey catches that they had on both land and water. And then I also did failed attempts. So a failed attempt was when they would jab at a prey item and then not catch it. I also estimated the percentage of time they spent waiting during an observation period. And I did other things I noticed, such as common behaviors, foraging disturbances, etc. So here's what I found. I observed a total of 11 adults across the entire study and 13 juveniles. And what's interesting about this is that I found a very similar amount of them foraging, except that the adults had about half of the time spent foraging than the juveniles did. And as you can see, the juveniles caught about 23 total prey items versus the adults who caught 9, but the adults spent less time. And overall, they ate a lot more things from the land than from the water, which if you remember earlier, I said they were wading birds, so this is a little bit interesting. I'll get into it later. And then I did calculate the prey captured per hour per individual bird that I observed, and I put adults and juveniles on the x-axis and then their prey capture per hour average. The adults were a lot higher. There was about 6 prey items per hour for adults, and then the juveniles were about 3, but there is an outlier up there on the adults. I think that adult specifically caught about 4 prey items in 5 minutes. So in this graph, I have the minutes spent foraging on the x-axis and then the failed attempts on the y-axis. And then the juveniles are the white dots and the adults are the black dots. And all of this is just to say that here you can see the adults only had 2 failed attempts the entire time, whereas the juveniles had around 33, I think, and we had an outlier up there with 17 in one observation. And then here I did a very similar graph, except instead the y-axis is percentage of time spent waiting, and we have 0%, 50%, and 100% of the observation period that I observed. And I only saw 2 adults waiting the entire time versus I think that's 6 juveniles waiting. But there were a lot more birds that just didn't wade through the entire observation period. And so what do these results show? The juveniles are less efficient foragers than the adults. That's what I found. They had a lot more failed attempts. They seemed to be clumsier in that way. They also had less prey per hour, so they were less successful. And I saw them wade more than the adults. And this is interesting because wading and capturing prey items in the water seemed to be a less efficient form of foraging. So it was interesting that the juveniles did this more, and I thought you could attribute this to them having less life experience. And I also thought that because I thought that wading was less efficient because it's harder to see in the water distorts where the prey items might be, whereas on land you can see the prey better, and so they were more successful. And yeah, they just spent more energy. In this video, there's a juvenile making an unsuccessful attempt, I think, at some piece of plastic. I'm not sure. And then this is a little bit confusing, but I'm going to explain it. So I observed a lot of territorial interactions between bare-throated tiger herons. And what I observed was that the adults and the juveniles had a total of 7 territorial interactions between each other, which meant that usually the adult would chase a juvenile out of its territory. I only observed 1 juvenile versus juvenile territorial interaction, and I saw none between adults. I'm actually not sure why the adults and the juveniles were more competitive in this way, but it might be due to adult mating or something else. And then I had some unexpected observations as well across both juveniles and adults, so now we're just talking about bare-throated tiger herons. And what I saw was that they ate primarily crabs, which was against the literature I found before that said they ate mostly fish. And then they mostly foraged on the land, hence eating mostly crabs. Like I also talked about before, they're considered wading birds, yet they were found foraging on the land most of the time. And then what's really interesting here is across both juveniles and adults, I found an average of 4.5 prey items per hour total. So in the study that I mentioned before by Kushlan and Anger, when they studied bare-throated tiger herons in Panama, they found that they averaged about 1 prey item per hour. So I found a much higher result. And I have some theories on why this might be. I think that because I was observing them in the mangroves, and the mangroves are such an abundant place for prey, that this might be due to, that this might cause the difference that I found here. Yeah, and in conclusion, there's a couple of takeaways from my study. And one is that for conservation, we need to meet the needs of the whole species, which means it's important to study the adults and the juveniles and to understand their differences, because mangroves are disappearing, and even though these birds aren't in threat right now, they may be in the future, so we should understand everything we can about them, and there is not much knowledge out there, so I'm trying to help close this gap. And also mangroves, they're an important environment for herons. This is already known, but I hope that my research just further adds to this point. And yeah, I would like to acknowledge the Kwahinikio locals, my advisors, and all of the staff, and some special mentions. Thank you all for, yeah, everything.
So you mentioned the paper that was studying these in Panama,
Mhm.
and a little bit about you studied obviously mangroves. Do you know where they studied, like what habitat they're studying tiger herons in? And then also, do you think that might affect, because you said that they're like primarily thought to be wading birds, if you observe that, do you think that would have been an effect of the study?
The habitat, you mean? Yeah, it's possible. In the other study, they studied them it was like along the coast on the rocky seashores. That's how they described it. I think they were observed wading more there. Yeah, it's possible that because of the tides of the mangroves, maybe they wade less. Not entirely sure the exact reason, but yeah, it's very possible. Kira. Yeah.
This species is highly territorial with like well-defined territories, and that's why they're not interacting, or are they more laid back and not super territorial?
They are known to be a territorial species. I did observe bare-throated tiger heron adults calling at each other, which might have been a mating thing. I'm not entirely sure. You can't tell the difference between females and males, so it's really hard for me to distinguish, but yeah, they are known to be territorial. Yes, Kaylin.
Were you able to differentiate between the individuals you were studying and somehow?
Yeah, thank you for asking. I actually did want to talk about this, but I didn't get a chance to. I recognized 3 different individuals across my observations in 3 observation periods each, and I actually named them. So this, let's see if this works. This is Kestrel, the bare-throated tiger heron. This is, oh, here's Flor Jr., which is named after a Coahuila local who helped me find the birds. And then here's Atlas. Thank you. Oh, I was able to differentiate them by using Frank's camera, and then I could take photos of the coloration on their face and then compare it when I got home, and then I could look and see if they were in the same places. Yeah. Their nesting season is in May, I believe. Yeah, around that time. Yeah, Alice.
So they're like foraging for crabs. What was the difference in technique that they used for swimming?
Yeah, I did observe this really weird body movement that they would do a lot, which was like a sway almost. So they would do the thing where they kind of crouch along the land, they would spot the crab, and then they would do this like swaying motion. And it would take them a while before they made the jab, but they were really successful at that. Versus the water, I saw them stab at the water a lot and not make any catches.
Thank you, Sophia.
Yeah. Thank you.
Thanks, bro. Where should I put this?
Anywhere you want. Here.
I'm gonna stick a clock here.
Then. All right. Forward, backward. This is the thingy.
Okay. Thank you very much, Frank. Hi, everybody. Thanks for being here. As Frank introduced, I'm Finnegan Men. I go to University of California, Santa Barbara, and I major in ecology. I came to Costa Rica and did this research about biodiversity, looking at plants and birds in pre-montane rainforest here in Monteverde. So I want to start off by saying biodiversity is essential. For those who aren't familiar with what it is, biodiversity pertains to a myriad of life the biosphere supports, both within genetic variation within a species to species variation, and that scales across ecosystems and entire regions. Biodiversity matters for a lot of reasons. It's becoming something that's pretty heavily studied as of late, and that's because it has a very functional role in climate resilience, which is becoming increasingly important. Additionally, it's very important for ecosystem services, which is another growing area of research. For those who aren't familiar with what an ecosystem service is, it is a service the ecosystem provides at no cost to us. An example of this is frog tadpoles can eat malaria-carrying larvae of mosquitoes, as well as the fact that our medicine cabinets contain chemicals that 50% of which were sourced from rainforests. They also are just more productive and functional when it's a biodiverse ecosystem. Tropical rainforests have the highest biodiversity out of any terrestrial ecosystem. And to highlight that, here in Costa Rica, it's only 0.03% of the Earth's landmass and land surface. However, it's 5 to 6% of its biodiversity. New species are discovered pretty much every single day in tropical rainforests. This led me to my central question. I was looking to ask how diversity differs between primary, secondary, or regenerating and degraded and disturbed rainforests here in Monteverde. So to do this, I went to the Curicancha Preserve. There they have all 3 forest types I was looking at. Their secondary forest was 30 years old, which was perfect for my study. And a key factor here is I was using birds as a proxy for overall biodiversity. They're a really good candidate taxa for that because birds are widely distributed, have a very important trophic level, are very diverse as a taxa, and are very readily and easy to observe. Additionally, I was doing underground quadrat data looking at the understory vegetation diversity as well. So I had two transects, each 100 meters across these 3 forest types. I walked each one frequently. I did 3 walks of each one. And then when I'd walk them, I'd stop every 20 meters for 10 minutes to collect avian observations. I used the Merlin Bird ID app developed by Cornell Ornithology Lab, as well as my own auditory acuteness and a Costa Rica field guide as well. That was handy. Additionally, to my right, I'd place the quadrat right off the trail in the forest that was there. I sampled on consistent times of day as well as weather patterns because I didn't want any confounding in bird activity. So this right here was one of my degraded transects in the Curry-Concha Preserve. This is an example of the old growth sections. And here's a keel-billed toucan, which I was seeing in secondary and primary forests. Here are two of the quadrats. This is a degraded one, and that is in the primary. Pretty stark difference. Let's get to the results. So I did a one-way ANOVA stat test to find statistical significance in my species richness. Here we have the forest type, primary, secondary, and degraded. I had 47 species unique to primary, 33 in my secondary, and 19 only found in my degraded. My total bird observations were 421. It followed a similar pattern with most being in primary at 180, and then going down in secondary and degraded as well. I had a high number of unknowns. Those were mainly centered in primary and secondary with very minimal and degraded at 31. An unknown is a bird that I wasn't able to identify or Merlin couldn't identify, but I could hear. It was really centered in the heavily forested areas because they were more cacophonous and sonically congested. This is my beta diversity. It's looking at the distribution of species across forest types. 41.5% of species were only found in primary, 15.4 were found in secondary and degraded, and 10.8 were generalist species found in all three. An important thing to note is that 64.6% of all species I ever encountered in this research were supported in primary rainforests. Here's another graph. This is looking at a few different abundance factors. I'm looking at the cumulative homogeneity of a sample. I measured that by looking at the three most abundant species. That was very high, proportionally degraded, with 59 being just those three species of my total 98 bird observations found in degraded. Contrast that to primary, which was same number, but a lot more total observations. I also looked at lesser violet ears and total hummingbird observations. Lesser violet ears were my most encountered bird. They're very vocal by nature and a generalist species, so I saw them a lot. Here's my last graph. This is looking at my ground coverage data by my forest type, and I grouped by morphology, bare ground, and leaf litter. Degraded was dominated by grasses and clovers, had very minimal leaf litter, and almost no trees of any kind, whereas primary was over 50% trees as well as 25% leaf litter. Secondary had very high bare ground and young trees. So now to get to my discussion. So these results demonstrate the replaceable nature and importance of primary rainforests in conserving biodiversity. They support so much because they're very vegetatively diverse and have a wide array of ecological niches that can be fulfilled by all sorts of habitat specialist species. They have developed leaf litter for insectivores and things like that. An example of this is I saw wood creepers and the barred forest falcon. Wood creepers need standing deadwood for their nesting site and foraging patterns, and I only saw those in those primary rainforests. The barred forest falcon is a species that has a preference for primary forest, but due to deforestation in South America has started taking advantage of secondary rainforest as well. Here's an example of standing deadwood that a wood creeper would use, and just the general complexity that you see in a primary rainforest is truly amazing. Here's that the barred forest falcon, and here is the northern barred wood creeper. So degraded rainforests were really homogeneous, as I mentioned earlier. They were a highly disturbed landscape exhibiting a high amount of successional species and grasses and very little complexity and room for ecological niches to be taken advantage of. An example of that is I saw the southern house wren, which was a species that could take advantage of the open area but wasn't found anywhere else. And pretty much all complex taxa disappeared from these ecosystems entirely. Here's an example of my degraded transect and just a monotypic landscape. Additionally, secondary rainforests were very important. They're sort of this functional middle ground where they did support more biodiversity than the degraded rainforest, However, has substantially less and not as many habitat specialists as the primary did. But they have this very important future ahead of us. Half of all rainforests in Costa Rica are now considered secondary rainforests, and they have their own ecological value. For example, the clay-colored thrush was only seen in this and takes advantage of more open woodlands. But restoration doesn't all come equally. These second forests are often culled for wood faster than biodiversity can really recover to a substantial amount. So the management goal in mind is very important. Here's an old growth tree you wouldn't find in secondary rainforest, and here's secondary forest, a lot of young trees. My understory ground coverage data pairs really well with my avian diversity data, and it highlights the structural drivers behind biodiversity differences across my 3 forest types. There are really simplified landscapes that generalist and human-adapted species could thrive in. Essentially, going forward, we need to focus on the dual imperative of restoring degraded or monotypic landscapes. This is deforestation, this is cattle ranching. These are really catastrophic for biodiversity worldwide and ecological function in general. And we need to pair restoring these ecosystems with conserving intact primary rainforest that's left, because those are just unparalleled in the amount of biodiversity and functionality that they have. This is my acknowledgement slide. I just wanted to thank my amazing advisors and all the staff that have brought me here and taught me, as well as my family and my loved ones, and Curry Concha Preserve for letting me research there.
Did you find any toucans?
Yeah, I saw a keel-billed toucan in secondary and primary. Favorite bird I saw and why? I saw a scarlet-throated hummingbird. That was really beautiful. And yeah, the barred forest falcon was gorgeous, for sure. Kira.
Were there any species you saw that had overlap among landscapes?
Yeah, so this slide shows the overlap. So these are grouped by species that I found in either only primary, only secondary, only degraded. All 3 are combinations of them. So yeah, the ones with the most overlap was primary and secondary, which would make sense. There are 7 bird species which were found in those 2. Laney.
How do you feel about using resources to plant trees in degraded forests rather than letting nature kind of recover on its own, give those resources for something else? Or is there a balance?
Yeah, sort of like all things in life, it's a bit of a balance. It's tough because sometimes seeding, when done right, can be beneficial and speed up the restoration process, as well as the biodiversity end result, because it was intentionally managed with that in mind. But as we saw in the ACG, it's not always necessary, and it's a lot more cost effective to do what the forest does best. So it's not necessary always.
Did you take in consideration the daytime for your bird?
I did. I sampled at consistent times and weather patterns throughout. Thanks.
Thank you, Finn.
Thanks, y'all.
Oh, fuck. Stickleback. But we need to be back in 10 minutes, okay? Because this symposium does not include dinner. So be back in 10 minutes, okay? Thank you.
Okay, I'm glad. I was hoping to win some favors, do birds, Cornell Ornithology Lab.
Yeah, I heard you aware. I got, oh man, yeah, I got it.
I'm presenting next. I was wondering if it's possible to put my laptop on here.
Is it, so you're the first one?
Yes.
Is it session 4 or 5? Not
5.
Finally. So it's not here, it's not working here.
It is. I've just made a lot of adjustments that I prefer.
Can you just like pass it? It's just easier to use with the same
Pass,
computer.
put it onto
we're going to start. This is the fifth session. Actually, it was a long day waiting for this, the most interesting session. First speaker from UC Berkeley, Scarlett Miller, going to tell us about behaviors of monkeys and diet. Welcome, and thank you.
Hi, thank you. I'm Scarlett, and this is howler monkey diet in fragmented coastal forests. All right. So a little introduction to howler monkeys. They're in the genus Oluwata, and they're neotropical primates that range from the southernmost of Mexico to the northernmost of Argentina. They're characterized through their loud vocalizations, also their howls, and most importantly, they're folivorous, which means that they primarily eat leaves, and they also eat nuts and fruits and seeds and flowers as part of that. So the species that I studied was the mantled howler monkey, also known as the Oluwata palliata. This species ranges from Colombia, Costa Rica, Ecuador, Guatemala, Honduras, Mexico, Nicaragua, Panama, and Peru. This species is vulnerable on the conservation status, and this is probably due to like climate change, fragmentation, habitat loss, and the pet trade industry. And this species is characterized physically through their golden long hairs on the side of their bodies. That's why they're called mantled howler monkeys. My question is, what is the diet of the mantled howler monkeys living in coastal fragmented forests? So the reason why I want to study diet is because it affects a lot of how they go about their life and why they are the way that they are. So howler monkeys mostly eat leaves, like I said, and leaves are very low in energy. They have low energy content, and this makes them sleep all day. They rest all day, because they spend a lot of time resting, they need to conserve their energy. They have very strategic movements. They are also, the diet also affects their anatomy. So they have a specialized gut microbiome that helps them digest the cellulose in the leaves. And they also have a slow metabolism so they can conserve their energy. They also have really good color vision. They can see just as many colors as you and I, and this helps them be able to determine what fruits are ripe and what leaves are young for when they eat. They also have a hyoid bone in their throat, and that's what makes them be able to howl. So their diet also influences why they howl. So they howl to let other troops know where they are, so that the way different types of troops don't have to come into contact, and they don't have to compete for resources, because howler monkeys really don't want to fight. They don't have the energy to fight. Methods. So first I searched for the monkeys, and I searched with a local from Guajiniquil named Jose, and I searched with my binoculars, and what we did was we listened and heard their howls and then went to them. And then we would observe them, and so I did 3 days of preliminary observations, and I was usually there from 9 a.m. to 5 p.m., but sometimes I was there at 5 a.m., or 5:30 a.m., some days. We observed 3 troops, and we definitely heard way more troops around the area. Then I documented them. I documented the trees that they fed from and the trees that they rested from. And then I would flag the trees and use an offline map to get the coordinates of the tree. Then with Roberto Espinoza, we identified each tree to help to see what they were eating. This is the study site. So there was regenerating tropical dry forest, and then there was primary growth thinned forest, and then there was a mangrove forest. To get a better picture, thinned primary forest is in green, mangrove forest is in the blue, and the red forest, the second growth forest is in the red, and that's where there's a big hill. Also, right here is pastures, and then this was also thinned primary forest. And you can see where the troops kind of are based on like the little pins. So the results. I collected 78 individual trees that were documented. Of the 78 trees, there was 13 species with feeding events. And then feeding events, there was 162 feeding events. And so I label feeding events as anytime I saw a monkey eating from a tree. So what they ate most, as you can clearly see, is Ficus inspedia, but they also ate Spondius mamen and Inga vera. Here's the same data, but interpreted in a different way. It's through a pie chart, and you can see it's a lot of Ficus inspedia. So how is the diet related to tree availability and abundance? With general area scans conducted by Roberto, we identified the most common trees in different areas within the home range of the troops and compared it to the number of the most common trees that these howler monkeys ate from. Here's a list of the common species in the area of the home range. I won't say every single one, but as you can see, there was a lot of Seminea salmon. It was present and common in every area that we were looking at, but they only ate a small amount of the trees. And then with Ficus inspedia, you can see there was a decent amount in the area, and most of the trees that were in the area they were eating from. I also want to point out Spondia mamen, which I said earlier had 22 feeding events. But it was like not present at all. I only saw one Spondia mamen tree, showing that it was eaten disproportionately from the relative abundance. So now we ask, why do these howlers choose these species? Ficus inspedia is part of the Moraceae family. It grows from 12 to 40 meters tall, and it has a wide crown, making it like ideal for a monkey to travel through. It experiences year-round fruiting, large per tree fruiting crops, and it has high values of protein, individual amino acids, and several minerals, including potassium and calcium. Spondius mamen is the second most eaten, and it's part of the Anacardiaceae family. It has 20 to 25 meters in length, and it's large and dense. The fruits are a great source of energy. It's the only time I saw a howler monkey eating the fruit. And it's an important amount of potassium. The pulp of the fruit has an important amount of potassium, phosphorus, magnesium, and copper, as well as it contains high levels of antioxidant compounds. Okay, Inga vera is part of the Fabaceae family. It was the third most eaten, and it has 12 to 20 meters in length. It contains secondary compounds, which is tannins and saponin, and it's rich in calcium, potassium, magnesium, and iron. Also, it has nitrogen-rich tissues. So, takeaway points: a diverse diet was not prevalent in the 2-week study that I was watching these howler troops. And in other studies, the howlers in fragmented forests have spent 23% of their time feeding on Ficus inspedia, whereas mine was, I think, 63%. But over those studies, it was over a year long, so it's possible that they could have just been eating the ficus only during this part of the year, which is like a transition between wet season and dry season. Most feeding events were Ficus inspedia, and Spondia mamen was ate disproportionately from the relative abundance. And then the opposite was that Seminea salmon had few feeding events relative to its abundance. Okay, acknowledgements. I want to acknowledge Frank and Federico, my first and second advisors, Jose, who helped me track the monkeys, Roberto, who helped me identify the trees, Sophia, who was with me for some of it, all the EAP staff in the Monteverde Institute. Thank you all for listening.
trees were fruiting. Was the long-beaning tree fruiting?
That was the only one I saw fruiting. But yeah, but it said that it fruits all year round, but I didn't see fruits from the ficus. Ew?
In your map, in your very beginning, you show a little gap in the distribution. You have any insight about this gap?
Oh.
No, in the whole distribution in Central America.
Which map are you talking about?
Can you go back and back and back? Back and back.
Oh, this.
Yeah, there is a big gap.
Oh.
The question is if you have an insight about, and if there is information about maybe molecular divergence.
Yeah, I don't know, but I know that there's 5 subspecies within this species, and so I assume that the species that's in the lower part of Mexico is probably a different subspecies than the species that are all around. Owen?
You mentioned that a lot of finding these monkeys was based on listening to their vocalizations. What was that process like? How long did that take at times? And did you know a specific place that you could consistently go to initially do them?
Yes, the place that I consistently went to was, oh, actually, I think I passed it. So the place that I went to was Cuatro por Cuatro, and it was in Coajinequil, and it was a certain road that led to the beach, and I knew that they were there every time. We went to one other place that we couldn't find monkeys at, and finding monkeys never took more than 2 hours. Yes.
You were recording what you saw them eat. Could that be like observation bias? Would it be better to do something in the future with like fecal analysis or something like that?
Yeah, I think there is an observation bias because I definitely noticed every single monkey made eye contact with me and was like, they knew I was there, they knew I was prevalent, and I think that they could have maybe chose to go in the higher trees rather than the lower trees because I'm obviously on the ground and they're probably not used to seeing a whole lot of humans just stay with them for hours at a time. So they probably was keeping their distance. That may have limited their like tree selection.
Gracias, Scarlett.
What?
I have a whole folder, sorry.
Whole folder. No se ha quitado la pantalla, pero está bien, ¿verdad? No entiendo por qué. Dale chance.
Oh, this one.
Pero está rarísimo que no.
And it's that one, the middle one. No, down. Uh-huh, yeah.
Cierre eso.
Oh.
Puede hacer pequeño.
Well, it's already there, look.
No quiero verle así. Lo que pasa es que. Ah, okay.
No.
No.
Forward, backward, and laser pointers red.
Okay.
Avery Larson from UC Santa Cruz is going to tell us about fishing bats.
Okay. Thanks, everyone. So I'm Avery Larson. Thanks, Fede, for introducing me, and I'm going to talk about the hind limb variations of Noctilio leperinus, more commonly known as greater fishing bats, sometimes also referred to as bulldog bats because they kind of look like bulldogs. If you see their smushed noses. Okay, so why greater fishing bats? One of the first reasons that they excited me was because they are extreme examples of morphometrics when specifically looking at their feet. They're huge in comparison to their wingspan and body mass. They are the second most diverse order of mammal, second most to Rodentia. They have around 1,500 species worldwide, and there's more to be discovered because they are fairly unstudied. As I point out here, especially the differences between male and female Noctilio leperinus, and that is what I'm focusing on in this study. They're key ecosystem indicators. They live in the mangroves, and that's where I study them in Kahini Keel. They prey on small fish that Tori talked about earlier. Sorry, Tori. And also, they're super cute, right? Okay, so here's a video of one capturing prey in the mangroves, and Jim Wolf was the one who took this video, and it's pretty extreme. They kind of like use their toes like this, and they rake their feet through the water. And so when I first learned about them, I was looking at different studies, and one main study that I looked at that kind of got me to my question focused on the hydrodynamics of their feet. So they were looking at multiple different bats and studying like what drag they have in the water. If they have higher drag or a greater surface area of their feet, does that affect hunting ability? And surprise, these guys have the highest drag, but because they have this highest surface area of feet, but this also helps them catch prey, and it makes them one of the best hunters in the study that they measured. And so that's why I'm asking, is there a difference in size between the hind limb, between the male and the female of Noctilia leporinus? I'm asking if there's a difference, maybe one, the female's toes are thinner to reduce drag, or if the males have thicker claws, something like that. And so that's what we studied. We were able to capture 31 Noctilia leporinus, which is an extreme amount. Actually, most studies I reference are captured around like 20 to 30 bats on average. And I just want to mention that these were very low moonlit nights. There was just a new moon, so it was pretty dark on average. But every night we would set up mist nets from 6 to 8 p.m. across the river or next to the river along the Rio Kahini Keel and the Rio Murcielago. And then this is me untangling, not a fishing mat, but from the mist net, and we would put them in bags for weight measurements. This is me measuring one of their middle digits. I measured the middle digits on every specimen caught to get uniform data. This is me measuring the length of one of the middle digits of the Noctilia leporinus. I would measure toe length, width, claw length, width, and height. I would measure wingspan, tibia length, ankle width, mass, sex, and I would see if they're reproductive or not. And so this is my first graph, and I know it's a lot. It's a little scary right now, but essentially what I measured here was the middle digit width, so how thick their knuckles were, or their toes. How thick were they in comparison to forearm? And I used forearm as a proxy for total size because weight varied so much. We caught pregnant bats, we caught bats that had just given birth, and also our nets were set up during feeding time, so a lot of them had big bellies that were full of food. So form is a good way to identify these bats, and so I use that as a proxy for size. I compared female and male. The females is the yellow line you see here, and the blue line is males, and this is what I measured. So essentially their claw comes out of here, and this is the thickest part of their middle finger. So as you can see, middle digit with the females actually decreases as forearm increases. So as the bat is getting bigger, the female's toes are getting slimmer. They have a slope of negative 0.55. And then males have a positive slope of almost 1. So it's a little more intuitive. As they get bigger, so do their knuckles, or the thickness of their fingers. And so why is this happening? Why are females having slimmer claws as they are increasing in size or getting older? And this led me to some of my conclusions. Calcium in female bats is often lacking. There's also usually some calcium deficiencies found in frugivorous or insectivorous bats, so that could be an explanation for what's happening here. However, fishing bats also, they do get a lot of calcium and vitamin D from the fish that they are eating, as they're mostly eating fish. However, when you're growing a baby or you're producing milk for that pup, it takes a lot of calcium. And if you don't have enough, then when the female bats are pregnant, and if they don't have enough calcium to support them and their pup, then the pup will start taking calcium from themselves. So they'll start thinning in bone development. They'll actually lose bone mass. And then another explanation I have for this graph is maybe that this is a selective pressure. Maybe females are, over time, choosing to have slimmer claws to reduce drag in waterways. Because when they're hunting, they're carrying their pups, and that's like 1/3 of their body weight they're carrying on them. So maybe they're selecting to have slimmer toes. Now, this is my next graph. I mapped middle digit claw width, so not their toe, but like their fingernail, essentially. Now, I measured the width of each claw, and I measured it against forearm length. And here it's a little interesting. So while males overall have a higher or thicker claws, they don't really move. It's pretty consistent. It's almost a slope of 0. So as male bats get bigger, their claw thickness is staying the same. However, while females are getting bigger, their claws are increasing proportionally in size. They have a slope of around 2. And this is really interesting because we can kind of correlate this back to the vitamin D that they get from fish. So they might have an increase in vitamin D from that fish, which helps with keratin growth, which is your hair and your nails. So they might be eating more fish, or it's just aiding in claw thickness. It also could be a selective pressure, which is a really interesting thing. So they're essentially, again, choosing to have thicker claws over time to help with roost stability. So when they're hanging upside down, remember they hang from their feet, right? And their claws, carrying their pups, it's often advantageous to have sturdier claws that are more supportive of a heavier weight. So that's another interesting thing that we can think about when looking at this graph. And then this is my last one. These are all statistically significant data points that I measured and found. So this is ankle width versus forearm. And so this is their little ankle. These are their feet. And this is kind of inverse of our last graph we looked at. The females now have a slope of almost 0. So as they are getting bigger, their ankles are not getting any bigger. But what's really interesting is the males have a really high slope compared to the females. It's almost 2.5. So as the males are getting bigger, their ankles are proportionally getting bigger. Well, not proportionally, but their ankles are also getting bigger. And this could be related to males eating more, if we didn't guess that based off of our human counterparts who like to eat a lot. Anyway, sorry, bad joke. They actually have been studied male fishing bats to have bigger cheek pouches so they can catch bigger fish and fit more fish in their mouth. Additionally, they travel farther to get prey, whereas females hunt together in groups and they stay closer to their roosts. So they might be getting different and more nutritious foods. And this helps aid in higher bone development. So once they've reached their maximum size or they continue growing, they often thicken out instead of getting longer. So tibia, I'll mention this in a second, but tibia is also another significantly different fact that we measured, where males had, it's kind of similar, males had a steeper slope than females. So they had longer tibias in correlation to that. Oh, and this also could be a roosting preference or mate preference. Maybe females are selecting males who have greater stability when roosting. Those thicker ankles will help with stability because often they hang on to pretty slippery sea caves or tree hollows. And often there's only 1 or 2 males in a group of females. Okay, so yes, tibia lengths showed significant differences as well between males and females. And one thing I just want to point out and kind of segue into our next thing is one of my field advisors, Eduardo Artavia, he talked about the misrepresentation of bats in the media. And essentially, I guess a lot of people think bats are, they carry diseases and they're scary and things like that. But what he said really kind of got to me was that if we study any animal looking specifically for pathogens, we're going to find more pathogens in other animals. These bats are specifically studied because they can carry pathogens without necessarily getting the effects of some of the viruses that they're carrying. So I think that this study allowed me to really get an idea of what they really are. And I want to thank Eduardo Artavia and Roberto Fernandez. Those are my 2 field assistants who really helped me out, and all the people. I couldn't have done it without you guys. And then Frank and Federico and all the students who helped me. Sophia? Okay, I'm gonna show you. So this is me. I'm a bat, and these are my wings. The baby's essentially gonna hold on to the front of them. Yeah, so it's pretty tricky when you're navigating. Rudy?
I just was wondering when you were talking about the statistical significance and how it could be a result of selective pressures. And you were saying that the females might be like choosing to have these different these differences. What do you mean by choosing? Like, do you think they're like actively like having
That's a good question.
control over their size of bones?
I don't think so. I think that like were you gonna say something?
Yeah, I'm just gonna say it. Sometimes biologists use shorthand, so you can talk about a tree wanting to flower, synchronously with others.
No. Thank you. That's okay. I'm more talking about their evolutionary path to get here, kind of thing. I don't think you or I could like choose to have bigger ankles or something like that. But yeah. Oh yes.
Is this kind of sexual dimorphism like typical in bats as far as like studies other bats that forage more similarly, or like bats that forage more typically?
Okay, that's a great question, and it's kind of the reason why I chose to ask this question about fishing mats. It's a pretty understudied topic from what I found in my research, and differences between male and females aren't necessarily represented in the research. There's a lot of research on bats. What is a bat? Like, what do they do? How do they hunt? Kind of thing. But there's not these little nuanced differences that are being studied, which are so important to like everyday life. Yeah. Thank you.
Yeah, responding, so let's wait. Okay. I think the clicker works. Okay. And this is the phone. Okay. Okay.
Okay, next speaker from UC Santa Barbara, Emily Desrone Boga, and she's going to tell us about interesting aspects of fungi and skin of bats.
Okay, my name's Amelie, and like Fede said, I've been studying bats, but I've specifically been studying the fungal communities that live on bats. So something that's important to know is that all organisms, you, me, plants, vultures, even fish, have microbial communities that live on them or within them. And these are kind of finding their place in the popular literature now as we're studying the health effects that both external and internal microbial communities have on hosts. Within this presentation, I'm going to be using the word mycobiome, which specifically refers to these fungal communities that will live on or within organisms. So why should we care about bats' microbiome? As I said, understanding microbiomes and mycobiomes is kind of a developing area of research, and specifically within wildlife populations, this is also a developing area of research. So what defenses do wild populations have against pathogens other than some behavioral things that they can do or their innate immunity? We think that there could be a correlation between microbiome and their ability to fend off against diseases. And this is specifically interesting in bats because we have a really prevalent disease in North American bat populations called the white nose disease, which is a fungal pathogen that is decimating North American bat populations. But recent studies have found that there are some fungi that may be symbiotic with bats that can have what we think is a competitive effect with these fungi and provide some resistance for these bats. So understanding what the makeup of healthy bats, fungi, and bacterial microbiomes could be an important part to protecting these communities. So I'm just going to go over some of the things that can affect an organism's microbiome. First, we can have the environmental fungal reservoir. So these are all the fungi that are just out in the world that we could pick up, touching something, or if I'm a bat flying around and picking up fruit. we have intermicrobial competition. So if I have 2 microbes living within my ears, for example, for a bat, and they are at war with each other, they can lead to the loss of certain bacteria or fungi, and so that can affect what we see. Next is we have host identity. So these can be a variety of things. Like in this study, we're going to look at a few different factors, but this could be species or genus, lifestyle choice, different lifestyle parts, and also this pertains to like part of this is what environment they are interacting with. The next is interspecies interactions. So what other species are they near, and could, again, this is part of kind of the environmental fungal reservoir sharing these fungi or bacteria with. And so these are just a few of the different things that can affect the microbiome of an organism. And in this study, I'm specifically looking at the environmental fungal reservoir and host identity. So with all these different factors, and with the importance of understanding a normal, healthy bat's microbiome, brought me to my question: what factors, both environmental and host-specific, influence the makeup of fungal communities on bats? So I set out to do this by trapping bats at 2 different sites here in Monteverde, and identifying the bats and swabbing their ears. And from these ear swabs, I would grow up my fungi. Here's an example of some of my beautiful fungi that grew up. And these are 3 different individuals, but they're all from the same species, Sternera hondurensis. And so once I got the swabs and grew up the fungi, I would look specifically at these plates and then divide my fungi into different groups based on morphology. So here, this is like a pretty good example. You can see there's a bunch of different colors, shapes, and these different characteristics I use to divide them into morphotypes. And so for each bat caught, I would look at their plate and I would create like a fungal profile for them. So I would say, this Sternera hondurensis has these different morphology. And from that, we get to our first results. So this is a heat map of the bats sampled and the fungal morphotypes that were found on each. And we can see on the y-axis is my bats labeled with their species. And then on the x-axis are the fungal morphotypes. And if it's purple, that means that bat has that morphotype. So this is kind of a summary. And as you can see, we had some dominant species. And we had 5 species overall that we caught at these 2 different sites. And so what was really interesting to me was looking at what is associated with these specific fungal profiles. So I took metadata while collecting these bats, and then I basically went through a series of analyses to determine which of these factors is important to determine what the fungal profile of bats are. So I looked at species, genus, sex, relative age, and then also I looked across these 2 different locations, which is not a host trait, but it is my environmental trait. So first, basically, kind of a scary graph, but what I did is taking this multidimensional data, I put it through PCOA, which takes this multidimensional data and puts it down onto 2 axes, and it's supposed to, or what it's trying to tell us is if there's an association between the profiles of these fungi, or of the bats, the fungi on the bats. So each dot represents a sampled bat, and each location on this graph kind of is a proxy for a specific fungal profile. So if you see clumping of spots together, that means they share those fungal profiles. And one thing that kind of becomes evident when you look at this is that, 1, there's not very much clumping, and number 2, there's not a lot of data to do a proper ordination plot, which is what this is. And so looking at this based off a few different factors, the factors here, I grouped them by species, genus, sex, relative age. Through all of those groupings, there wasn't any significant association. Through a Permanova analysis, we saw that there wasn't a specific statistical association between any of these groups. But if we kind of move to look at this on a more general scale, the next study that I chose to do was looking at the fungal richness on each of these bats. So this is where we start to see some results that are kind of exciting. So if we look at the mean fungal richness on bats caught over here in the red is Bajo del Tigre, and then over here in the blue is the Estación, And the y-axis is the fungal morphs presence, and the thick bar is going to be our mean. And based on a two-sided t-test, this is statistically significant, and we have a higher fungal richness at Bajo del Tigre. So this was particularly interesting to me because if we circle back to what the different factors that I introduced at the beginning, host identity, environment, of the two that we looked at, based on this plot saying that the bats that we caught at one location had significantly different richness, we can say that there is perhaps a correlation between the environmental fungal reservoirs, the environment, and the fungal profile of these bats. I think beyond this, it's important to mention that there is a bit of a caveat to this. At Bajo del Tigre, we also caught a greater diversity of bats, so that could perhaps be what is associating with the greater diversity of fungi. And this could also suggest that diversity. But this study needs to be expanded, and perhaps using molecular techniques to identify fungi, we could do this in even more depth and get a really nice ordination analysis. Yeah, so that's kind of some future directions, expanding the sample set, examining natural history factors like which bats live together and are they sharing fungi, and perhaps finding a way to parse out what is an environmental contamination of fungi and what's actually a symbiotic fungi, which is a pretty interesting factor, especially when we circle back to understanding white nose disease. Yeah, and then thank you for everyone who helped me, for Fede, Naomi, as my advisors, for Adriana, for my homestay family, for the staff, and all of my classmates. Ruby?
Well, I was wondering if you have any ideas of the environmental factors that could be causing the difference between Bajo del Tigre and the Estación.
Yeah, so there's a lot of different things that can affect like the fungal makeup of a place. One that I kind of briefly looked at in literature review is elevation. There is a pretty distinct difference between Bajo del Tigre and the Estación, and just there's a variety of things that could influence, what fungi are present in what areas. And I think what would be most telling would be to look at like where are these bats residing for most of their time, and is there a fungal association between like this bat roosts here and they all have the same type of fungi. But basically, there's so many confounding factors that could be causing different fungi to be present in different areas that I think it takes like a really widespread study to be able to figure that out. Yes.
Do you know if these fungi like solely live on bat ears, or like were you able to identify them, or do they how do they colonize from other places?
Yeah, so we were not able to identify any fungi. It's really hard to morphologically identify fungi, especially when you're growing it up on a plate. I think to be able to do that, like genetic analysis is really essential. And so I'm not sure if they're growing other places. It's likely that these are environmentally acquired fungi, as most external fungi are. So if any fungi that we have on our skin, we probably picked up somewhere. So just spores can be in the air, they can be things they're touching, but they're most likely environmentally acquired. Yeah. Mm-hmm. Yes.
Are you talking about spores or living fungi?
So this is kind of unclear. They're swabs. They're not visible fungi to us, but like all organisms, bats have like small amounts of, or they have fungi that are present on their skin, and so they could be in the spore form, but they can also be in like a kind of like a smaller growth form, and they basically we were able to visualize them because we put them on these plates that have high nutrients and give them the perfect conditions to grow, but it's not like they're growing in these mats in the bat's ears. Mm-hmm.
There are some questions there.
Kaylin? Oh.
In the lab, I remember you did some swabs of some of your classmates.
I wish I had the photo up here. Yes, we swabbed eight humans, their ears to compare them to fungi. Sadly, they were not as pretty as my bat fungal plates. Sorry. There was one student that had more ear fungi than anyone else, but I'm not gonna say who. But there are actually a lot of studies looking at the fungal communities on humans and proposing that perhaps mammals have conserved fungal communities. So if we were to look at us versus bats, there may be some similarities between our ears and bat ears. Yes, Frank? Yes. Yes.
Maybe a quick suggestion, if you haven't already done this, and that is for species, or at least genera, Balotigre and higher up, did you actually do a species by species comparison to see who had more fungal morphospecies?
Yeah, I did. I did it both with richness and using a PCOA to look at, except PCOA, we don't have enough data for that, but sorry, yes.
I'll show you what I had in mind. It's not sort of a bigger analysis. It's a species by species thing.
Oh, okay, yeah, great.
This is for you. Okay. Here. This is the clicker. This is what everyone sees. This is the forward slide.
Okay.
And
Does
this
it-
works forward, backward, and this is the laser pointer.
Okay.
Okay.
In this case, it's going to happen a new analysis and stuff like that. So take advantage of this and look at a positive input. Okay, next speaker from UC Santa Barbara, Noel Ross, is going to speak about an interesting aspect in neurology in coatics.
Thank you, Fede. Hi, I'm Noelle. I'm an undergrad. I'm an undergrad biology student at UC Santa Barbara, and I decided to do my study on white-nosed coatis, scientific name Nasua norica, and how they interpret potential threats in their environment. So just an introduction, when I was developing this study, I really wanted to focus on a perspective that's often not highlighted when it comes to field studies of mammal behavior. A lot of us know of traditional behavioral ecology, and I also am going to explain it as the outside-in approach, in which we look at an animal's environment and interpret their behavior based on what we're seeing externally. On the other hand, we have neuroecology, which is the inside-out approach. Instead of assuming that behavior is just a response to the external environment, we look at internal neurons and also take behavior and look at it as just a clue to sensory processing. Here, I decided to use coatis because they are best suited when trying to translate behavioral patterns into a neuroecological perspective. They are known for their exploratory behavior. They have social complexity and also keen sensory abilities, making it very easy to connect their behavior to the brain. So some study objectives here is I wanted to go in and examine how coatis respond to a novel, non-threatening stimulus. Also look at their behavior as a proxy for internal sensory, and compare coatis with sympatric agoutis, which I started to study along the way as they were a little surprised to me. So in addition to that, I wanted to evaluate the influence of some key factors, two of them being more deeply rooted in ecology, the other one's kind of leading into neuroecology. So for the first one, we have environmental context. We can look at different environments, use that study site, and connect it. We also have sex, age, and class, which is a demographic used when looking at external environment. On the other hand, we have social structure, which it connects more to the neuroecology sense, as there are solitary males as well as juvenile and females within bands. So looking at the difference between those two can also help us identify what's really going on here. And we have phylogenetic differences, which is a huge piece of neuroecology. It has a lot to do with internal sensory, but I'm also going to add in something called phylogenetic inertia, and we'll see more about what that means. So using all these things, the approach was to integrate neuroecology into a comparative method, looking at all of these things, maybe doing a process of elimination to see what is the big thing going into this research question. Is a wild coatis threat response shaped more by environment, evolution, or neural processing? So my methods, I had two different areas that I conducted field studies on. I had the Estación Biológica as well as the Bajo del Tigre area. At the station, I had the lower compost area, the upper compost area, as well as the greenhouse. And this is an incredibly rough estimate of Frank's property and where I put the sites there. So we have Frank's shed somewhere over here, as as well as the garden patch somewhere over there. So there are 5 field sites. The experimental setup was to use a rubber snake, which was a visual stimulus, placing it on the ground, and also placing food lure beside the snake. I then set up a camera trap in order to capture these moments, also to minimize human presence and have a consistent setup throughout all of these different environments. So also I took the behavioral variables down of sniff duration, hesitation, and reaction type. Not all reactions are something that we can visibly see or just automatically pick up because of movement. It can be them ignoring something, a brief investigation, sustained investigation, to freeze or retreat. And just a little side placement of this, I also looked at social context, so the difference between a solitary male and then band organization. So for results, I had 29 total trials. Coatis were present in 22. There was comparisons possible among the adult males, adult females, and juveniles as I collected data for all of these groups. We once again collected sniff duration, hesitation, and investigation. There was also agoutis present in 7 of these trials. A lot of the time they were snatching the lure, and I thought it was very, very mean, but it ended up being a really, really good thing when looking at this study. The agoutis showed 0 hesitation or investigation toward the fake snake, and that's why I was able to use agoutis as the baseline. Here are the agouti decision pathway versus the coati decision pathway. As you can see, it's linear. There's nothing going on there, just them completely ignoring the fake snake. Looking at this complex graph, we can see that the percent investigation of the coatis was 100, and the agoutis was 0. The coati showed high curiosity and threat evaluation behavior, and the agouti showed complete avoidance. Second graph, a little bit more difficult, was the sniff duration of coatis during the fake snake trials. We have the 3 different groups there. All of them within the same sniff duration, but the ranges varied across the different groups. The females having a 2.5 to 4.5 sniff duration, juveniles having a smaller one, and the males having one in between that. Looking at the juveniles, I wasn't expecting to see that the juveniles had the shortest sniff duration. I was actually expecting the opposite. But looking at the social context within the bands, I saw that the juveniles would only investigate the snake once the female had already approached it and made sure it wasn't of a danger, which was really awesome to see. There was one male outlier up there, which was 8 seconds of sniff duration, very curious, and then one at the bottom as well. So some results. There was some key behavioral findings. The coatis and agoutis show markedly different investigation patterns toward the same stimulus, and these differences reflect species-specific approaches to assessing risk. In addition, there were environmental differences. So I wanted to use Frank's shed because it had the most unpredictable disturbance. And within that, I saw longer sniff durations. And in the lower compost area at the Estación Biológica, I saw that I knew that it was a familiar space, and in result, it translated in short, repetitive investigations of the coatis. Just to highlight here, there were differences in sniff duration at both of these sites, but at the same time, the fact that there was sniff duration at all kind of shows that there might be some underlying things when looking at the sensory. And because of this, I wanted to look more at the comparative species contrast, in which the responses reveal fundamental differences in sensory evaluation across species. So for my discussion, I interpreted these threat response, the coatis treating the model snake as the novel object. There was sensory filtering and social information use, and the complete non-responsiveness of the other species made it very clear of this thing called phylogenetic inertia, which shows that there is evolution within a species, but environment is not the only factor here. There's also this idea that there is a select range in which a species can evolve. And why this supports neuroecology, it shows us how each species' nervous system prioritizes sensory cues. We saw a lot of social cue following, especially in between the bands, and there was sensory processing, ecological context, and evolutionary history. Also, this was another huge thing that I found that I think really helped my study, which was the Darwinian comparative method. It clarified why related species differ in their threat responses. I talked about phylogenetic inertia, which this article emphasized. Their behavior shaped the evolutionary constraints plus ecological relevance, not just immediate context. And the coatis versus agoutis demonstrate that similar environments do not produce similar behaviors. So it shows species sensory / neurosystems and how they determine and interpret risk. Acknowledgements, I'd like to thank Federico as well as Emmy for challenging me to look more into this study and find my path and how to go. Frank Joyce for allowing me to use his fake snakes. The Estación Biológica, EAP, all the instructors there, as well as CIEE for the camera traps. In addition, Gray, very awesome job helping me do the camera traps and data collection. Haley and Antonella for showing me how to set it up and also helping me when it comes to camera positions. Oh, Taryn?
I might have missed this, but why did you choose to use fake snakes? And do they tend to prey on coatis?
Yes, they do tend to prey on coatis. I wanted to use fake snakes because when doing research about this project, I was introduced to this thing called coati bodies, which are cytoplasmic inclusions within the coati's brain. It was done on a captive coati, and with this, these inclusions were PMNT positive, which means that they had norepinephrine synthesis, and that triggered a fight or flight response. So I wanted to find something that was scary, and snakes being one of the predators, being a potential predator of the coatis, I wanted to investigate that. Leo.
Two questions. Do coatis feed on snakes? And can you define Keen's sensory abilities?
Do coatis feed on I think when talking to Federico, I heard that in a band they were able to attack a snake, but I'm not sure if they completely feed on it. I don't have full information there. And then you said keen sensory abilities.
It was in your first snakes.
Keen sensory. Let me look back at it. Oh, I know exactly what you're talking about. Keen sensory abilities. I was talking about the olfactory response within the nose and how they sniff out things like that. Yeah. Yes.
Do the agoutis and the coatis have more like the agoutis, are they more olfactory based than the coatis in foraging? Because that could, I was thinking that might explain the difference in recognizing the other thing.
Yeah, at least based on what I saw, the agoutis lacked that olfactory response. They weren't very tentative to things on the ground when it came to sniffing. It was more like see what they want, grab it, and go. And then on the other hand, coatis were very like investigative. So yes, there is a difference there, but I want to say that it was more of an evolutionary trait, and because of that, it kind of connects back to that neuroecology point of view. Does that answer? Sorry.
I guess I was asking, of course it's an evolutionary trait. I was asking that, like, do they forage more by smell, or do you know if they forage more by smell and sight than the coatis?
Okay, sorry. I'm not 100% sure. I didn't do much research when it came to the agoutis, but that is a really interesting point to add. Yeah.
I'm sorry if you have questions, but we need to stop now. Thank you.
Thank you.
Yeah, that's a very interesting question. Yes, we decided to have very few sets of examples. Old fashioned.
Sí, pero lo está abriendo. This is for you. I'm gonna put it here.
You were thinking how this agoutis didn't pay that much attention. Next speaker is going to tell us about how coatis are advertising everybody around. So, Adela Corey from UC Santa Barbara, thank you.
Thank you, Fede. Hi, thanks, Fede. My name's Ella, and today I'm going to talk about inter-individual variation in chirp vocalizations of white-nosed coati. So Noelle talked a little bit about coatis, but they're in the family Procyonidae, related to raccoons. Unique to their family for multiple reasons, one of which is their social structure. Females and juveniles live in bands or social groups called bands that are comprised of anywhere from 4 to over 30 individuals. And around the age of 2, males will leave their mothers and life with the band to live pretty much entirely solitary. Additionally, they are diurnal, active during the day, and they spend around 90% of their day being active, foraging on the ground and low in the trees, and then at night they'll climb higher up into the trees to sleep. So they are super social, which means they are also very vocal. They have a whole plethora of vocalizations. One that they use quite frequently is a chirp. A chirp is characterized by having a high frequency and short duration. They use it all the time. They use it during locomotion, foraging, even during rest sometimes. And it is assumed to be a contact call. The structure of a chirp is useful as a contact call because it is a short duration, so they're not attracting predators, or predators aren't able to locate them, but they're able to communicate in dense areas where they usually frequent. So this, all observing of coatis talking to each other led me to the question of, do coatis have unique voices? And I wanted to discover that if there was a variation in coati voices, if there were any patterns that might point to the functionality of this variation. My process involved a whole lot of waiting for coatis, not as much finding, but when I did find them, all my data for this project is used from the Estación Biológica, though I did have multiple other study sites. Anyways, when I did encounter the coatis, I used a directional condenser microphone connected to a recorder, and so I was able to isolate the chirps of the coatis. I also simultaneously recorded on my cell phone so that I could later go back and cross-reference the vocalizations with individuals. I collected a total of 18 vocalizations from 6 individuals, so 3 chirps per individual, from 4 adult females, 1 adult male, and 1 juvenile male. And then to analyze these chirps, I split them up. This is a sonogram of a chirp. I split the chirps up into 3 elements. Element 1 is a high frequency element, element 2 is a mid frequency, and element 3 is low frequency or low to mid frequency. And all the chirps contained these 3 elements, but they varied in shape and frequency. So here are my results. On the x-axis, we have the elements 1, 2, and 3, and the y-axis is their frequency, which the top number is 20,000 hertz. So each color represents an individual, and each box plot is the interquartile range mean and the distribution of each of the 3 calls from each individual in that given element. So you can see that there is a kind of consistent pattern within each Kawadi's vocalizations. So I did some statistical analysis. I ran 12 separate linear mixed effects repeatability tests, which test for repeatability. Repeatability is essentially the proportion of variance that can be attributed to differences between individuals rather than within individuals' vocalizations. So the 12 tests were comprised of 6 for comparing all individuals. I compared both the low and high frequency elements or high frequency components of each element. And when comparing all individuals, repeatability varied from high to very high, which signifies that there is distinguishability between chirps in Kawadis. And then I reran those tests again to compare only females. And interestingly, the results came back even stronger. And now we get to discuss it. Here are 4 of my study subjects. Naomi and Emmy are both adult females. Leo and Frank are a juvenile male and the adult male. This is another representation of what chirps look like. There's kind of tape on that one, but hopefully you can see that the structure of the male's calls are actually very similar. And when you compare the females, they have more of a unique shape. So an interesting element I found in the males is that the juvenile had a component in the ultrasonic range, which I did not document in my data, but just something to point out. And along with the fact that the male produced the lowest frequencies, this is consistent with the assumption in bioacoustics that larger animals produce lower frequencies and smaller produce higher. Okay, back to the differences. The similarities in the males and the differences between the females. A possible reason for the fact that females have a more significant variance in their vocalizations is that their lifestyle is more dependent on individual identification because they're so social, whereas the males don't necessarily interact with Kawadis as much, and so it might not be as useful. So to conclude, there is inter-individual variation in Kawadi vocalizations. They have their own voices. Some possible functions of this variation is, like I talked about earlier, group cohesion. It allows them to know the dynamic of who is around them when they're traveling in groups together. It allows them to recognize individuals within their band, but more importantly, it could allow them to identify unfamiliar individuals. And because coatis are so mobile, they overlap in territory a lot, both bands and individuals. And so knowing when there's a potential conflict is important. I would love to have done so much more research on this topic. I would have loved to analyze more vocalizations and also conduct playback experiments in which you play unfamiliar calls to coatis and analyze their behavior. This topic is important not only because it helps us further our understanding in the cognition and communication of coatis, but it also helps us understand how future for increasing human interaction and noise pollution might affect the lifestyle of these creatures. Lastly, I want to give a huge thank you to my advisors, Fede and Emmy, for all their support. Also Frank and to the Hotel Belmar, which were two sites that I did not use in my data, but spent much time trying to collect data at. And Elsa and the staff, Elsa was taking care of Frank's house at the time, and the staff at the Belmar who were adamant in helping me find the coatis. And I just want to appreciate the Institute for providing this opportunity. These are a lot of cool things that I got to see while not seeing coatis. Thank you. Oh, sorry. Oh, it.
Interested in experimenting with unfamiliar sounds. What do you mean by that? Like coatis from a different place? Because I know that different like dolphins and orcas have accents if they're from different locations. So would you be interested in that?
Yeah. So I actually I had hoped to be able to do this in this experiment, but was challenged. I was hoping to collect data from multiple different bands and play back unfamiliar calls from one band to a different band to observe how they might react. Yeah, Monica? Yeah. So what I tested for was to see how much of the variance in their calls could be attributed to variances between individuals. And so this showed that within an individual it was very consistent, and so the majority of the variance was between individuals. So yeah. Yeah.
So their social structure, do all the females take care of all of their pups, or are they unique and are they thus possibly communicating with their own pups and band groups?
There is some research into this. There is a group dynamic in which it's not just the female taking care of her pup. It's definitely a group effort. But I'm not sure how specific that is, so I don't know how much I could speak on that. Yeah. Yeah. Thanks. Thank you. Thank you. Thanks. Thank you. Thank you. Thank you.
You can keep the mic too.
Thank you. Not everyone can hear me. Thank you. Thank you.
Bueno, por esta sesión sí.
Sí, es la última.
Thank you.
Okay.
Esta es la última de esta sesión.
Okay.
Muchas gracias también.
Okay. Yeah. Thank you.
Are you ready?
And yeah, this is forward, backward. This is the laser pointer. Okay. Yeah. This doesn't work, but I'll use this. All right. Yeah. Okay.
Okay, we're ready for the last presentation of this session about biology of mammals. And our speaker from UC Berkeley, Greg Comersford, is going to tell us about the movement of mammals, how abundant they are, and how to understand farms and forests living together.
Yeah. Thank you. Yeah, I'm Gray, and I will be talking about mammalian habitat connectivity in Monteverde, Costa Rica. Oops. So we know that Costa Rica is one of the world's leaders when it comes to protected area. Way back in October, when we first stepped into this room for the first time as an EAP group, we learned that around 28% of Costa Rica's land mass is protected area. And it's a super impressive number and really, really great for Costa Rica's wildlife conservation. But what if I told you that the future of wildlife conservation doesn't only rely on these protected areas, but also on the choices of local farmers? In Monteverde, agriculture often fragments essential wildlife corridors, but my study focuses on an alternate perspective. By examining mammal habitat use across a multi-use landscape and comparing a protected reserve with an agricultural farm, I'm hoping to study what the differences are between habitat use in these two places. So this led to my central question of what are the sighting patterns, temporal dynamics, and species richness of mammals in the Bajo del Tigre protected area and agricultural fields in Finca Laguna Verde. And so this is a map of my study sites. In the yellow points, we see the names of the sites. We have site 1, which is Finca Laguna Verde, and site 2, which is the Bajo del Tigre Reserve. And in blue, we have each camera site documented by number. And some back.
No, it's
I really.
I think it's closed.
Oh no! What?
I would check. It's
Okay.
closed at 5.
Why am I so
After time again. He doesn't learn. He never learns.
Okay. Huh? I'll ask him. Yay. Yep. Frank. Next.
Friends presentation.
Yep. Classic. We're live.
Wait, but
I'm gonna shout.
Is it your
Huh?
I'm about to
Yeah.
shout you and Jared and Gray out in my
Yep.
presentation. What? You guys are talking about conservation is important and biodiversity is important, and I'm talking about why pesticide is bad biodiversity, and it because it diminishes all that, and why we gotta not do that. And
Uh-huh.
and you guys talk about that.
It's all connected. Thank you.
oh,
Yep. Do I need the clicker? Okay, I could also just do this, right? Oh,
yeah.
Stop.
But you can also just like click.
Do you know why it's like this?
I have no idea. Maybe because it's a PDF.
Okay.
Right. We're gonna start.
Okay.
All right, thank you everyone. And we're coming back to our very last session, which is going to be about agroecology. And we're going to start with Naomi Prince, who's going to teach us about the effects of compost in growth rates.
Yes.
Plant growth rates. Thank you, Naomi.
All right, thank you so much, Emmy, for the introduction. My name's Naomi, and I'm going to be presenting on my research on radishes, Raphinus sativus, growth rate in different types of compost. And here are some images of the radishes at different times in their life cycle throughout the 2 weeks. So first, it's going to be really important to give you guys a background on what composting is. So I have 2 images here. The one on the right is the compost piles at Life Monteverde, which I used in my project. So Life Monteverde uses a conventional compost method, and then the institute here uses a vermicomposting method. So conventional compost is where essentially big piles of organic matter are collected and create heat to break down organic material. They have 3 phases. First is the mesophilic phase, where the microbes start to break down, and it's normally at 20 to 40 degrees Celsius. And then it really starts to heat up and get working and breaking down tougher organic materials in the thermophilic phase, which is from 45 to 70 degrees Celsius. And then the third phase is the maturation phase, where it starts to cool down again and the nutrients stabilize, creating a rich organic fertilizer. Over on the right is a vermicompost from the Institute. Vermicompost is a composting method using worms, typically Esnia fetida, as you can see in the image right there. And the worms essentially eat the organic matter and digest it and create worm castings that are very rich in nutrients and can be used as organic fertilizer. This is a cooler and more aerobic environment. And these are the 2 composts that I used for my project. All right, so my research question after visiting some of the farms in Monteverde was, what compost type is most productive for seed germination and growth? And you can see all of the pottings here. I'll talk a little bit about the different treatments I did in order to test this.
So why did I choose radish as opposed to kale or lettuce or any other type of vegetable? This is the seedling on day 3, day 7, and day 11. They grow really fast, and I wanted to compare not only growth rate, but also germination, and they were one of the only types of seeds that I could do this with in such a short period of time, so that's why I chose radish. All right, so now on to methods. On the top right, you can see the seeds that I used, and essentially I had 2 days of potting, and in these 2 days, I planted 10 control pottings and 10 of each of the compost treatments with the help of some of the CIE interns as well as Esteban. And the 10 different treatments, there's going to be 5 treatments of the vermicompost and 5 of the conventional compost from Life. The conventional compost from Life actually also adds, I forgot to mention this earlier, but biochar, which is a form of charcoal that they use from their prunings from their coffee trees. And the 5 of each have different compost to soil ratios, 20%, 40%, 60%, 80%, and 100%. And then I set them up in the greenhouse in the Monteverde Institute and watered them with a little spray bottle, and then I measured their above ground growth in centimeters per day. All right, so a little bit about my results. My results have 2 sections, the germination section and the growth rate section. And I'll start with the germination section because that's earlier on in the life cycle. But this graph right here shows on the x-axis is the type of potting and the y-axis is percent of seeds germinated. We had a really high success rate. 94% of the vermicompost pottings germinated and 72% of the life compost pottings germinated. And the ones that are missing here, the vermicompost 80% had 70% germination, and then the life Monteverde 80% had 20 and 100% had 40% germination. All right, so the next graph that I have here is the average height of the treatments through from day 6 through 10. I started on day 6 because that's when they were germinated and had a steady growth rate that I could compare. And on the top, you can see the vermicompost 20% was consistently the tallest, and then the bright green kind of in the center is the control, so you can see what was better than the control and what was shorter than the control. And I'll talk a little bit about my hypotheses for why some of the treatments didn't do as well. All right, so next is my graph on growth rate. And on the x-axis is the compost to soil ratios. In yellow is the life compost, and then green is the vermicompost from the Institute. And on the y-axis is growth rate in centimeters per day. So I have some stats. The winner, so to speak, was the Life Monteverde 20% with 1.47 centimeters per day. Next was the Life 60% with 1.416 centimeters per day. And then coming in third is the Vermicompost 20% with 1.32 centimeters growth per day. And all 3 of these had 100% germination rate. So this is kind of like the top 3 compost to soil ratios and types of compost. I also conducted a one-way ANOVA test, and I got a p-value of less than 0.01 with an F statistic of 27.94, which means that it is statistically significant and that the type of compost and compost to soil ratio does impact the growth rate of the radishes. All right, so now moving on to discussion, I really wanted to talk about the kind of unique strengths and weaknesses that I found within using the vermicompost and the conventional compost with biochar from Life. In some of my background research, I learned, or like research after my project, I learned that biochar is known to retain water and nutrients in soil and increase carbon sequestration. And so this is one of the pros of using this as a form of natural fertilizer in your compost. And this is really good for what Life does, where they add their compost to their bigger coffee trees and helps with water retention for those like deeper roots. And then the vermicompost is known to be richer than regular conventional compost in a lot of macronutrients and micronutrients. And so this is really good for earlier on in a seed's life cycle for really getting it going and germinating like the Institute does with their smaller seedlings. So this kind of shows how different types of compost are better for different types of farming. And then I also had a few more questions that I wanted to ask and go over.
as we talked about in one of the earlier graphs, there was some growth rates that were actually lower than the control, and this posed the question, how did potentially too much compost inhibit the growth and germination? And so some of my hypotheses for these are that the density of the compost is denser than normal soil and might have created waterlogged soil, which created like excessive water retention and didn't allow for proper drainage. It could also possibly lead to too much nutrients could lead to a biome that allows for fungal growth, which could inhibit the growth of the seedling. And then a few questions that I would love to look into if I had more time or for further projects looking into regenerative ag and composting are how can industrial farms incorporate regenerative agriculture practices and reduce chemical inputs? So this is really important when looking into how we can kind of transition our farms into a way that works with the environment and with the earth rather than breaking it down by using chemical fertilizers, which are very commonly used. And then how do the microbial and fungal communities of plants that use vermicompost versus conventional compost differ? And yeah, this would just be really interesting to look into the more like microbiology of the different soil to compost compositions. And then I would like to acknowledge some people that this project would not have been possible without the help of. Naomi and Emmy were my two advisors. They helped me so much. And then Esteban from the Institute provided me with a space to put my pottings as well as help me with my pottings and a lot of advice on agriculture with his background knowledge. The Monteverde Institute, Jerison and Deanna, and Life Monteverde for giving me the compost that I ended up using, and then my host family. All right, I don't know why the thank yous don't show up, but next. Oh, thank you. And this is also a picture of all the pottings on the last day. They're pretty tall, so. Avery?
I was curious because when we went to life, I learned that compost had like a certain temperature it's supposed to be at, and I was wondering, like when you were transferring the compost, did like you try to maintain temperature, or was that something you didn't measure because you were like taking it out from this pile, and like if so, did it affect?
So when I got the compost from Life and from the Institute, I got them when they were complete, so in their like maturation phase, which is after they've cooled down and the nutrients have stabilized. So yeah, but in conventional composting methods where they have the piles, it gets up to 70 degrees Celsius, which is really hot. Like you can see steam literally rising from piles when you kind of open them up. But the vermicompost doesn't get hot because they don't use heat to decompose. They use the worms' digestive systems. So. Yeah.
Does the species of worm make a difference? So do people vary their worm species?
That's a really good point and something that I looked a little bit into. But the Esnia fetida is the most common, and it's because of their preference for consuming like organic materials. I don't know too much about that, but I would love to look into it. I do know that other vermicomposts have used different species of worms, but I would assume that would more be based on like the availability in the area, because you can vermicompost anywhere, but you maybe can't get any worms anywhere, if that makes sense. Monica? Yeah, the pottings were used for, so the soil and compost ratios were used for some medicinal pottings in the Institute, but the radishes, they went to bed. But yeah, I would, in the future, if I had more time, I would love to do like a similar research project where I got to see them reach maturity and like harvest them and maybe measure like the weight of the root vegetable and the length of the taproot. Yeah.
Any other questions? No?
Yes. Oh, everyone forgets. Thank you.
Yeah, Sarah. It's not my turn.
Right.
This isn't Sarah.
No.
Oh, it's not Sarah's.
Hola. Here, this is the clicker thing, so forward, backward, and this is the laser pointer if you want it this way.
Okay. Perfect.
All right.
Our next presenter for this last session about agroecology is going to be Antonella Alarco, who's going to teach us about repellent in cattle. Thank you, Antonella.
Hi everyone, my name is Antonella and today I'll be talking about 3 different pest repellents in 3 different dairy farms and seeing the abundance in pests and seeing their stress-related behaviors to those pests in dairy cattle. Yeah. So some background information is that all over the world, most dairy farms are managed by chemical pests, and over the years, it has been released that chemical repellents have been detrimental to the Earth's ecosystem, especially their soils, and also harmful to animals, especially the cow's physiological impacts. And so people have been transitioning to more natural-based repellents, and these natural-based repellents have been more food additives that these farms have complemented. So my study will focus on 3 farms with 3 different approaches. The first farm is Finca Paraíso that feeds their cows 40 grams of garlic and aromatic plants. The second farm that I will talk about is called Finca Pablo Herrera, which is a chemical pesticide called Cengage that he gives every month when he sees an abundance of pests on the cow. And then the third farm that I'll be talking about is Finca Bello Amanecer, which is a sulfur-based additive in their diets and gives monthly sulfur baths. All of these farms are based in the province of Punta Arenas, and the pests that Costa Rica has an issue with, at least that the cows suffer with, are flies, ticks, and vampire bats. And the common behaviors that they express when these pests are around them are head tossing, skin twitching, and kicking into the abdomen, as well as constantly licking themselves and leg lifting and tail flicking, which a past study has done before about. So the question that I wish to address today is, do insect repellent strategies influence stress-related behaviors and pest abundance in dairy cattle? So the way that I measured this is that I randomly selected 5 active lactating cows at each farm. I considered active lactating as if they were being milked twice a day, and I recorded their ear tag number to ensure that I was keeping constant with the subject. And then I would go to that farm, observe them for a total of 30 minutes, and every 2 minutes, I would record the stress behavior that they would express or display among those farms. The behaviors that I took notes of was head tossing, which what I described as when the cow rapidly lifts, jerks, or swings its head up and down in a repetitive motion, as well as when they twitch their ears back and forth. And then licking of their skin is when the cow grooms its skin for more than 3 seconds. Skin twitching, it's the contraction of the skin, and then kicking into the abdomen, which is when the cow abruptly lifts its hind legs to strike its udder area. Those are the 4 that I'm going to talk about today due to time constraints. And then I also logged the number of pest abundance I experienced among each cow. So for flies, I also observed each cow for 30 minutes, and every 10 minutes I counted how many flies I visually see on their backside of the cow. And then for number of ticks, I looked at their right side of the cow from their hook to their wither, and within 5 feet, I counted how many ticks there was. And for vampire bats, those are really noticeable because they're normally 5 to 10 millimeters long, and it's a bite, and you can see like blood dripping down on the cow if they have a vampire bite. So I know this looks like a lot, and I realize I should have made it bigger, but I'll explain each of my results. So the color of the bar represents a different farm. So the green bar represents Finca Bello Amanecer, which is the sulfur-based farm that feeds their cow sulfur to
as a repellent. And then the purple farm, the purple bars, is the Finca Paraíso that feeds their cows garlic, and then Finca Pablo Herrera, which is the synthetic one. And on the left side, you can see their head tossing behaviors, and then on the right side, you can see their licking behavior of the individual. There was no significant difference between both farms in displaying these behaviors. So, and then there was an outlier in Finca Pablo Herrera, which suggests that one cow appeared to have a higher result of displaying that behavior of head tossing on the left graph. It also just tells me that, performing both of these behaviors also affect their time grazing. So the more time that they spent grazing and the less time that they affect grazing also affects their milk productivity and their quality of life. So, sorry, I'm nervous and I'm blinking out, but yeah. So there was no significant difference amongst these two behaviors as the p-value was greater than 0.05. So another behavior that I took note of was skin twitching and their average kicking to the abdomen. There was also no significant behaviors among these farms. However, you can notice that in Finca Pablo Herrera, there was zero display of average of the kicking to the abdomen. This behavior is mainly displayed as it's more of aggressive, like if it's being very persistent to the cow, these pests, and then less cows in general perform the kicking to the abdomen behavior, meaning that they had less pests in that lower part of their body. And then there you have skin twitching was a baseline defensive behavior for them that was presented in all farms that should suggest that these flies were present and regularly irritating, but more to a manageable level. What was interesting to me was the difference of pest found in each farm. So in Finca Bello Amanecer, which did the sulfur-based farm, you can notice that they had way less ticks in the cow compared to the amount of flies. There's a statistical difference between all the farms with ticks. So it kind of is telling me that sulfur is really good if you want to get rid of ticks. And then comparing to Finca Paraíso, which was the garlic-based farm, it was also good at reducing ticks, but it was even better at reducing flies. And then looking at the synthetic farm repellent, which was the blue graph, which did the best in reducing flies. So there was no significant difference in fly abundance using garlic or chemical repellents, and there was not enough data on vampire bites to conclude an effect because they weren't as common in the farms. So to conclude, stress-related pest behaviors were not reliable indicators of pest load because there's a lot of factors why cows get stressed out, and those behaviors don't necessarily indicate it because it's due to pest. And then sulfur-based feed supplements were the most effective at reducing ticks, how I mentioned, and then garlic-based supplements showed moderate effect among ticks and flies. And then natural repellents show a promise as alternates to synthetic chemicals, but effective depends on the pest types. And then these findings can help farmers that are transitioning to a more regenerative approach and helping the Earth's ecosystem, and yeah, conservationalist. And lastly, I just wanted to acknowledge Fede, Naomi, and all the farms for letting me participate, as well as Noel and Haley for coming. Thank you. Any questions? Yes.
Do you think there's anything more you would have done or changed depending on if you were given a longer timeline?
Yeah, I honestly only had 7 days to conduct this study because it was hard to get approval from these farms. But what I would change was I really wanted to do more lab work. Like I wanted to do a hair cortisol to actually see if there is difference in stress. Yeah, so I would implement more lab stuff, more lab work, if possible. Yeah. Yes.
So the sulfur-based additive, what kind of sulfur-based additive was it? Was it like the antibiotic was based?
It was a powder that they added on top of their diet that they were giving regularly. Yeah. Yes.
Do you have any idea how the stress level of, say, cows in the Monterey zone compares to stress levels in, let's say, California or cows in California, not people, but or other places? Because I'm wondering if the cows are, I don't know, certain of those 3 farms are all kind of in the same range.
Yeah.
And yeah, do you have any experience with that?
Well, at least here they were like herded in groups of 11. So I know that the ones in California, they're normally really big farms. They're like 40. So being around a lot of them can regulate more stress. Yeah, I don't know. Yes. Yeah.
Did you happen to observe which treatment resulted in the stinkiest breath?
And the what?
Stinkiest breath.
Oh, no, sorry. Yes.
Sorry, this is more of a comment. It's pretty remarkable that the synthetics don't do anything better than like garlic or salt. So if you like had salt and garlic together,
Yeah, I kind of wanted, yeah, I wanted to help like, I wanted to show that synthetics are, or no, natural repellents are equally as good as synthetics repellent. That was like the purpose of my study, but yeah. Yes.
Do you think if you combined all the treatments, would it be most effective at keeping everything away, or would it be a more mediocre medium of all of them, not really satisfying in one category?
That's a good question. Like, I would love to measure pest abundance in a farm that does both sulfur and garlic and compare that. But yeah, that would be a future study. But yeah, I would expect to find a difference in results. Yeah.
Last question.
No, can I answer?
Oh, yeah.
Joyce will answer.
So on our farm, we do combine them, but we combine them differently. We give them sulfur to eat to avoid the bites from vampires, actually, not so much for flies. And we use garlic with red strings. We use mashed up garlic, put it on red strings and tie it on them to avoid a vampire test too. So there is a combination. There's more you could do to test.
Yeah.
Thank you.
Thank you guys. Thank you.
Okay. That's great.
Whoa, it looks so yellow.
I don't know why.
Why is it yellow?
I think it's Leo's computer.
Leo, what did you do?
Como está hecho para que se vea amarillento a esta hora? Se refleja en
What are you taking? It's okay. It's not a big deal.
It's for everyone to rest their eyes.
I mean, they can. That's totally fine. The yellow doesn't really make any difference. It's green. As long as the yellow's not too bright for you guys, then like it's okay. Oh, I see. Okay. Present.
Oh, yeah. Okay. Not a big difference, but
Where's the clicker?
there's a clicker, forward, backward. This is the next slide. It's what people are looking at.
Awesome.
All right, thank you everyone again for your patience. Our next presenter is Sarah Stout, who's going to teach us about the potential effect of castor bean plant, on leafcutter ants.
Yes.
Thank you, Sarah.
Thank you, Emmy.
Thank you, Emmy. Yes, I'm Sarah. I'm studying plant sciences at the University of California, Santa Cruz. And in the last month or so, I have studied the effect of castor bean plants on leafcutter ant herbivory. So why did I choose this study? On our agroecology field trips, I noticed a lot that there were leafcutter activity as a problem for farmers, and with lots of herbivory and pest problems, they tend to warrant the use of pest controls. And many farmers are steering away from the use of pesticides and looking for alternative uses that are a little more environmentally friendly. Farmers and restorers alike in the Monteverde region and all of Costa Rica are working really hard to restore their land to healthy natural states, and described in Jared's, Finn's, Antonella's, and Gray's presentations all discussed why bringing back natural states of nature is really important. So yeah, let's see. So organic alternatives are really important for water quality, soil quality, and the overall health of humans and any species that live. So I started to think what natural methods could deter the leafcutter ants. And herbivory in the tropics is really special because there's really fast rates of evolution and a lot of specialized insects that are like to go after specific plants, and plants that have developed secondary compounds to combat these different insects. Sorry. So leafcutter ants make up 25% of all herbivory in the tropics, which is really, really insane. They're very persistent and they have a preference for young leaves, so it's not great for farmers who want to keep their product yields up, and it's not good for restoration efforts in treelets and saplings in the forest. It's important to note that the leafcutters feed on fungi, not the actual leaves. They farm the fungi and feed the leaves to the fungi. So Ricinus communis is also known as the castor bean plant. It's in the family Euphorbiaceae, and it's very resilient. It caught my attention because it's invasive to the tropics and it has paradoxical uses in cosmetics and medicine and lethal gases. So I thought, how could all of this come into one? But the processing of the secondary compound in Ricinus communis is what determines that outcome, and that compound is ricin. So all of this combined, persistent pests, invasive plants that have insect deterrent effects, and a demand for natural methods of pest control led me to ask the question, how effectively do castor bean plant solutions deter leafcutter ants? So I studied here in Monteverde and in San Luis. In Monteverde, I studied at the Gwindon farm, and anywhere in town I could find a trail. So sometimes you'd see me sat on the side of the road for about an hour. And in San Luis at Finca Florencia and Finca Elisa Mata. So I made 5 different solutions using the castor bean and 2 methods of preparation to begin with. One was the fresh plant or the fresh solution, which I harvested castor bean and blended it with water immediately and let it soak for a couple of days. And the dry was I harvested the plant material, let it dry, and for about 3 days blended it up with water and let that steep again. There were 2 different compositions in these solutions. One was the partial plant, which was just the stem and the leaves, and one was the full plant, which was the stems, the leaves, and the flowers. The only solution that I didn't make was the oil, because making the oil can sometimes be dangerous because of the ricin content. So that was bought by Emmy from the store, and I mixed it with water as to give it a broader area when I applied it. There were 2 experiments that were run and ways of application. So one was the piles treatment. I found and identified leafcutter ants' leaves and ripped them up into 4 piles of 15 pieces, treated 2 of the piles and controlled 2 of the piles. They were left for 40 minutes, and I counted how many leaves were left. They were set up in that order you see over on the right. The second one was applied to stems of kale experiencing herbivory, and I would only do a portion of the stem, wait 10 minutes for it to dry, and then do a 10-minute observation period to observe their interactions, the ant interactions with the treated area. So my first experiment yielded this graph. On the x-axis, we have the treatments, and on the y-axis, we have the percent of leaves left over by the ants, and this kind of tells us how much of a deterrent effect that the treatment had. The oil had the most deterrent effect, pretty obvious to the rest of them. I also ran a two-factor t-test with unequal variances to find our p-values to see if there's any significance between the two. 4 out of 5 of them were significant. The only one not showing any significance was the fresh partial plant. And then my second experiment, I recorded the deterrent effect of it and the recognition of the treatment on the stem. For the control, the fresh partial plant and the dry partial plant, there was no recognition and no deterrent effect. They ran up and down the stem with ease. The dry partial plant, the fresh partial plant had no deterrence, but they did recognize it, the treated area, and on the dry full plant, they sometimes were deterred, and they definitely did recognize that there was a treated area. For the oil, both had a major effect. So the differences between dry and fresh treatments I found to be pretty interesting. I again ran the same t-test and found a p-value of 0.01 for the fresh and dry partial plant differences, and a p-value of 0.05 for the full, and both of them favored the dry solutions. Those were more effective than the fresh ones. So my observations and effectiveness, there was no deterrent effectiveness really observed for any of the partial plant mixtures, and for the full plant mixtures, there was little deterrent effect. Although there was significance, there wasn't enough to maybe keep ants from eating more than half of your orange tree. And the fresh and dry, there was dry leaves had definitely more of an effect, because drying leaves, when making teas, it concentrates the compounds in the leaves and makes them more available when they're steeped. Yes. So plant tolerance to herbivory kind of fluctuates. It depends on the species and the location, the health of the soil and the environment, and their age and their size. And fruit production for trees is usually affected with more than 50% herbivory. So those treatments that didn't have more than a 50% deterrence rate probably wouldn't be the best options, even though they had statistical difference from their controls. So other uses for the castor bean plant could be in ferments, fertilizers, compost, and mulch. The breakdown of these plants in organic ways allows the ricin deterrent properties to be exposed and expressed in the soil. So, it's also a little bit of a less dangerous way to deal with castor bean, as some people really don't want to touch it or be in contact with some dangerous compounds such as ricin. And there's definitely some limitations to my study. The ants showed to have some bias. Different trails had different biases on if they wanted to pick up leaves or if they didn't. And there's a bit of a smaller sample size. So for future studies, although it did pose a lot of new questions. For future studies, I was thinking about different compositions and concentrations of the castor bean, different oil treatments, because the oil really seemed to work. There was a lot of cleaning activity and other behaviors that happened that were not exhibited in the solution treatments that I had made myself. Castor bean effects on other insects, and also how the castor bean affects the fungi in the leaf's nest as well. I'd like to thank Emmy, my primary advisor, and Naomi, my secondary advisor, as well as Eliza Marto, Benito, Gwendon, and Mauricio Liton for opening up their farms to me and letting me study there, as well as all of my peers, the Monteverde Institute, and yeah, everyone, and my homestay family, everyone that made my life really fun and awesome. So thank you. Ricin is dangerous because when it enters the cell, it when it enters the cell, it can block protein production. So, too much is ingested in humans, it can be lethal or fatal, and can cause like stomach aches and headaches and other negative effects. So, yeah, just the compound is not dealt with correctly. It can just be negative to cells' protein production. I think so, but they don't necessarily eat the plants. So I wasn't necessarily looking at the direct effects it had on them, but just the deterrent effects to see if it just made them go away rather than killing them all. Yeah. Sophia?
In the graphs before where you did recognition, yeah, that one. So for the fresh partial and dry partial, there was no deterrence and no recognition, but the herbivory was still lower, correct?
For the fresh partial and the dry partial?
Yeah.
Yeah, there was still less herbivory, and in here you can see the dry partial and the fresh partial. They're kind of the same, but here there's this is the amount of leaves that they left, so there was more herbivory. Does that make sense?
Yeah.
Okay. Yeah. So when there was no recognition and no deterrence, it just didn't really work out.
Thank you so much.
Sorry. Thank you. Thank you. I don't know. Oh. For you.
Does it go here?
There, forward, backward. This is a laser pointer. I don't know if this is what
All right, our next presenter is Nick Bennett, who's going to teach us about potential antibacterial properties of fungi here in Monteverde. Thank you, Nick.
Hi, I'm Nick. I studied the antibacterial properties of the fungi in the Monteverde cloud forest. I asked the question, are there antibacterial fungi in the Monteverde cloud forest? This is important to look into because antibiotic resistance has been on the rise, especially with bacteria like E. coli and Staphylococcus that I studied. These are leading to a rise in healthcare-associated infections, which can lead to mortality or, in better cases, just longer hospital stays. This usually happens because of equipment that wasn't sanitized properly, which could be because the things we use to sanitize them, the bacteria were immune to. And so unless you just douse it in bleach for a few hours, then it's just gonna survive whatever you throw at it. Fungi have been used as our earliest antibiotics, with penicillin coming from the Penicillium mold. We've had this since the 1940s, and in the early 1900s, we were quickly developing new antibiotics, which allowed us to outpace the evolution of bacteria. But now that we've slowed down and have been overprescribing the current antibiotics that we've been using, the bacteria can easily evolve to beat it. So if we can make novel antibiotics off of new fungi, we might be able to outpace the bacteria again. So I did this by first scouting out fungi in the forest. I did this about a week before I collected them, and then a few days before I would get my bacteria, I went out, collected them, brought them back, and dried them. I got my bacteria later than I thought, so they were dried for a lot longer than I expected, which was good, actually. Then once I got the bacteria, I got the fungi that were dried, cut them up, and then crushed them with mortar and pestle. They didn't really become powder like I wanted them to because fungi are kind of squishy. So they just kind of ignored all the force I put into it. But you can see this one got pretty messed up. It's a little blurry, but then I put them in 10 times their water, 20 times for ones that were like really small and didn't have that much mass. So 10 times their mass in water. And I put them in the fridge overnight, stirred them, left them there. Then the next morning, I pulled them out and I stirred them again, dipped filter discs into them for a Kirby-Bauer disc test. And then I made agar plates with Mueller-Hinton agar and plated diluted E. coli and Staphylococcus bacteria on them. It was 1 bacteria per plate except for this plate because it would have been an odd number and I wanted to do a blind test, so I needed it to be an even number. So I did 4 filter discs per plate, 2 discs per sample per bacteria, so 92 discs. And then I incubated them for 36 hours at 37°C. Then the next day, or not the next day, after 36 hours, I pulled them out and I took pictures and measured any zones of inhibition that I found. Zones of inhibition are clear areas you can see around the discs that don't have any bacteria growth, which you'll see in a bit. As you can see, so minuses mean they didn't have any activity. As you can see, there's a lot of minuses. Only the control had a zone of inhibition, which does show that my extracts did permeate through the agar. So I know that wasn't the issue. So this here is the control. It has a really clear zone of inhibition around it. As you can see with some of these plates, actually all of them, the bacteria didn't fully grow across the plate. I think that might be because some of the bacteria died in transit, especially with Staphylococcus, which is the one in the middle, which only grew around the areas I streaked it. The E. coli did a bit better, but didn't really fully cover the plate. So I can't definitively say that that's the reason, or that I didn't get any zones of inhibition, because you can see that there's areas without growth, but that might just be because the bacteria never reached it. You can also see here that there was something growing out of the filter discs, especially on the Staphylococcus. I believe that it's either the fungi survived somehow, the drying process, and then being incubated for 36 hours, and was able to grow, or the filter discs were contaminated. But they have distinct morphologies on the unknown colonies, so I think it might be the fungi, because the spores might have survived. So, as I said, nothing showed any activity. This might just be because I didn't choose anything that actually produced antibacterial compounds. I looked at several studies afterwards that showed that some of the genera that I tested did have antibacterial properties, but not all of their species did. So I probably just chose the ones that didn't have those properties. These are some of the ones that were expected to. This one is also maybe edible. I'm not going to test it, but I've been told it might be edible. Another study in Kenya also looked into similar genre that I did, and they used much more advanced methods than I did. They had access to more advanced equipment like a centrifuge, an incubator shaker, and other things. I believe that might have been one of the reasons they got better results than I did, is because they extract they used was able to permeate better throughout the solvent. And for the solvent, they also used chloroform, ethanol, and hot water, and I just used distilled water. So it's possible that that was able to pull the compounds out of the fungi. It's also possible that some of the compounds were in the substrate that it was on, because I didn't collect any of the substrate, I just collected the fruiting bodies. So they might have just been in the substrate, just around it, which would make some sense because that's where the bacteria that they'd be competing with would be coming from. And it's also where the mycelium underneath the fruiting body would be. So that's what would be competing the most with it. A big fruiting body like this probably doesn't have to worry about bacteria too much. Special thanks to Naomi, my primary advisor, who is probably the only reason this succeeded at all, because she helped me with everything. Thanks to Emmy, who helped me with my blind test, which, I mean, you could kind of tell what the control was already when you looked at it, but it wasn't a good idea. Thanks to Trinity and Noel for helping me bushwhack for like 3 hours in the forest looking for fungi and getting bit by mosquitoes. Thanks to Jose Murillo, who helped me identify the fungi, and to the Monteverde Institute and La Estación Biológica Monteverde for letting me use their facilities and their land. Questions?
There's time for questions. Happened to the bacteria that did not grow correctly?
So in transit, it got defrosted, and for about 3 hours, it was unrefrigerated. So I have the feeling that a lot of it died, which is why you can see with the Staphylococcus had like nothing other than the streaks and some contamination. But the E. coli did better. I believe that if I let that incubate longer, it would have spread out across the plate. The Staphylococcus probably died though.
Yeah, your point about the mushroom versus the mycelium is really important, because the mycelium is more like the plant, if you want to call it that, and the mushroom is more like the fruit.
Mm-hmm.
And so they have different properties. And most of the bacterial properties are in the mycelium, because the mushroom is meant to be a temporary structure, just to distribute spores. As a matter of fact, some mushrooms, of course, are highly edible because that helps distribute spores. They're not trying to keep animals away. You know? So it's important probably in the future to take a look at mycelium versus mushrooms, because they're different things.
That is definitely something I want to look into, because once I had collected the fruiting bodies and gotten my results, I started having the feeling that it probably wouldn't be the flower, for lack of a better term, that produces those substances. It would probably be the plant itself, like you said, because that's what would actually be in danger from bacteria. Owen.
The unknown growth, did you get a chance to look at it through a microscope?
No, and I'm very curious what I would see if I did. I was kind of constrained on time. It was the last day, and I had about like 3 hours left of lab time. So, but I do really want to know what it was. In the back. Oh.
Into selecting the fungi that you took, or was it just any of the fungi you found?
So I found ones that were large enough to take as a sample. Some that I took were really small and they disintegrated while drying. I tried to avoid those kinds, but it was usually just what I found.
Thank you so much.
Thanks. Oh, might want that.
Gordon Stanley, who's also going to teach us about antibacterial properties, but in this case of Gavilana. Thank you, Gordon.
Thank you, Emmy. Hi, everybody. My name is Gordon. I am a biology major at UCLA, and I'm here to share a little bit about the bacterial growth inhibition effects of Gavilana. So, starting with my introduction here, a little bit about Gavilana. Latin name is Neuralena lobata. It also has a multitude of other names, which kind of speaks to how widely used it is. It's a herbaceous shrubby aster distributed throughout the tropics, so it's found from central Mexico all the way down to Bolivia. Commonly found next to roadsides, in home gardens, places where humans have been, especially because humans like to plant it. So who uses it and how? It's commonly used by indigenous groups, and it's very also common in folk medicine because it boasts these medicinal properties. It's got pretty well documented antibiotic, anti-parasitic, and antifungal properties. And it's commonly prepared in teas, powders, tinctures, and juices. All of these preparations vary regionally with different indigenous groups and different folk medicine techniques. Although there are some overlaps, the tea, for example, is probably the most common mode of preparation. And that comes in two forms. Basically, leaves steeped in hot water and root material steeped in hot water. So, why I think it's important that we study this plant is with the advent of Western medicine, we're experiencing a widespread loss of indigenous and folk knowledge in terms of orally passed down medicinal traditions. And given that Gavilana has pretty well documented antibacterial and medicinal effects, the plant could serve as a viable bridge to kind of help preserve tradition while also incorporate pre-existing knowledge into Western medicine. So that's kind of the reason in which I'm looking into it. Now, my research question, sort of getting at that point, is do any of the historical folk medicinal preparations of Neuralena lobata inhibit E. coli, Bacillus subtilis, or Staphylococcus aureus growth in vitro? So I was doing this in the lab setting. So my materials and methods were adapted from Kirby-Bauer disc diffusion procedure, which is a pretty commonly used pharmacological procedure in which various solutions or drugs are used and filter paper discs are dipped into them and then placed over plated bacteria to determine the drug's efficacy in reducing bacterial growth or inhibiting bacterial growth in vitro. So these zones of inhibition here, which are the circles around the filter paper discs, are kind of, by measuring them, you can gauge how successful your experimental solutions are in exhibiting inhibition. So my first step in my methods was to collect plant material. I was very fortunate in that there's two growing literally in the backyard. So I just had to walk outside and collect some leaf and root material. Next, I prepared my various plant solutions. So all of these are documented pre-existing historical uses of the plant and how that it's actually been prepared. With the only exception being in the leaf infusions, I added varying kind of numbers of leaves. And I say numbers like this because I found an average weight of all the leaves and then added the corresponding amount of grams to create like a 1 leaf solution, a 5, and a 10. I also I submerged 10 grams of root material to make a root infusion. I pulverized a leaf with the mortar and pestle and extracted its juice for the leaf extract. And then I placed 5 leaves in the dry box until they were completely dry and then ground them up into a powder. I included 2 controls in my study. My positive control was an antibiotic solution with the active ingredient tobramycin. And then my negative control was distilled water. These were obviously used to get the most and least possible inhibition, so I had something to compare my experimental trials to. My next step was to prepare my agar plates. You make the mixture, you pour it into sterilized plates, you allow it to cool. Next, I plated my bacteria and plants. Oh, I just forgot something very important. Let me go back too. Thank you, Nick, for helping me implement a blind labeling system in my study. So after the point of preparing all my solutions, I had no clue in measuring them which strain was which strain and which solution was which solution. So this helped me avoid any kind of bias in measuring my zones of inhibition. All right, back to bacteria. After I plated my different bacterial strains, I placed the filtered papers that had been submerged and in each of the different experimental solutions and placed it over the bacterium. I did so for 2 18-hour stints, totaling 36 hours of inhibition. The only reason I took them out was to photograph their progress halfway through. And then after the 36 hours had elapsed, I put all the pictures that I took of my trials in ImageJ, which is software on my laptop, and I set the scale of the image, and then using the software, I was able to trace the perimeters of all the zones of inhibition to get an area reading for all of the zones in millimeters squared. So getting into some of these results. The only experimental trials that exhibited any zones of inhibition after 36 hours were the root infusions. And it only did so against B. subtilis and S. aureus. It did not exhibit any inhibition against E. coli strains. The leaf infusions, the leaf powder, and the leaf extract all did not exhibit any zones of inhibition. And then here I've got an example of one of my trials with the root infusion against B. subtilis. You can see here that although they're smaller than the previous slide, this is the antibiotic. There are still pronounced zones of inhibition in 3 out of these 4 trials, with the exception being this one. This is my one and only graph. It's a bar chart of the various inhibition areas in millimeters squared. So that's the y-axis here. And the x-axis is separated on different bacterial strains, with the blue representing the root infusions. So notably against B. subtilis, the root infusion exhibited statistically significant inhibition when compared with distilled water. It did not exhibit any statistically significant inhibition against distilled water in either other bacterial strain, and also quite obviously, the antibiotic outperformed everything in every strain. So getting into the discussion, why might this matter? The roots contain chemical derivatives of a compound called thymol, which is pretty commonly found in a lot of over-the-counter oral antiseptics and mouthwashes, most notably probably Listerine is the active ingredient is thymol. The exact derivative has a very long name, which I will not get at, but something about this molecule specifically, which is found in the roots of Gavillana, is that it passes a whole trove of filters and rules used by pharmacologists to decide a compound's likeliness of becoming a good drug or making a good drug. And so this one notably passes 3 of the most popular, Ghost's filter, the Lipinski rule of 5, and the Egan rule. And each one of these kind of delves into different chemical properties each compound maintains, like its molecular weight and things of that nature. So hence, this compound that's found in Gavillana roots has very substantial medicinal and pharmacological applications. Probably the most unexpected result was that the leaf infusions did not exhibit any zones of inhibition. Prior research has indicated that although it would not have been as significant as the antibiotics, there would be some inhibition. And notably, a lit review revealed that the leaves contain alkaloids, which the plant has developed as a chemical defense against herbivory to, that have antiparasitic and antibacterial capacity. So for those reasons, I was expecting the leaf infusion to be effective. Obviously, it wasn't. Why might it not have been? It's notable that the plants were in their early flowering stages at the time that I collected these leaves. And it's commonly studied in flowering plants that compounds move around to different areas of the plants that most need them. So during this stage, it's possible that the compounds could actually have been more present in the flowers of the plant and not in the leaves. And thus, when I made the infusions, it exhibited less inhibition as a result. So I'd be interested in studying them outside of their normal flowering times. To conclude, the Gavialana root infusion exhibited the only bacterial inhibition against B. subtilis. Despite this, Gavialana shows great promise in pharmacology and in medicine, and definitely my findings beget further study on this plant. I would like to thank Naomi Solano and Federico as my two advisors for their support and guidance throughout. I'd also like to thank Eladio, Esteban, and Jorge for their knowledge on the plant. I'd like to thank as well the UC EAP faculty and the Monteverde Institute faculty for providing such a great work environment. And you all, because you're very hard to follow in presentations. You guys all killed it. So thank you.
Is there, do you know anything in the traditional use of Gavialana root infusion specifically that could have independently suggested that it's more effective in situations that are very severe?
So not exactly in bacterial inhibition. The review I did initially suggested that the root infusions are used as a blood cleanser. And so they're not necessarily for something that you would expect antibiotics to be especially helpful in, but obviously I think this begets further study into their use as such. Sarah?
How would you go about doing future studies?
So there's a lot of moving parts in this experiment, but I think the most effective one would try to combine chemical analysis with all of the 3 different steps. So running specific chemical analyses on the multiple different parts of the plants, on each of the different solutions and preparations of the plant, and then looking at the resulting bacterial inhibition to try to kind of isolate what compounds are producing what results. Anyone else? Trinity.
So I'm wondering if this study inspires you to want to use more natural remedies or natural medicines in your own practice in the future.
Yeah, absolutely. I think it's imperative that we kind of preserve this knowledge and traditional uses of plants, because Nick got at this quite a bit in his presentation. Bacteria is growing ever more resistant to our antibiotics, and looking in other places is kind of kicking the can down the road, but I think a more effective solution will be found if people continue to look in places like this.
Thank you, Gordon.
Thank you.
Actually, would you mind turning that on?
Of course.
So this is going to be super fast. Just to remind you that we met 10 weeks ago with the Cubs. It's very hard to see the future as much as I like to think that I can see the future. And these projects really are not scripted ahead of time. We have no idea really how they're going to turn out. And I was fantastically impressed by the effort that you put into them and the professionalism of your presentation. And I kept thinking, like, who is teaching these guys? But obviously, I sincerely thank my coworkers and everyone else who helped make the project grow. For this particular event, I would especially like to thank Joyce for translating. To the Monteverde Institute for hosting this, and for some of our really amazing audience members. So Silvio, who is a cousin of Naomi, and your name is Danielle? Michael. Sorry. And Kathy, and Amelia's parents, Andre and Noel, and I don't know your name yet.
Pia, her sister.
Pia. Fully engaged, really impressive, fantastic. like to say quickly that I'd like to thank you, the students, for not giving up. I know that some of you, maybe all of you, did some sweating. Some of you shed some tears. Some of you led a little bit in your projects. So all the sweat and tears paid off. It's really, really a super day. I can't remember symposium in recent history, so that would be the last 10 years or more, where everybody has just done such a wonderful job. I thought it would be really good for you and therapeutic to walk to the stage. I only insisted and said no. There will be a ride for you, yes. And also, thanks to you for your patience and for people who are troubleshooting all day long with our technological challenges. That just comes with territory. That just came in the zero bar. You're done, Frank. You're done. I'm done. Thank you.